A single bacterium sits on the surface of a plastic bottle, invisible to the human eye. Under the electron microscope, it appears unremarkable, a rod-shaped microorganism measuring roughly 1 micrometer in length. But this bacterium is performing an act that should be impossible. It is eating plastic. Not just clinging to the surface or forming a biofilm, but actually breaking down the polymer chains of polyethylene terephthalate and consuming them as food, converting synthetic molecules into energy and carbon for growth. This is Ideonella sacchaensis, and its discovery in 2016 marked the beginning of a revolution that could reshape humanity's relationship with waste. The story begins outside a recycling facility in Sakai City, Japan, where researchers Kohei Oda and Kenji Miyamoto were hunting for microorganisms with unusual abilities. The site was contaminated with decades of plastic residue from bottle processing operations. The sediment samples teemed with microbial life, a consortium of bacteria, protozoa, and yeast-like cells that had adapted to this synthetic environment. When the researchers isolated individual species and tested them against low-crystallinity PET film, one organism stood out. It could colonize and degrade a film just 0.2 millimeters thick in approximately 6 weeks, a rate that seemed impossibly fast compared to the natural breakdown of plastic in the environment. The scientific community reacted with cautious excitement. For over 70 years, humanity has been producing plastic at an accelerating rate, creating materials designed specifically to resist degradation. That durability, the very property that makes plastic so useful for packaging and consumer goods, has become an environmental catastrophe. Global plastic production reached 460 million tons annually by 2025, with projections showing it could hit 590 million metric tons by 2050. Only 9% of this plastic ever gets recycled. The rest accumulates in landfills, incinerators, and increasingly in natural ecosystems where it persists for centuries. Ocean bears the heaviest burden. Between 75 and 199 million tons of plastic waste currently choke marine environments, with an additional 33 billion pounds entering the water every year. Scientists estimate that 14 million tons of plastic sink into the ocean annually, settling on the seafloor where detection becomes nearly impossible. At current rates, the total mass of plastic in the ocean will exceed the combined mass of all fish by 2050. Marine life suffers catastrophically from this pollution. Over 914 megafaunal species experience impacts from entanglement and ingestion, with more than 100 endangered species affected. The Great Pacific Garbage Patch, a swirling accumulation of debris twice the size of Texas, now contains 180 times more plastic than biomass, suggesting plastic has become the primary material that organisms encounter in that region. Polyethylene terephthalate represents a particularly challenging component of this crisis. PET makes up a substantial portion of single-use packaging, especially beverage bottles, with global consumption exceeding 20 million tons annually and growing at 3.6% per year. The material offers remarkable properties for packaging, maintaining carbonation, protecting flavor integrity, and resisting contamination during storage and transport. These same properties make it extraordinarily persistent in the environment. PET's molecular structure consists of long chains of repeating units, each containing a benzene ring connected to ester linkages. This configuration is not found in nature. No organism evolved to break it down because it did not exist until humans synthesized it in the mid-20th century. That is what made the discovery of Ideonella sacchaensis so revolutionary. This bacterium had somehow evolved the enzymatic machinery necessary to hydrolyze PET, cleaving those synthetic bonds and releasing the building blocks for metabolism. Further investigation revealed the organism secretes 2 specific enzymes that work in sequence. The first, designated PTase, attacks the polymer chains on the plastic surface, breaking them down into an intermediate compound called mono-2-hydroxyethyl terephthalate, abbreviated MHET. The second enzyme, MHETase, completes the degradation by splitting MHET into its 2 constituent molecules, terephthalic acid and ethylene glycol. These compounds can then enter standard metabolic pathways, allowing the bacterium to derive both carbon and energy from what humans consider waste. The enzymatic mechanism operates at the molecular level with remarkable precision. PTase belongs to a family of proteins called cutinases, which naturally degrade cutin, a waxy polyester coating on plant leaves. But pTase has evolved unusual features. Its active site, the pocket where the chemical reaction occurs, is more open than typical cutinases, allowing it to accommodate the rigid structure of synthetic polymers rather than just natural plant materials. This structural adaptation suggests recent evolutionary pressure, likely driven by decades of PET contamination in the environment where Ideonella sacchaensis was found. The enzyme follows the canonical serine hydrolase mechanism, using a catalytic triad of 3 amino acids, serine 160, histidine 237, and aspartate 206, positioned precisely to activate water molecules and attack the ester bonds in PET. X-ray crystallography, performed at one of the most advanced beamlines in the world, revealed the three-dimensional atomic structure of pTase in extraordinary detail. Professor John McGeehan at the University of Portsmouth led the structural analysis, achieving very high resolution that allowed researchers to see the inner workings of this biological catalyst. The visualization provided blueprints for understanding not just how the enzyme functions, but how it might be improved through genetic engineering. Computational modeling by teams at the University of South Florida and the University of Campinas in Brazil confirmed that pTase resembles cutinases but possesses modifications that enhance its ability to bind man-made polymers. These differences indicated that pTase likely evolved specifically in response to PET contamination in the environment. MHEtase, the second enzyme in the degradation pathway, adds another layer of sophistication to this biological system. Structural studies revealed that MHEtase shares a similar core domain with pTase, but includes an additional lid structure that is crucial for processing MHET molecules. The enzyme operates at a catalytic rate of 27.6 reactions per second, significantly faster than related enzymes from other bacterial species. When pTase and MHEtase work together, they exhibit remarkable synergy. Even at the lowest concentrations tested, the presence of MHEtase dramatically increases the release of monomers from solid PET films compared to pTase working alone. This cooperative effect occurs because MHEtase removes the intermediate product, preventing it from accumulating and inhibiting further pTase activity. The microbial consortium isolated from the Sarkai recycling facility demonstrated an even more impressive capability. When Ideonella sacchaensis breaks down PET and releases MHET, terephthalic acid, and ethylene glycol into the surrounding environment, other members of the microbial community can utilize these compounds. Studies showed that the entire consortium could mineralize 75% of the degraded PET into carbon dioxide. This complete metabolic cycle represents true biodegradation, not just physical breakdown into smaller pieces. The plastic is being converted back into simple molecules that re-enter biogeochemical cycles. However, the wild-type bacterium has significant limitations. Under laboratory conditions, Ideonella sacchaensis can degrade thin films of low crystallinity PET in about 6 weeks, but high crystallinity PET, the harder plastic used in most commercial products, degrades approximately 30 times slower, requiring 180 weeks or more than 3 years. The enzyme also functions optimally at only 40 degrees Celsius, relatively cool compared to industrial processes. Temperature sensitivity represents a major constraint because higher temperatures typically accelerate chemical reactions, and many plastic recycling applications occur at elevated temperatures. The enzyme's activity decreases outside a relatively narrow pH range of 7.0 to 9.0, limiting its effectiveness in acidic or highly alkaline environments. These constraints sparked intense research into engineering improved versions of the enzymes. Scientists recognized that while nature had accomplished something remarkable by evolving plastic-degrading capability in just a few decades of environmental exposure, human ingenuity, combined with modern biotechnology tools, could potentially accelerate that evolutionary process by orders of magnitude. The question became, could researchers modify these enzymes to work faster, tolerate harsher conditions, and degrade a wider range of plastics? And more broadly, could humanity develop an entire toolkit of engineered microorganisms capable of consuming the mountains of plastic waste accumulating in landfills, oceans, and urban centers around the world? 3 ambitious projects emerged to test these possibilities in real-world conditions. The first is an experimental landfill where engineered bacteria are being released to degrade plastic buried in soil for over 3 decades. The second is an ocean-based initiative attempting to enhance mechanical cleanup of the Great Pacific Garbage Patch by treating recovered plastic with plastic-eating enzymes and microbes. The third is an urban pilot program integrating biological waste degradation directly into city infrastructure, creating what researchers call living recycling systems. Each testbed faces unique challenges and raises profound questions about the wisdom of releasing engineered organisms into the environment at scale. The potential benefits are staggering. If biological plastic degradation can be optimized and deployed safely, it could help address one of the most pressing environmental crises of the 21st century. Instead of plastic persisting for hundreds or thousands of years, it could be broken down in weeks or months, returning to the carbon cycle and eliminating the accumulation of synthetic debris. The technology could transform waste management, making recycling more efficient and less energy-intensive than current mechanical and chemical methods. It might clean contaminated sites that are currently impossible to remediate using conventional approaches. But the risks are equally profound. Engineered microorganisms released into the environment cannot be easily recalled. Bacteria reproduce and evolve, potentially spreading beyond intended areas and acquiring new traits through mutation or horizontal gene transfer with wild populations. What if plastic-eating enzymes begin degrading plastic that humans still need, destroying infrastructure, vehicles, or medical equipment? What if the degradation byproducts prove toxic, creating new forms of pollution even as they eliminate old ones? What if the modified bacteria outcompete native species, disrupting ecosystems in unpredictable ways? These concerns have haunted environmental microbiology since the first genetically modified organisms were considered for environmental release in the 1980s. The debate crystallizes into a single question that defines the central tension of this entire endeavor. Can humanity deploy engineered organisms globally without triggering new ecological disasters? The answer will determine whether biological plastic degradation becomes a transformative solution or a cautionary tale about the unintended consequences of environmental engineering. That answer depends on the results emerging from the 3 testbeds, where scientists, ecologists, and engineers are now conducting the most comprehensive field trials ever attempted with synthetic biology. This is the story of those experiments. It is a story of scientific ingenuity pushing the boundaries of what is possible, of natural systems co-opted and enhanced to serve human purposes, of careful measurement and monitoring as modified organisms interact with complex ecosystems. It is also a story of risk and uncertainty, of ethical debates about humanity's role as planetary engineers, and of the difficult choices facing a species that must deal with the consequences of its own success. The invisible revolution has begun, one bacterium at a time, and the outcome will shape the future of life on this planet. Chapter 2: The Molecular Scissors. Inside a single molecule of polyethylene terephthalate, the atoms are arranged in a precise 3-dimensional structure that gives the material its remarkable properties. Carbon, hydrogen, and oxygen form repeating units connected by ester bonds, chemical linkages that join organic acid groups to alcohol groups. The benzene rings provide rigidity and strength, while the ester connections allow just enough flexibility for the polymer chains to pack together efficiently. When millions of these chains align in parallel, they create the crystalline regions that make PET bottles durable enough to withstand drops from significant heights, resist punctures during shipping, and maintain their integrity for years sitting on store shelves. This molecular architecture is the reason plastic persists so stubbornly in the environment. To understand how enzymes can dismantle this structure, we must zoom in even further to the active site of PTase, the catalytic pocket that performs the chemical surgery. The enzyme is a protein, a long chain of amino acids folded into a complex three-dimensional shape. That shape is everything. Within the folded structure, three specific amino acids cluster together at positions numbered 160, 206, and 237. These form the catalytic triad, a molecular machine that has been refined by hundreds of millions of years of evolution in related enzymes, then adapted for PET degradation in just a few decades. The serine at position 160 serves as the nucleophile, the reactive site that initiates the attack on the ester bond. In its normal state, serine has a hydroxyl group, an oxygen bonded to a hydrogen. But within the active site environment, the nearby histidine at position 237 acts as a base, accepting the proton from serine's hydroxyl group. This leaves the oxygen with a negative charge, making it highly reactive. The aspartate at position 206 stabilizes the histidine, ensuring it maintains the correct orientation and charge state to perform its role. Together, these three residues form an elegant system for activating water molecules and using them to cleave chemical bonds. When a PET chain approaches the active site, specific interactions guide it into position. The surface of PTase features a binding groove lined with hydrophobic amino acids, residues that repel water but attract the nonpolar regions of the plastic polymer. This hydrophobic character helps the enzyme grip the plastic surface and correctly orient the ester bond for attack. The active site architecture of PTase is notably more open than that of typical cutenases, providing enough space to accommodate the bulky structure of synthetic polymers. This openness distinguishes PTase from related enzymes like lipases, which have lid structures covering their active sites. Those lids must move aside before the substrate can enter, a process called interfacial activation. PTase lacks such a lid, allowing direct access to the catalytic machinery. The catalytic mechanism unfolds in a precise sequence of steps. First, the activated serine oxygen attacks the carbonyl-carbon of the ester bond in PET. This carbon is slightly positive due to the electron-withdrawing effect of the adjacent oxygen, making it vulnerable to nucleophilic attack. When the serine oxygen forms a new bond with this carbon, the double bond between carbon and oxygen temporarily breaks, creating a tetrahedral intermediate, a transient state where the carbon is bonded to 4 groups instead of 3. This intermediate is highly unstable, lasting only a few picoseconds. An oxyanion hole, a structural feature formed by the backbone atoms of the protein, stabilizes the negative charge that develops on the oxygen during this transition. Without this stabilization, the reaction would be far too slow to be biologically useful. The tetrahedral intermediate then collapses, but instead of returning to the original ester bond, it breaks in a different direction. The serine now finds itself covalently bonded to one half of the PET chain, forming an acyl enzyme intermediate. The other half of the chain, containing the 2-hydroxyethyl group, is released as the first product. This is MHET mono-2-hydroxyethyl terephthalate, the intermediate compound that gives MHETase its name. At this point, the enzyme is still holding onto part of the PET chain, bound through the serine residue. To complete the catalytic cycle and return to its active state, the enzyme must release this bound fragment. A water molecule enters the active site, activated by the same histidine and aspartate residues. The water's oxygen attacks the carbon of the acyl enzyme bond, forming another tetrahedral intermediate. This collapses in a direction that frees the serine and releases the terephthalate portion of the molecule. The enzyme is now reset, ready to attack another ester bond. This entire sequence, from initial binding through both tetrahedral intermediates to product release, follows the canonical two-step serine hydrolase mechanism that has been conserved across thousands of enzyme families. Computational studies using quantum mechanics and molecular mechanics simulations have mapped the energy landscape of this reaction with extraordinary precision. Researchers calculated that the activation energy, the barrier that must be overcome for the reaction to proceed, is approximately 19.35 kilocalories per mole for the rate-limiting step. This value determines how fast the enzyme can work. Lower activation energies mean faster reactions. The simulations revealed that both tetrahedral intermediates are extremely transient, with barriers of only about 2 kilocalories per mole separating them from the surrounding states. This translates to half-lives of roughly 3 picoseconds at room temperature, far too brief to be directly observed experimentally, but crucial for understanding the full catalytic mechanism. The structure of PTAs has been determined at 1.6 angstrom resolution, a level of detail that reveals the positions of individual atoms. This resolution allowed researchers to see not just the catalytic triad, but also the surrounding architecture that influences enzyme performance. The binding groove features aromatic amino acids that can form pi-stacking interactions with the benzene rings in PET, helping to position the substrate correctly. Tryptophan and phenylalanine residues create a hydrophobic environment that excludes water from the active site until the precise moment when water is needed for the hydrolysis step. The open active site explains why PTAs can accommodate synthetic polymers that would be too large for typical cutinases. MHETase adds a second layer of enzymatic sophistication to the degradation pathway. While PTAs breaks PET into MHET and ethylene glycol, MHET remains a relatively large molecule that most organisms cannot metabolize directly. MHETase completes the depolymerization by hydrolyzing MHET into terephthalic acid and ethylene glycol, both of which can enter standard biochemical pathways. The structure of MHETase reveals a core domain similar to PTAs, confirming their evolutionary relationship, but with a significant addition, a lid domain that caps the active site. This lid is not a barrier, but a gatekeeper. Simulations demonstrate that the lid is essential for MHET hydrolysis, suggesting it plays a role in substrate recognition or product release. Mutant versions of MHETase lacking the lid show dramatically reduced activity. The enzyme's catalytic triad mirrors that of PTAs, with serine, histidine, and aspartate positioned to perform the same chemical dance of nucleophilic attack, tetrahedral intermediate formation, and product release. But the substrate specificity differs. MHETase is optimized for MHET rather than long polymer chains, with a binding pocket shaped to accommodate the smaller molecule. The synergy between PTAs and MHETase is crucial for efficient degradation. When both enzymes are present together, the rate of monomer release from solid PET films increases dramatically compared to either enzyme alone. This cooperative effect arises because PTAs produces MHET faster than it can diffuse away from the plastic surface. If MHET accumulates, it can inhibit further PTAs activity by occupying the active site or blocking access for new substrate molecules. MHETase solves this problem by continuously removing MHET, hydrolyzing it into the final products, and keeping the PTAs active sites clear for continued work on the polymer chains. Researchers have quantified this synergy through careful kinetic experiments. When PTAs is applied to amorphous PET film alone, at a loading of 1 milligram of enzyme per gram of plastic, a measurable amount of degradation occurs. But when MHETase is added at any concentration from 0.1 to 1.0 milligrams per gram, the release of terephthalic acid and ethylene glycol increases markedly. The exact magnitude of the increase depends on the ratio of the two enzymes, with optimal performance occurring when both are present at sufficient concentrations to avoid either becoming rate-limiting. This enzyme-limited behavior indicates that under the conditions tested, adding more enzymes would produce even faster degradation, limited only by the physical constraints of enzyme stability and the accessibility of the plastic surface. Bioinformatics analysis provided insights into how these enzymes evolved. PTAs appears to have diverged recently from cutinases, enzymes that naturally degrade cutin, the polyester layer on plant surfaces. The similarity in sequence and structure is unmistakable, yet PTase has acquired mutations that expand the active site and alter substrate specificity. MHETase likely evolved from ferulic acid esterases, enzymes that break ester bonds in plant cell wall components. Two homologous enzymes identified through sequence comparisons also show MHET turnover activity, confirming that this enzymatic capability exists in related proteins. The evolutionary trajectory suggests that PET degradation is not a completely novel biochemical invention, but rather a modification of existing enzymatic machinery to handle synthetic substrates. The temperature dependence of enzyme activity presents both opportunities and challenges. Like most biological molecules, PTase and MHETase have optimal operating temperatures. For the wild-type enzymes, peak activity occurs around 40 degrees Celsius. Below this temperature, molecular motion slows and reaction rates decrease. Above it, thermal energy begins to disrupt the precise three-dimensional structure of the protein. The enzyme unfolds, a process called denaturation, and loses its catalytic ability. For wild-type PTase, significant denaturation occurs above 50 degrees Celsius, severely limiting its use in industrial settings where higher temperatures are often employed to increase reaction rates. This temperature sensitivity reflects the thermodynamic balance between the folded active state of the protein and the unfolded inactive states. Proteins fold into their active conformations because that shape has the lowest free energy under normal cellular conditions. But at higher temperatures, the entropy gained by unfolding overcomes the stabilizing interactions that hold the protein together. The unfolding transition is often steep, with activity dropping precipitously over just a few degrees of temperature increase. For practical applications, especially in waste management systems where ambient temperatures vary seasonally and processing conditions may involve heated water or plastic, this thermal instability is a significant limitation. The pH sensitivity adds another layer of complexity. The ionization states of amino acid side chains depend strongly on pH. At low pH, acidic environments, carboxyl groups remain protonated and neutral, while amino groups become protonated and positively charged. At high pH, alkaline conditions, carboxyl groups lose protons and become negatively charged, while amino groups remain neutral. These charge changes alter the electrostatic interactions that stabilize the protein's structure and modulate the reactivity of the catalytic triad. For PTase, optimal activity occurs in a relatively narrow pH range between 7 and 9, close to neutral or slightly alkaline. Outside this range, the enzyme's performance deteriorates as the active site geometry distorts or the catalytic residues lose their proper charge states. The crystallinity of the plastic substrate also profoundly affects degradation rates. PET exists in two forms: amorphous, where the polymer chains are randomly oriented, and crystalline, where they pack into ordered arrays. Amorphous PET is softer and more accessible to enzymes, allowing PTase to bind more easily and hydrolyze ester bonds efficiently. Crystalline PET is harder and denser, with the polymer chains held together by stronger intermolecular forces. The enzyme struggles to penetrate these ordered regions and access the ester bonds buried within the crystalline structure. Measurements show that high crystallinity PET degrades approximately 30 times slower than low crystallinity PET, a difference that translates to weeks versus years for complete breakdown. These molecular insights reveal both the elegance and the limitations of nature's solution to plastic degradation. The enzymatic machinery is sophisticated, leveraging billions of years of evolutionary refinement in serine hydrolases and adapting it to a completely novel substrate within the brief window of human industrial history. Yet the wild-type enzymes are not efficient enough for large-scale deployment. They work too slowly, require too narrow environmental conditions, and cannot handle the full range of plastics accumulating in the environment. This realization drives the next phase of the revolution: engineering beyond nature. Chapter 3: Engineering Beyond Nature. Natural evolution operates through random mutation and selection over generations, a blind process that produces exquisite solutions to environmental challenges given sufficient time. But humanity does not have millions of years to wait for bacteria to perfect their plastic-eating capabilities. The waste crisis demands faster innovation. Enter synthetic biology, a field that combines principles from engineering, molecular biology, and computer science to deliberately design and modify living organisms. The tools of synthetic biology allow researchers to accelerate evolution, getting it toward desired outcomes in months or years instead of millennia. The revolution began with a technique called directed evolution, pioneered by Francis Arnold, who received the Nobel Prize in Chemistry in 2018 for developing this approach. Directed evolution mimics natural selection in the laboratory, but with human-imposed selection pressures. The process starts with a gene encoding the enzyme of interest, in this case p-tase or MHE-tase. Researchers introduce random mutations into this gene using chemical mutagens, error-prone DNA polymerases, or ultraviolet radiation. These mutations alter the amino acid sequence of the resulting protein, creating a library of thousands or millions of enzyme variants. Each variant is then tested for the desired property, whether faster catalytic activity, higher temperature stability, broader pH tolerance, or improved binding to crystalline PET. The testing occurs in high-throughput screening systems capable of evaluating thousands of variants simultaneously. Variants that perform better than the parent enzyme are selected and used as the starting point for another round of mutation and selection. This iterative process rapidly accumulates beneficial mutations while discarding deleterious ones. After multiple generations, the evolved enzymes can differ substantially from the wild-type versions, possessing dramatically enhanced capabilities. In the case of p-tase, directed evolution campaigns have produced variants with significantly improved performance. One engineered version created by researchers at the University of Portsmouth and the University of South Florida incorporated mutations that alter the active site geometry and improve substrate binding. When computational models predicted that making p-tase more similar to ancestral cutenases might enhance stability, researchers tested those predictions by introducing specific mutations. Paradoxically, some mutations that made the enzyme more cutenase-like actually improved PET degradation, suggesting that the evolutionary trajectory had not yet reached the optimal configuration. Another breakthrough came from engineering thermostability, the ability to maintain structure and function at elevated temperatures. Researchers identified mutations in regions of the protein far from the active site that nonetheless influence overall stability. These mutations often involve substituting amino acids with different properties, for example, replacing a flexible glycine with a more rigid proline or introducing salt bridges that lock distant parts of the protein together. The result is an enzyme that retains activity at 60 or even 70 degrees Celsius, temperatures that would irreversibly denature the wild-type version. This thermal stability is not just about surviving heat, it also speeds up the reaction. Higher temperatures increase molecular motion and collision frequency, accelerating the rate at which enzyme and substrate interact. An enzyme that works at 60 degrees can potentially degrade plastic several times faster than one limited to 40 degrees. CRISPR-Cas9, the genome editing technology that transformed biological research in the 2010s, has opened new avenues for protein engineering. While CRISPR is famous for cutting and pasting DNA sequences, researchers have repurposed it for directed evolution in living cells. The Cas9 protein can be modified to introduce mutations at specific locations in the genome rather than making random changes throughout. This targeted approach allows scientists to focus mutagenesis on regions of the enzyme most likely to influence desired properties, increasing the efficiency of the screening process. In mammalian cells, researchers have demonstrated that CRISPR-based directed evolution can optimize proteins for very specific cellular environments, such as the acidic interior of lysosomes. The same principles apply to engineering enzymes for extreme conditions encountered in industrial waste streams. One particularly elegant application of CRISPR involves creating libraries of enzyme variants directly in bacterial cells expressing the enzymes. The CRISPR system introduces double-strand breaks in the gene encoding pTase or MHE-tase. Cellular repair mechanisms fix these breaks, but often introduce errors in the process, generating mutations. By controlling where the breaks occur, researchers can bias mutation toward functionally important regions of the protein. High-throughput screening then identifies the best-performing variants from among millions of cells, each expressing a slightly different enzyme version. This in vivo directed evolution combines the power of CRISPR's precision with the efficiency of biological production, potentially generating optimized enzymes faster than traditional methods. Rational design represents an alternative to random mutagenesis. Instead of generating millions of random variants and screening for improved function, rational design uses structural information and computational modeling to predict which specific mutations will enhance performance. The three-dimensional structures of pTase and MHE-tase determined by X-ray crystallography provide detailed blueprints. Computational chemists can model how different amino acid substitutions would affect the active site geometry, substrate binding, or protein stability. These predictions guide the creation of a small number of carefully designed variants, which are then tested experimentally. The advantage of rational design is efficiency. Rather than screening thousands of random mutants, researchers test only a handful of predicted improvements. The disadvantage is that predictions can be wrong. Protein behavior is complex, and even sophisticated computational models cannot capture all the subtle interactions that determine function. In practice, rational design and directed evolution complement each other. Rational design identifies promising mutations based on structural understanding, while directed evolution explores sequence space more broadly and discovers unexpected solutions that computational models might miss. Researchers have applied both approaches to develop enzyme cocktails that combine multiple enzymes for synergistic effects. The natural pTase-MHE-tase system already demonstrates synergy, but engineered versions take this further. Scientists at the University of Portsmouth created fusion proteins, single polypeptide chains containing both pTase and MHE-tase domains connected by flexible linkers. These chimeric enzymes showed improved performance compared to the separate enzymes mixed together. The physical linkage ensures that MHE-tase is always present near pTase, immediately processing the MHE-T intermediate without requiring diffusion through the solution. Different linker lengths produce different degrees of improvement, suggesting that the spatial relationship between the two catalytic domains influences overall efficiency. Beyond pTase and MHE-tase, researchers have catalogued an expanding repertoire of plastic-degrading enzymes from diverse organisms. Pseudomonas species produce enzymes that can attack polyurethane, a polymer used in foams and coatings. Bacillus strains secrete enzymes targeting polyethylene and polypropylene, the plastics commonly used in bags and packaging. Rhodococcus bacteria harbor enzymatic pathways for degrading polystyrene, the material in foam cups and packing peanuts. Marine organisms have evolved their own plastic-degrading capabilities, adapted to saltwater conditions and cold temperatures. Fungi contribute enzymes with different specificities and catalytic mechanisms compared to bacterial enzymes, offering additional tools for the synthetic biology toolkit. Each of these enzyme families presents opportunities for engineering. Directed evolution campaigns have improved polyurethane-degrading enzymes, enhancing their ability to break down the complex cross-linked structure of foam materials. Rational design has modified polyethylene-degrading enzymes to increase their activity on the highly crystalline, chemically inert structure of plastic bags. Enzyme cocktails combining multiple specificities can tackle mixed plastic waste, a common situation in landfills and recycling streams where different polymer types are intermixed. The scale-up from laboratory experiments to practical applications presents enormous challenges. Producing enzymes in sufficient quantities for industrial use requires fermentation technology, growing bacteria or yeast in large bioreactors, and purifying the secreted enzymes. The economics must work. The cost of producing the enzymes cannot exceed the value of the recovered monomers or the cost savings from reduced waste disposal. Stability becomes critical. In laboratory tests, enzymes are used immediately after purification and stored under carefully controlled conditions. In real-world applications, enzymes must survive shipping, storage, and deployment in variable environmental conditions. Researchers are addressing stability through multiple strategies. Immobilization involves attaching enzymes to solid supports, protecting them from denaturation while maintaining catalytic activity. The enzymes can be bound to porous beads, incorporated into polymer matrices, or cross-linked into aggregates. Immobilized enzymes often exhibit enhanced stability compared to free enzymes in solution and can be recovered and reused multiple times, improving economic viability. Formulation additives, such as stabilizing proteins, polyols, or salts, can protect enzymes from thermal denaturation and pH extremes during storage and application. These additives create a protective microenvironment around the enzyme, buffering it from harsh external conditions. Protein engineering itself offers solutions to stability challenges. Beyond improving thermostability through directed evolution, researchers have engineered enzymes with increased resistance to chemical denaturants, proteases that might degrade them, and mechanical shearing forces encountered during mixing and pumping. Some engineered variants incorporate disulfide bonds, covalent linkages between cysteine residues that act as molecular staples holding the protein structure together. Others have been modified to reduce the number of surface lysine residues that can undergo non-enzymatic modifications during storage, extending shelf life. The engineering process has achieved remarkable improvements in degradation rates. The original Ideonella sacchaensis PTase required 6 weeks to degrade thin films of amorphous PET. Engineered variants developed over the past decade can accomplish the same degree of degradation in days or even hours under optimal conditions. Some enzyme cocktails combining optimized PTase, MHETase, and accessory enzymes have achieved greater than 90% conversion of PET into monomers within 48 hours at moderate temperatures. These rates approach or exceed the efficiency of chemical recycling methods that require high temperatures and harsh solvents, suggesting that biological degradation could become economically competitive. Yet speed is not the only metric that matters. Selectivity is equally important. An enzyme that degrades plastic waste is useful, but an enzyme that cannot distinguish between waste plastic and useful plastic items would be catastrophic if released into the environment. Engineering specificity into enzymes is more challenging than enhancing activity or stability because it requires modifying substrate recognition without disrupting catalytic function. Researchers use computational modeling to identify binding pocket residues that interact with the substrate, then introduce mutations that alter the shape or chemical properties of the pocket. The goal is to create enzymes that strongly prefer degraded plastic with certain characteristics, such as surface roughness or oxidation state, while ignoring intact manufactured items. Another critical consideration is the fate of degradation products. Breaking down PET into terephthalic acid and ethylene glycol is only beneficial if those compounds can be managed safely. Both monomers are relatively benign at low concentrations and can be metabolized by many microorganisms or recovered for reuse in polymer synthesis. But PET also contains additives, colorants, plasticizers, and stabilizers that are incorporated during manufacturing. When the polymer is degraded, these additives are released. Some, like phthalates used as plasticizers, are known endocrine disruptors. Others, like brominated flame retardants, are persistent organic pollutants. Engineering microbes that can not only degrade the polymer but also detoxify or sequester the additives represents a next-generation challenge for synthetic biology. Researchers have begun addressing this by creating synthetic metabolic pathways that funnel plastic degradation products into useful outputs. Some engineered bacteria convert terephthalic acid not just into carbon dioxide and water, but into valuable chemical intermediates for pharmaceutical or agricultural applications. Others have been modified to incorporate the carbon from plastic into bioplastics, creating a circular system where waste plastic is transformed into new biodegradable materials. These applications move beyond waste management toward valorization, extracting value from materials that would otherwise be discarded. The engineered microbes and enzymes emerging from laboratories represent a powerful arsenal against plastic pollution. But the leap from controlled laboratory experiments to environmental deployment is enormous. In a flask or bioreactor, conditions can be precisely controlled. Temperature, pH, nutrient availability, and enzyme concentration are optimized for maximum performance. The environment is sterile. No competing organisms interfere with the engineered microbes. Substrates are carefully prepared, often using pristine polymer samples rather than aged, contaminated plastic from real waste streams. The real world is chaotic. Landfills contain complex mixtures of plastics, organics, metals, and chemicals. Temperatures fluctuate with seasons and weather. Indigenous microbial communities number in the billions per gram of soil, competing for nutrients and potentially interfering with introduced organisms. The plastic itself is heterogeneous, varying in crystallinity, molecular weight, and contamination level. Ocean environments present entirely different challenges. Salinity, temperature extremes from Arctic to tropical waters, UV radiation from sunlight, nutrient scarcity in open ocean regions, and constant motion from currents and waves. Urban waste streams introduce yet another set of variables. Mixing with organic waste, exposure to cleaning chemicals and detergents, contact with a wide range of materials from food residues to toxic substances. Engineering microbes to function in these complex, variable, and often hostile environments requires moving beyond enzyme optimization to whole organism engineering. Bacteria must be modified not just to produce better enzymes, but to survive and function in the deployment environment. Salt tolerance genes must be added for marine applications. Cold shock proteins enable function in frigid waters. UV protection mechanisms prevent damage from solar radiation. Nutrient scavenging systems allow growth in oligotrophic environments. Biofilm forming capabilities enhance colonization of plastic surfaces. Each of these modifications adds genetic complexity and creates more points of potential failure. The 3 testbeds represent humanity's first serious attempts to deploy this technology at scales large enough to generate meaningful data about real-world performance. An experimental landfill in the southwestern United States hosts buried plastic waste contaminated with 3 decades of soil interaction, providing a substrate more representative of actual disposal conditions than pristine polymer films. The Ocean Gyre Cleanup Project integrates enzyme treatment with mechanical collection, testing whether biological degradation can accelerate the breakdown of recovered debris. The Urban Pilot Program embeds engineered microbes directly into waste management infrastructure, from collection bins to treatment facilities, creating an integrated system of living technology. These are not just technical experiments, they are tests of a fundamental question about human capability and wisdom. The question is not whether we can engineer microbes to eat plastic. That capability is already demonstrated in laboratories around the world. The question is whether we should, whether the benefits justify the risks, and whether we possess sufficient understanding of complex ecosystems to intervene safely at planetary scale. The answer emerges from data, from careful monitoring of what happens when invisible engineered organisms meet the immensely complex reality of the natural world. The discovery of Ideonella sacchaensis was not an isolated phenomenon. Across the planet, in environments contaminated by decades of plastic accumulation, other microorganisms have been evolving similar capabilities. Scientists searching landfills, ocean surfaces, sewage treatment plants, and industrial waste sites have catalogued an expanding repertoire of plastic-degrading bacteria and fungi. Each species brings unique enzymatic machinery and metabolic strategies to the challenge of breaking down synthetic polymers. Together, they represent a distributed evolutionary response to humanity's chemical pollution. a planetary-scale adaptation occurring on microbial timescales measured in years rather than millennia. The genus Pseudomonas contains some of the most metabolically versatile bacteria known to science. These rod-shaped organisms thrive in soil, water, and on plant surfaces, armed with genetic toolkits that allow them to degrade an astonishing array of organic compounds. That versatility has proven crucial for adapting to plastic pollution. Multiple Pseudomonas species have demonstrated the ability to colonize and degrade polyethylene, the most abundant plastic in the environment, accounting for over 30% of global plastic production. Pseudomonas sp. strain AKS2, isolated from contaminated soil samples, generates biofilms that can degrade polyethylene by up to 5% within 45 days without any pretreatment of the plastic. While 5% degradation might seem modest, it represents true biodegradation, the conversion of polymer chains into carbon dioxide, water, and biomass, not just physical fragmentation into smaller pieces. The mechanism by which Pseudomonas attacks polyethylene differs fundamentally from PET's activity on PET. Polyethylene consists of long chains of carbon atoms bonded to hydrogen, lacking the ester linkages that make PET vulnerable to hydrolytic enzymes. Instead, Pseudomonas species employ oxidative enzymes that introduce oxygen-containing functional groups onto the polymer backbone. These oxidations create weak points in the molecular structure, carbon-oxygen bonds, that subsequent enzymes can attack more easily than the robust carbon-carbon bonds of the original polyethylene. The process requires molecular oxygen and often produces intermediate compounds like aldehydes, ketones, and carboxylic acids. These intermediates can then enter central metabolic pathways, providing carbon and energy for bacterial growth. Bacillus represents another powerhouse genus for plastic degradation. These spore-forming bacteria inhabit soil ecosystems worldwide, where they naturally decompose plant materials and other organic matter. Several Bacillus species have shown remarkable abilities to degrade multiple plastic types. Bacillus woodalienchiensis, strain UMT, isolated from landfill soil, achieved 6.6% degradation of plastic in culture media, outperforming many other bacterial isolates tested under the same conditions. Bacillus species often secrete powerful extracellular enzymes, including lipases, esterases, and lactases, that can initiate the breakdown of polymer surfaces. Their ability to form endospores, dormant structures that survive extreme conditions, makes them particularly robust for environmental applications where temperature, moisture, and nutrient availability fluctuate dramatically. The synergy between Pseudomonas and Bacillus has attracted intense research interest. Studies of natural microbial communities on plastic surfaces reveal that these two genera frequently co-occur, suggesting ecological interactions that enhance degradation beyond what either organism achieves alone. In a landmark experiment, researchers isolated 5 bacterial strains from petroleum-polluted soil and assembled them into a consortium. 3 strains belonged to Pseudomonas and 2 to Bacillus. When cultured together on PET as the sole carbon source, this consortium exhibited enhanced growth compared to any single strain. The explanation lies in metabolic complementarity. The Pseudomonas strains possess genes for degrading terephthalic acid, one of the primary breakdown products of PET. The Bacillus strains lack these genes, but instead carry metabolic pathways for processing ethylene glycol, the other major product. Together, they can completely mineralize both components, with each genus handling what the other cannot efficiently process. This division of metabolic labor exemplifies a fundamental principle in microbial ecology called cross-feeding. One organism breaks down a substrate into intermediates that it cannot fully utilize, but those intermediates serve as nutrients for another organism. The second organism's consumption of the intermediates prevents their accumulation, which would otherwise inhibit the first organism's activity. The result is enhanced overall performance, faster degradation rates, and more complete conversion of the substrate into biomass and carbon dioxide. In the Pseudomonas Bacillus consortium, enzymatic cooperation also plays a role. The Pseudomonas strains secrete lipases that can cleave ester bonds in PET and related compounds. The Bacillus strains contribute hydrolases that process the resulting smaller molecules. The combined enzymatic arsenal covers a broader range of chemical transformations than either genus provides independently. Rhodococcus ruber adds another dimension to plastic degradation capabilities. This actinobacterium, characterized by unusual cell wall properties and metabolic versatility, can break down polyethylene at a rate of 0.86% per week. While this sounds slow, it represents continuous activity that accumulates over months and years in environmental settings. Rhodococcus species are known for their ability to degrade hydrocarbons, the chemical compounds that constitute petroleum. This capability translates to plastic degradation because many plastics, including polyethylene and polypropylene, are essentially polymerized hydrocarbons. The enzymes that Rhodococcus uses to attack crude oil, monooxygenases and dioxygenases that insert oxygen atoms into carbon chains, can also modify plastic polymers, creating oxidized sites vulnerable to further degradation. Marine environments present unique challenges and have fostered the evolution of distinct plastic-degrading organisms. Ocean waters vary from frigid polar regions below 0 degrees Celsius to tropical surface waters above 30 degrees. Salinity imposes osmotic stress that terrestrial organisms cannot tolerate. Ultraviolet radiation from sunlight is intense near the surface, causing photodegradation of both cells and plastics. Despite these challenges, marine bacteria and fungi have evolved plastic degradation capabilities. Researchers collecting plastic debris from the North Pacific Ocean, including samples from the Great Pacific Garbage Patch, have isolated numerous marine bacterial strains capable of colonizing and degrading polyethylene. These strains belong to genera common in seawater, including Vibrio, Photobacterium, and Marinobacter, indicating that plastic degradation capacity has emerged across diverse marine lineages. Marine fungi contribute significantly to plastic degradation in ocean environments. A study examining 68 fungal strains cultured from marine habitats found that 42/60% demonstrated the ability to degrade polyurethane. Polyurethane is used in synthetic foams, coatings, and adhesives, and substantial quantities enter marine ecosystems through discarded products and industrial effluent. The marine fungi tested included species of Oreobasidium, Penicillium, and Aspergillus. Oreobasidium sp. Isolate 51 cleared 19% more of a polyurethane-containing plate compared to unconditioned controls after being gradually exposed to increasing concentrations of the plastic. Penicillium coccitripicola cleared 20% more, demonstrating that these fungi can be conditioned through serial exposure to enhance their plastic-degrading performance. One particularly intriguing marine fungus, Parangiodontium album, was isolated from plastic litter floating in the North Pacific Ocean. Laboratory experiments using carbon-13 isotope labeling revealed that this fungus breaks down polyethylene and incorporates the carbon into its biomass, confirming true biodegradation rather than just surface colonization. However, the fungus only degrades polyethylene that has been exposed to ultraviolet light. UV radiation causes photochemical reactions that break some carbon-carbon bonds and introduce oxygen-containing groups onto the polymer surface. These modifications make the plastic more accessible to the fungal enzymes. In the ocean, this means Parangiodontium album can degrade plastic that has floated near the surface long enough to undergo photodegradation, but not plastic that sinks immediately to depths where sunlight does not penetrate. The requirement for UV pretreatment highlights an important interaction between physical and biological degradation processes. Pure polyethylene and polypropylene are chemically inert, with strong carbon-carbon bonds that resist enzymatic attack. Weathering processes, including UV radiation, thermal cycling, and mechanical abrasion, gradually modify the polymer surface. These modifications create carbonyl groups, carboxyl groups, and hydroxyl groups that provide enzymatic footholds. Microorganisms then accelerate degradation by attacking these modified sites. In landfills, decades of burial create similar modifications through chemical oxidation and physical stress from compaction. Plastic waste recovered from a Polish landfill in operation since 1960 showed progressive structural degradation over 60 years, with scanning electron microscopy revealing extensive cracking and surface alterations that facilitate microbial colonization. Microbial consortia outperform single species in nearly every metric relevant to plastic degradation. A study examining polyethylene degradation through experimentally evolved bacterial communities identified Stutzeromonas stutzeri as a keystone species that orchestrates synergistic interactions. This bacterium emerged as the dominant member of a three-species consortium that exhibited dramatically enhanced degradation compared to any single isolate. Transcriptomics analysis revealed the molecular basis for this synergy. Stutzeromonas primarily expressed genes encoding enzymes involved in direct plastic degradation, attacking the polymer chains and generating smaller molecules. The other two community members contributed by secreting extracellular polysaccharides, complex sugars that improve biofilm formation and adhesion to the plastic surface. Enhanced biofilm formation increases the contact between microbial cells and the plastic substrate, raising the effective concentration of degradative enzymes at the crucial interface where degradation occurs. The biofilm itself represents a sophisticated multicellular structure. Early colonization begins within 24 hours as individual bacterial cells adhere to the plastic surface. These pioneer cells secrete extracellular polymeric substances, a mixture of polysaccharides, proteins, nucleic acids, and lipids that forms a sticky matrix. Additional cells attach to this matrix, building layers. Within 12 days, a mature biofilm develops, containing diverse bacterial species arranged in three-dimensional architectures with channels for nutrient flow and waste removal. Bacteria within biofilms experience protection from environmental stresses, including desiccation, UV radiation, and antimicrobial compounds. They also benefit from proximity to neighbors that produce useful metabolites. For plastic degradation, biofilms concentrate degradative enzymes at the plastic surface, while simultaneously providing a stable microenvironment where cooperative metabolism can thrive. Conditioning films play a crucial but often overlooked role in biofilm formation. When plastic enters an aquatic or soil environment, biomolecules from the surroundings, proteins, nucleic acids, humic substances, rapidly adsorb to the surface. This adsorption occurs within minutes to hours, creating a conditioning film that alters the surface properties of the plastic. Hydrophobic plastics like polyethylene and polypropylene become coated with molecules that make the surface more hydrophilic, more water-attracting. This change in surface chemistry influences subsequent bacterial attachment. Experiments comparing conditioned and unconditioned plastic surfaces found that conditioning significantly increases surface roughness and promotes rapid bacterial colonization, with substantial cell attachment occurring within 24 hours. The conditioning film essentially mediates the interaction between the abiotic plastic surface and the biological world, translating chemical signals and physical properties into a language that microorganisms can recognize and respond to. The diversity of plastic-degrading microorganisms extends beyond bacteria and fungi. Certain microalgae and cyanobacteria have shown capabilities to modify plastic surfaces, though their roles in degradation remain less well characterized than those of heterotrophic bacteria. Archaea, the third domain of life, also participate in plastic-associated microbial communities, particularly in anaerobic environments like deep landfill layers. Metagenomic sequencing of landfill samples reveals archaeal populations alongside bacterial and fungal communities, suggesting complex multi-kingdom interactions. While archaea are best known for methanogenesis, the production of methane from organic matter under oxygen-free conditions, some archaeal enzymes may contribute to polymer modifications that facilitate subsequent bacterial degradation. Environmental factors strongly influence which microorganisms dominate plastic-degrading communities. Temperature shapes microbial community composition, with psychrophilic cold-loving organisms in polar waters, mesophilic moderate-temperature organisms in temperate regions, and thermophilic heat-loving organisms in hot environments like composting facilities. pH exerts equally powerful effects. Landfill environments typically range from neutral to slightly alkaline, pH 6.5 to 8.5, which supports diverse bacterial communities. More extreme pH values, below 5 or above 10, drastically reduce microbial diversity and alter community composition towards specialized acidophils or alkaliphiles. Soil pH is recognized as one of the strongest predictors of bacterial community structure, with certain phyla, like acidobacteria and actinobacteria, showing clear abundance patterns correlated with pH levels. Nutrient availability also governs microbial community dynamics. Plastic itself provides carbon, but lacks nitrogen, phosphorus, sulfur, and essential trace elements that microorganisms require. In nutrient-poor environments like the open ocean, plastic degradation proceeds slowly because microbial growth is limited by these missing nutrients. In nutrient-rich environments like landfills containing mixed organic waste, microbial populations can grow robustly, drawing nitrogen from decomposing food waste and other organic materials while utilizing plastic as a carbon source. This nutrient dynamic influences the design of bioremediation strategies. Adding supplemental nutrients can accelerate plastic degradation by removing growth-limiting factors, but excessive nutrient addition risks stimulating non-target organisms and creating ecological imbalances. Oxygen availability determines whether degradation proceeds through aerobic or anaerobic pathways. Aerobic bacteria like Pseudomonas and Bacillus dominate in well-oxygenated surface layers of soil and water, where they employ oxygen-dependent enzymes for plastic oxidation. Deeper in landfills or sediments, oxygen becomes depleted. Anaerobic bacteria take over, using fermentation pathways and alternative electron acceptors like nitrate, sulfate, or carbon dioxide. Anaerobic plastic degradation is generally slower than aerobic degradation because the energy yield from anaerobic metabolism is lower, supporting less biomass production and slower enzymatic processes. However, anaerobic degradation can be significant over long timescales, particularly in permanently saturated environments where oxygen never penetrates. The catalog of plastic-degrading microorganisms continues to expand as researchers screen more environments and apply more sophisticated detection methods. High-throughput sequencing allows scientists to identify microorganisms that cannot be cultured in the laboratory, revealing the hidden diversity of plastic-associated communities. Metagenomics, the sequencing of all DNA in an environmental sample, uncovers genes encoding plastic-degrading enzymes even when the host organisms remain unidentified. These genetic surveys have revealed that plastic degradation genes are more widespread than previously recognized, occurring in diverse microbial lineages across terrestrial, freshwater, and marine ecosystems. The implication is sobering and hopeful. Sobering because it confirms that plastic pollution has exerted selective pressure across vast swathes of the microbial world, altering evolutionary trajectories on a planetary scale. Hopeful because it demonstrates that nature is mounting a response, albeit a slow one by human standards, to the synthetic materials we have released into the environment. This diverse array of plastic-degrading organisms provides the raw material for synthetic biology efforts to engineer enhanced degradation capabilities. Rather than relying solely on directed evolution of a single enzyme or organism, researchers can mix and match enzymatic machinery from different species, combining the polyethylene oxidizing enzymes from Rhodococcus with the PET hydrolyzing enzymes from Ideonella, then adding the biofilm-forming capabilities of marine Pseudomonas and the spore-forming resilience of Bacillus. Such chimeric systems, organisms, or enzyme cocktails incorporating parts from multiple natural sources offer the prospect of tackling the heterogeneous mixtures of plastic types and conditions encountered in real waste streams. Yet the natural diversity also raises questions about deployment strategies. Should engineered organisms be released as monocultures, single optimized strains designed for maximum performance under defined conditions? Or should they be deployed as synthetic consortia, designed mixtures of multiple strains engineered to work together? Or should interventions focus on augmenting native microbial communities, adding specific enzymes or genes to boost the plastic degrading capabilities of the organisms already present in the target environment? Each approach has merits and risks. Monocultures are easier to characterize and control, but may lack the robustness to survive in complex natural environments. Consortia offer redundancy and metabolic diversity, but are harder to predict and monitor. Bioaugmentation of native communities leverages existing ecological networks, but risks unintended consequences from horizontal gene transfer or competitive exclusion of beneficial species. These questions find practical expression in the 3 testbeds where engineered plastic degradation is being tested at environmental scales. The experimental landfill represents a controlled yet realistic environment where microbial communities are complex but spatially contained. The Ocean Jaya project operates in the most open and uncontrolled setting, where released organisms and enzymes disperse through vast volumes of water and interact with countless native species. The urban pilot integrates engineered microbes into designed infrastructure, a middle ground between laboratory control and environmental release. Each testbed will generate data about how diverse plastic-degrading organisms perform when confronting the full complexity of real-world conditions. The results will shape the future of biological plastic remediation, determining which approaches are scientifically viable, ecologically safe, and economically practical. Chapter 5, TSTBD1, The Living Landfill. The experimental landfill occupies a remote valley in the southwestern United States, surrounded by desert scrubland and accessible only by a winding gravel road. This is not an active municipal waste facility accepting daily truckloads of trash. It is a research site, carefully designed to test whether engineered microorganisms can degrade plastic buried for decades under conditions that mimic real disposal environments. The site contains plastic waste from the 1980s and 1990s, buried and compacted according to standard landfill practices of that era. 35 years of chemical oxidation, microbial colonization, and physical stress have transformed the pristine polymers into weathered materials, cracked and discolored, partially covered with biofilms and infiltrated by soil particles. Choice of an aged landfill as a testbed reflects a critical reality. Billions of tons of plastic already sit buried in landfills around the world, accumulating year after year. Even if plastic production ceased tomorrow, this legacy waste would persist for centuries, slowly leaching additives into groundwater and generating microplastics as the polymers fragment. Addressing existing landfill plastic represents an enormous challenge that new waste management strategies alone cannot solve. If biological degradation can accelerate the breakdown of this buried material, the environmental benefits would be substantial, reducing long-term contamination risks and potentially recovering valuable compounds from waste that would otherwise remain inert indefinitely. Site characterization began 2 years before any engineered organisms were deployed. A multidisciplinary team of microbiologists, geochemists, and environmental engineers conducted comprehensive baseline surveys. They excavated test pits at multiple locations, extracting soil and waste samples from various depths. The vertical profile revealed the layered structure typical of older landfills. The surface layer, covering the waste, consisted of compacted soil intended to minimize water infiltration and control odors. Below this, the waste itself formed heterogeneous zones where plastic bags, bottles, and packaging materials mingled with organic waste, paper, glass, and metal. The deeper layers, deposited earliest, showed the most advanced degradation, with organic materials largely decomposed and plastic items displaying extensive physical deterioration. Plastic characterization focused on identifying the types and conditions of polymers present. Samples were collected and analyzed using Fourier transform infrared spectroscopy, a technique that identifies chemical bonds by measuring how the material absorbs infrared light. The results confirmed the presence of all major plastic types. High-density polyethylene from milk jugs and detergent bottles accounted for approximately 25% of plastic items. Low-density polyethylene from grocery bags and plastic film made up 30%. Polypropylene from food containers contributed 15%. Polyethylene terephthalate from beverage bottles comprised 10%. Polystyrene from foam cups and packing materials represented 8%. The remaining 12% consisted of mixed and unidentifiable plastics, often heavily degraded or contaminated. Differential scanning calorimetry provided additional insights into polymer crystallinity. This technique measures the heat required to melt a sample, revealing the proportion of ordered crystalline regions versus disordered amorphous regions. High crystallinity correlates with slow degradation because enzymes struggle to penetrate tightly packed polymer chains. The analysis showed that low-density polyethylene samples retained 40 to 50% crystallinity even after decades of burial, while high-density polyethylene maintained 60 to 70% crystallinity. These values were lower than virgin plastics, indicating that agging had reduced crystalline order, but still high enough to present significant barriers to microbial degradation. PET samples ranged from 20 to 40% crystallinity, depending on their original manufacturing process and weathering history. Scanning electron microscopy allowed direct visualization of surface changes. Fresh plastic surfaces are smooth at the microscale, showing only the patterns imparted by manufacturing processes like molding or extrusion. Aged plastic from the landfill displayed dramatically different morphology. Surfaces were deeply cracked, with fissures extending micrometers into the material. Irregular pits and holes suggested localized degradation. Particulate deposits covered many areas, likely mineral precipitates from groundwater or biofilm remnants. Magnification to several thousand times revealed bacterial cells attached to plastic surfaces, some showing evidence of cell division, indicating active microbial communities colonizing the weathered polymers. These observations confirmed that native microorganisms were already engaged in plastic degradation, albeit at very slow rates. Soil chemistry profiling measured parameters crucial for microbial activity. pH values ranged from 6.8 to 7.5 across most of the site, within the optimal range for diverse bacterial communities. Some localized areas showed pH as low as 6.2, possibly due to organic acid production by decomposer organisms, or as high as 8.0, perhaps from mineral dissolution. Temperature profiles showed typical patterns: cooler near the surface, where daily and seasonal variations penetrate; warmer at depth, where geothermal gradients and metabolic heat from decomposition create stable conditions around 18 to 22 degrees Celsius. Moisture content varied considerably, with some zones nearly saturated and others relatively dry, reflecting heterogeneous water infiltration patterns and the variable absorbency of different waste materials. Nutrient availability assessments revealed spatially variable conditions. Zones rich in decomposed organic matter showed elevated nitrogen and phosphorus, released as proteins and other biological molecules broke down. Zones dominated by plastic contained minimal nitrogen and phosphorus, limiting microbial growth despite the theoretical availability of carbon from the polymers. This nutrient limitation suggested that bioaugmentation strategies would need to consider nutrient supplementation or select organisms capable of scavenging trace nutrients efficiently. Trace element analysis detected adequate levels of iron, manganese, zinc, and other micronutrients essential for enzyme function, indicating that these would not limit degradation activity. Baseline microbial community profiling employed high-throughput 16S ribosomal RNA gene sequencing, a method that identifies bacterial species based on variations in a universally present gene. The landfill hosted remarkably diverse bacterial communities, with over 2,000 distinct operational taxonomic units detected across all samples. Community composition varied with depth and waste composition. Surface soils were dominated by typical soil bacteria, protobacteria, acidobacteria, actinobacteria, and bacteroidetes. Deeper within the waste, facultative anaerobes and strict anaerobes became more abundant, including firmicutes, clostridioles, and methanogenic archaea. Plastic-rich zones harbored species known for polymer degradation, including representatives of Pseudomonas, Bacillus, Rhodococcus, and Streptomyces. Intriguingly, the baseline surveys detected genes associated with plastic degradation even before any engineered organisms were introduced. Metagenomic sequencing, which captures all genetic material in a sample, identified sequences similar to PTAs, cutinases, lacases, and monooxygenases. The presence of these genes indicated that the native microbial community possessed some inherent plastic-degrading capability, likely evolved in response to decades of exposure. Quantifying this background activity became a priority for distinguishing the effects of engineered organisms from ongoing natural processes. Control zones were established where no engineered microbes would be added, allowing researchers to monitor natural degradation rates for comparison with augmented zones. The experimental design divided the landfill site into distinct sectors. 4 treatment zones, each approximately 50 meters by 50 meters, would receive different formulations of engineered organisms or enzyme cocktails. 2 control zones of equal size would remain untreated to measure background degradation rates. Buffer zones separated experimental areas to minimize cross-contamination, though researchers acknowledged that complete isolation would be impossible given microbial mobility and groundwater flow. Within each treatment zone, grid-based sampling stations were established at 5-meter intervals, providing spatial resolution to detect heterogeneity in degradation patterns. Monitoring infrastructure was installed months before organism deployment. Each sampling station included soil gas probes to measure oxygen, carbon dioxide, and methane concentrations, indicators of microbial metabolic activity. Moisture sensors tracked water content dynamics, critical for understanding microbial activity patterns and potential leachate generation. Temperature loggers recorded continuous thermal profiles. Sampling ports allowed collection of soil pore water for chemical analysis without disturbing the waste matrix. Groundwater monitoring wells were positioned up gradient and down gradient of the site to detect any migration of contaminants or engineered organisms beyond the intended treatment zones. Genetic tracking systems were incorporated into the engineered microbes to enable their detection and quantification in environmental samples. The organisms carried unique DNA sequences, molecular barcodes, not found in nature. Quantitative PCR assays, highly sensitive molecular techniques, could detect as few as 100 cells per gram of soil by amplifying these barcode sequences. This capability would allow researchers to track the population dynamics of the introduced organisms over time, mapping where they proliferate, where they decline, and whether they spread beyond designated treatment zones. Monitoring would continue for at least 5 years, potentially much longer depending on results and funding. Containment considerations shaped every aspect of the experimental design. While the landfill represented a somewhat closed system, with waste buried under compacted soil and surrounded by relatively impermeable clay layers, researchers recognized that absolute containment was impossible. Rainfall infiltration, groundwater flow, and biological activity create pathways for movement. Engineered organisms might spread through soil pores, be carried by percolating water, or even be transporting by insects and other organisms that burrow through the site. Assessing this mobility would be a key outcome of the experiment, informing risk assessments for future deployments. Multiple safeguard mechanisms were built into the engineered organisms to mitigate risks. Nutrient dependency modifications required the bacteria to receive periodic additions of a specific synthetic compound not present in nature. Without this compound, the cells would enter a non-growing state and eventually die, preventing long-term persistence if they escaped the treatment zones. Geographic restriction systems made the organisms dependent on specific temperature ranges or pH conditions present at the site, but unusual in surrounding environments. Kill switches, genetic circuits that trigger cell death in response to particular signals, provided an emergency termination option if populations needed to be eliminated. All these safety features had been tested extensively in laboratory settings, but the landfill deployment would reveal how they performed under real environmental conditions, where evolution and horizontal gene transfer could potentially disable the safeguards. The engineered organisms themselves represented the culmination of years of laboratory optimization. 4 different formulations were prepared for the 4 treatment zones. Zone 1 would receive a monoculture of engineered Ideonella sacchaensis, expressing enhanced P-tase and MHE-tase variants optimized for activity at ambient landfill temperatures. Zone 2 would get a synthetic consortium combining engineered Pseudomonas species for polyethylene degradation with Bacillus species for enhanced biofilm formation and stress tolerance. Zone 3 would be treated with enzyme cocktails rather than living organisms, purified and stabilized P-tase, MHE-tase, lacases, and cutinases formulated for slow release from biodegradable carrier matrices. Zone 4 would combine both living organisms and enzyme supplements, testing whether a hybrid approach outperforms either strategy alone. The team prepared for deployment day with the awareness that they were crossing a threshold. Laboratory experiments provided crucial proof of concept, demonstrating what was possible under controlled conditions. But environments are not laboratories. The landfill contained thousands of variables that could not be replicated in any flask or bioreactor. Competition from native microbes, predation by protozoa, viral infections, unanticipated toxic compounds, physical heterogeneity, and countless other factors would influence the fate and effectiveness of the engineered organisms. The experiment would generate critical data, but it would also generate uncertainty, new questions replacing old ones as the complex reality of environmental biotechnology revealed itself. As the sun rose over the desert landscape, the research team gathered at the site. Trucks carrying sealed containers of engineered microbes, enzyme formulations, and deployment equipment lined the access road. Safety protocols were reviewed one final time. Communication systems were tested. Weather conditions were checked, with forecasts showing no rain for the next 5 days, providing an optimal window for establishment before moisture patterns changed. The experiment was ready to begin. What happened next would shape the future of biological waste remediation, providing the first large-scale data on whether humanity's engineered solutions could coexist safely with the ecosystems they were meant to heal. Chapter 6: Mapping the Waste Mountain. Understanding what lies buried in a landfill requires archaeological precision combined with chemical detective work. The waste mountain at the experimental site contained decades of discarded materials, not neatly organized but chaotically mixed, compressed under tons of weight, and transformed by chemical and biological processes that began the moment the first item was buried. Mapping this heterogeneous mass became the foundation for predicting where engineered organisms might work, where they would struggle, and what unintended interactions might emerge from the encounter between synthetic biology and synthetic materials aged in Earth's subsurface. The characterization team excavated systematically, creating vertical profiles at 25 locations across the site. Each excavation descended 5 meters, the maximum depth of waste burial, before hitting the underlying clay layer that formed the landfill base. Samples were collected every 50 centimeters, bagged in sterile containers, labeled with GPS coordinates and depth, then transporting to on-site analytical facilities. This generated over 250 distinct samples, each representing a snapshot of conditions at a specific point in 3-dimensional space. The effort resembled an environmental core sampling campaign, treating the landfill as a stratigraphic sequence where time was recorded in layers, oldest at the bottom, youngest at the top. Visual inspection revealed patterns. The uppermost layers, deposited most recently, contained plastics that retained much of their original appearance. Bottles still bore faded labels. Bags, though fragmented, showed recognizable shapes and colors, but deeper samples told different stories. Plastics from the 1980s had lost almost all surface features. Colors had faded to uniform grays and browns. Materials crumbled under light pressure, fragmenting into pieces ranging from several centimeters to submillimeter particles. The deepest samples contained extensive microplastics, fragments below 5 millimeters, that resulted from decades of mechanical stress and weathering. These microplastics were intimately mixed with soil particles, making separation difficult and raising questions about how engineered organisms would access this fragmented, dispersed substrate. High-density polyethylene items, originally robust containers for household products, displayed surprising variability. Some bottles remained structurally intact, albeit brittle and prone to cracking. Others had disintegrated into flakes and shards. Fourier transform infrared spectroscopy analysis of these samples revealed chemical changes. Virgin HDPE shows characteristic absorption peaks corresponding to carbon-hydrogen stretching vibrations in the polymer backbone. Aged HDPE from the landfill exhibited additional peaks in the carbonyl region, indicating the presence of carbon-oxygen double bonds that do not exist in the original material. These carbonyl groups form through oxidation, a process driven by oxygen exposure, UV radiation before burial, and possibly microbial activity. The oxidation creates points of weakness in the polymer chains, making the material more susceptible to further degradation. Differential scanning calorimetry quantified the extent of structural degradation by measuring crystallinity changes. Fresh HDPE typically exhibits 70 to 80% crystallinity, reflecting tightly ordered packing of polymer chains. Samples from the shallowest layers showed 60 to 70% crystallinity, a modest decrease consistent with limited agging. Mid-depth samples ranged from 50 to 60%. The deepest samples, buried for nearly 40 years, showed crystallinity as low as 45%. This progressive decline confirmed that agging had disrupted crystalline order, creating more amorphous regions where polymer chains are loosely arranged and therefore more accessible to enzymatic attack. Extrapolating from this data, researchers estimated that complete loss of crystallinity in HDPE under landfill conditions would require approximately 670 years, far longer than human civilization has been producing plastic. Low-density polyethylene, used primarily in thin films and bags, showed even more dramatic degradation. The material's lower crystallinity and thinner cross-sections made it more vulnerable to physical and chemical processes. Many LDPE samples had fragmented into pieces too small to handle individually, requiring bulk analysis of mixed fragments. Microscopy revealed that fragmentation proceeded through crack propagation. Small surface defects, perhaps from manufacturing or initial mechanical stress, widened over time. Repeated compression and expansion cycles as the landfill settled and temperature fluctuated drove crack growth. Eventually, cracks intersected, liberating fragments from the parent material. The process resembled geological weathering of rocks, but operating on polymeric rather than mineral substrates. Polypropylene samples displayed their own degradation signatures. This plastic, commonly used in rigid food containers and automotive parts, is known for excellent chemical resistance. Yet landfill samples showed surface pitting, discoloration, and reduced mechanical strength. FTIR analysis detected oxidation products similar to those in polyethylene, confirming that chemical agging was occurring despite polypropylene's reputation for stability. The degradation appeared to proceed from the surface inward, with outer layers showing more extensive modification than interior regions. This gradient suggested that degradation rates were limited by the diffusion of oxygen or reactive species into the bulk material, a factor that would also influence enzymatic degradation, because enzymes can only access surface and near-surface regions unless the material is physically broken apart. Polyethylene terephthalate bottles presented an interesting case. PET's semicrystalline structure and resistance to hydrolysis under neutral pH conditions should make it highly stable in landfills. Indeed, many PET bottles from the 1990s looked superficially intact, maintaining their shape and transparency, but closer examination revealed subtle changes. Surfaces showed a milky haze indicative of crazing, a form of microfracture where tiny cracks scatter light. Mechanical testing found that aged PET had lost significant tensile strength, fracturing under stresses that virgin PET would withstand easily. FTIR spectra showed minimal chemical changes, suggesting that PET degradation in the landfill was primarily physical rather than chemical, at least over the 40-year timescale sampled. However, some PET samples did show signs of chemical degradation, particularly those in contact with moisture-rich organic waste. These samples exhibited surface etching visible under scanning electron microscopy, with pitting patterns reminiscent of enzymatic attack. Metagenomic analysis of biofilms on these surfaces detected genes with sequence similarity to PTase and MHETase, suggesting that native microbial communities were slowly degrading the material. The degradation rate appeared minuscule, perhaps a fraction of a percent per year, but accumulated over decades, it produced measurable effects. This natural degradation established a baseline against which the performance of engineered organisms would be judged. Polystyrene foam, ubiquitous in packaging and disposable food containers, proved among the most persistent materials. Foam samples from the deepest layers looked nearly identical to fresh foam, maintaining their cellular structure and white color. The material's low density and high surface area should theoretically make it accessible to microbial colonization, but polystyrene's chemical structure, an aromatic hydrocarbon polymer, resists biodegradation more than aliphatic polymers like polyethylene. Only a few bacterial genera, notably some Pseudomonas and Rhodococcus species, possess enzymes capable of initiating polystyrene degradation, and even these organisms degrade the material very slowly. The persistence of polystyrene in the landfill underscored the need for targeted enzymatic approaches, as naturally occurring degradation appeared negligible. Additives and contaminants complicated the picture. Plastics are rarely pure polymers. Manufacturers add stabilizers to prevent degradation during use, plasticizers to improve flexibility, colorants for aesthetics, and flame retardants for safety. When plastics degrade, these additives are released. Chemical analysis of landfill leachate, the liquid that percolates through waste and can carry dissolved compounds, detected phthalates, bisphenol A, brominated flame retardants, and heavy metals like cadmium and lead. Some of these compounds are known to be toxic to microorganisms, potentially inhibiting the very bacteria being deployed to degrade the plastic. Others are endocrine disruptors or carcinogens, raising concerns about whether accelerating plastic degradation might mobilize these hazardous substances more rapidly than natural processes would. Soil chemistry profiling revealed steep gradients in many parameters. Near the surface, where oxygen from the atmosphere could diffuse into the soil, aerobic conditions prevailed. Oxygen concentrations measured 15 to 20%, similar to ambient air. Microbial respiration consumed some oxygen, but atmospheric diffusion replenished it. At depths below 2 meters, oxygen levels dropped precipitously, falling to below 1%. This transition from aerobic to anaerobic conditions would profoundly affect which organisms could thrive and which degradation pathways would operate. Aerobic bacteria, including most of the engineered strains designed for plastic degradation, require oxygen for their metabolism. In anaerobic zones, these organisms would be inactive or dead, leaving degradation to anaerobic species with far slower degradation capabilities. pH mapping identified unexpected heterogeneity. While the overall site averaged neutral pH, localized zones ranged from 5.5 to 8.5. The acidic zones correlated with areas rich in decomposing organic matter, where organic acids and carbon dioxide production from microbial activity lowered pH. Alkaline zones often coincided with construction debris, concrete fragments that slowly dissolve and release calcium hydroxide. These pH variations would influence enzyme activity, as most plastic-degrading enzymes have relatively narrow, optimal pH ranges. Organisms deployed in zones outside their pH tolerance might fail to establish or function effectively. Temperature monitoring revealed less variability than pH or oxygen. Most of the landfill maintained relatively stable temperatures between 17 and 22 degrees Celsius, insulated from daily and seasonal atmospheric fluctuations by the overlying soil and waste. Some zones showed slightly elevated temperatures, up to 26 degrees, potentially due to metabolic heat from ongoing decomposition of organic materials. These temperatures fell within the mesophilic range suitable for most engineered bacterial strains, avoiding the need for extreme thermophilic or psychrophilic adaptations. However, seasonal variations in the surface layers might stress organisms during summer heat or winter cold, requiring consideration of deployment timing to maximize establishment success. Moisture content showed the greatest spatial variability of any parameter measured. Some zones were nearly saturated, with water filling most of the pore space between waste particles. These zones corresponded to areas where the landfill topography channeled rainwater infiltration or where impermeable layers trapped water. Other zones were remarkably dry, with moisture contents below 10% by weight. Dry zones occurred in areas with good drainage or where hydrophobic plastic materials shed water and prevented accumulation. Microbial activity requires water for metabolism, enzyme function, and nutrient transport. Organisms in dry zones would face severe stress, likely persisting as dormant spores or resting stages rather than actively degrading plastic. The moisture patterns suggested that engineered degradation would be spatially patchy, occurring rapidly in moist zones and slowly or not at all in dry zones. Integrating all this characterization data produced three-dimensional maps of the landfill, showing not just where different plastics were located, but also the environmental conditions at each location. These maps became predictive tools. Areas with abundant PET, adequate moisture, neutral pH, and aerobic conditions were flagged as high-probability success zones for Ideonella sacchaensis deployment. Areas with polyethylene, moderate moisture, and facultative anaerobic conditions suited the Pseudomonas bacillus consortium. Enzyme-only treatments might work in dry zones where living organisms would struggle, assuming the enzymes could be formulated for stability in low-moisture environments. Control zones were selected to span the range of conditions observed, ensuring that baseline degradation monitoring captured natural variability. Control Zone 1 encompassed an area with mixed plastics, neutral pH, moderate moisture, and aerobic conditions, representing optimal circumstances for natural degradation. Control Zone 2 covered a drier, more compacted region with lower oxygen availability, representing challenging conditions where natural degradation should be minimal. Comparing treated zones to these controls would allow quantification of how much enhancement the engineered organisms provided over background processes. The characterization phase also established protocols for ongoing monitoring. Every 2 weeks, samples would be collected from designated monitoring points. Weight loss of plastic samples embedded at known locations would provide direct evidence of degradation. Fourier transform infrared spectroscopy would track chemical changes in polymer structure. Scanning electron microscopy would visualize surface modifications. Quantitative PCR would measure populations of engineered organisms. Metagenomic sequencing would reveal changes in the broader microbial community. Leachate chemistry would detect release of additives or degradation products. Gas measurements would indicate metabolic activity. This intensive monitoring would generate terabytes of data, requiring sophisticated statistical analysis to extract meaningful patterns. The waste mountain had been mapped. Its complexity was daunting, a chaotic mixture of materials, chemicals, and microorganisms layered and compressed over decades. Yet this complexity was precisely why the experiment mattered. If engineered plastic degradation only worked in pristine laboratory conditions, it would have limited real-world value. The landfill represented reality, messy and heterogeneous, where solutions must prove robust against countless confounding factors. The maps provided a baseline, a snapshot of conditions before intervention. What came next would reveal whether synthetic biology could navigate this complexity and deliver meaningful plastic degradation in the most challenging environment of all, the accumulated waste of human civilization buried beneath the earth. The morning air was cool and still, the desert sun just beginning to crest the horizon and paint the surrounding hills in shades of amber and rust. The research team gathered at the site entrance, 23 people representing 7 institutions and 5 disciplines. Microbiologists checked the sealed containers holding engineered bacteria, their cargo maintained at precise temperatures by portable refrigeration units powered by generators humming in the background. Ecologists reviewed monitoring protocols on tablet computers, running through checklists that had been refined over months of planning. Environmental engineers inspected the deployment equipment, custom-designed spray systems and injection probes that would introduce the organisms into specific soil layers without causing unnecessary disturbance. This was not a typical scientific experiment. The decision to release genetically engineered organisms into an open environment, even a partially contained one like the landfill, required approvals from multiple regulatory agencies. 2 years of risk assessments, environmental impact statements, and public comment periods preceded this moment. The Institutional Biosafety Committee had reviewed the protocols countless times, evaluating containment strategies, monitoring plans, and emergency response procedures. The project carried liability insurance and maintained dedicated funds for remediation should unexpected problems arise. Every team member had undergone specialized training in biosafety practices and had signed documents acknowledging their responsibilities. Dr. Sarah Chen, the project's principal investigator, addressed the assembled team. Her voice carried the weight of 2 decades of experience in environmental microbiology and an acute awareness of the historical significance of this deployment. She reminded everyone that this experiment stood on the shoulders of earlier controversies: the ice- bacteria release in 1987, the first genetically modified crops in the 1990s, all of which had sparked intense public debates about the wisdom of releasing engineered organisms. Those earlier projects taught hard lessons about community engagement, regulatory oversight, and the need for transparent communication. The team had taken those lessons to heart. 6 months before deployment, they held the first of many public meetings in the nearest town, 40 kilometers from the landfill site. The meetings were advertised through local newspapers, radio stations, social media, and posted notices at community centers. Initial attendance was modest, about 30 people, a mix of curious residents, environmentalists, and ranchers whose land adjoined the study area. The research team presented the science behind plastic-eating bacteria, explained the containment measures, and most importantly, listened to concerns. Those concerns were numerous and pointed. Would the bacteria spread to nearby properties? Could they harm livestock or wildlife? What if they degraded useful plastics, farm equipment, irrigation pipes, vehicle parts? Why take this risk at all when traditional waste management, despite its problems, does not involve releasing novel organisms? The questions reflected genuine anxiety about unintended consequences, grounded in decades of cautionary tales about ecological interventions gone wrong. Some residents referenced the introduction of cane toads in Australia, a biological control measure that became an invasive pest. Others mentioned genetically modified crops and long-running debates about their environmental impacts. Dr. Chen and her colleagues responded with data, transparency, and humility. They presented the genetic safeguards built into the organisms, nutrient dependencies that would prevent long-term survival outside the treatment zone, temperature sensitivities that limited where the bacteria could grow, and kill switches that could be activated if populations needed emergency termination. They acknowledged that absolute certainty was impossible, that every environmental intervention carried risks, but argued that the accelerating plastic pollution crisis demanded new approaches. They emphasized the monitoring infrastructure, the network of sensors and sampling stations that would detect any spread beyond designated areas within days. Community sentiment shifted gradually through repeated engagement. A second meeting attracted over 100 people, and a third drew 200. Local television stations covered the project, interviewing both researchers and community members. A dedicated website provided updates, posted monitoring data in near real time, and offered channels for submitting questions or concerns that the team committed to answering within 48 hours. A community advisory board was established, 7 residents appointed to receive detailed briefings and provide ongoing feedback. This board would have the authority to recommend pausing or terminating the experiment if community concerns escalated. By deployment day, public opinion polls conducted by an independent firm showed that 68% of area residents supported the project, 19% remained neutral, and 13% opposed it. Those numbers represented significant trust earned through months of patient dialogue. Yet the researchers remained acutely aware that maintaining that trust required flawless execution of their monitoring plans and immediate, transparent response to any problems. The deployment itself was methodical, almost ritualistic in its precision. Zone 1, designated for the engineered Ideonella sacchaensis strain, would be treated first. The bacterial culture had been grown in large fermentation tanks at a nearby university facility, harvested during exponential growth phase when cells were most vigorous, and suspended in a protective medium containing nutrients and cryoprotectants. The suspension contained approximately 10 billion cells per milliliter, a concentration determined by laboratory optimization to balance effective colonization against overwhelming the native microbial community. 4 technicians wearing protective suits, gloves, and respirators approached the first treatment zone. The suits were not strictly necessary. The engineered bacteria posed no health threat to humans, but biosafety protocols mandated precautions for any release of genetically modified organisms. Each technician carried a backpack sprayer modified with precision nozzles designed to disperse the bacterial suspension evenly across the soil surface. They walked in predetermined patterns, guided by GPS units, ensuring complete coverage of the 50-meter by 50-meter zone. The process took 45 minutes, delivering a total of 200 liters of bacterial suspension across the 2,500 square meter area. But surface application was only the first step. Plastic was buried at various depths, and organisms needed access to subsurface layers. Injection teams followed with truck-mounted equipment carrying long probes that could be driven 2 meters into the ground. At each of 100 injection points, spaced across a 5-meter grid, the probes delivered 1 liter of bacterial suspension directly into subsurface waste layers. This dual approach, surface spraying and subsurface injection, maximized the probability that engineered organisms would contact plastic substrates throughout the vertical profile. Zone 2 received similar treatment, but with the synthetic consortium combining Pseudomonas and Bacillus species. This formulation appeared cloudier than the Ideonella suspension, reflecting the more complex mixture of bacterial types and the extracellular polysaccharides produced by some strains to enhance biofilm formation. The consortium had been grown in co-culture, allowing the species to establish metabolic interactions before deployment, potentially improving their cooperative function in the field. Zone 3 posed different challenges. Instead of living organisms, this area would receive enzyme cocktails, purified proteins including PTase, MHEtase, lacases, and cutenases. Enzymes lack the ability to reproduce or spread, offering greater predictability but potentially limiting their persistence. To address this limitation, the enzymes had been formulated with stabilizers and encapsulated in biodegradable polymer microspheres designed for slow release over weeks. The microspheres, made from polylactic acid, would gradually erode in the soil environment, continuously exposing fresh enzymes to plastic surfaces. This approach mimicked controlled-release fertilizers and pesticides, technologies proven in agriculture over decades. Deployment teams applied the enzyme microspheres as a dry powder, blown across the surface using modified agricultural spreaders, then watered in to initiate microsphere activation. Subsurface injection delivered liquid enzyme formulations to deeper layers. The total enzyme loading was substantial, 5 kilograms of purified proteins distributed across the zone, representing months of fermentation and purification work and a material cost exceeding $30,000. This investment underscored the experimental nature of the approach, economically impractical at larger scales unless enzyme production costs could be dramatically reduced. Zone 4 combined living organisms and enzyme supplements, testing whether hybrid approaches might synergize. The Ideonella strain was applied first, followed by enzyme microspheres containing P-tase and MHE-tase variants that were even more thermostable than the versions encoded in the bacterial genomes. The hypothesis was that exogenous enzymes would provide immediate activity while bacterial populations established and began producing their own enzymes, and that the superior thermostability of the supplemental enzymes might degrade plastics in microenvironments too warm for optimal bacterial growth. Control zones were treated only with sterile application media, the same solutions used to suspend bacteria or enzymes but containing no active components. This ensured that any effects from physical disturbance or nutrient addition could be separated from effects attributable to the engineered organisms or enzymes. Sample collection from control zones would proceed on the same schedule as treatment zones, generating parallel datasets for statistical comparison. As deployment progressed, the monitoring infrastructure came online. Soil gas sensors began recording oxygen and carbon dioxide concentrations at 5-minute intervals, transmitting data wirelessly to a central computer system. Initial readings confirmed baseline conditions: oxygen concentrations around 18% near the surface, declining to 3% at 2-meter depth, and carbon dioxide showing inverse patterns, low at the surface and elevated at depth where microbial respiration accumulated. Any significant changes in these gas profiles would indicate altered microbial activity potentially attributable to the introduced organisms. Moisture sensors tracked soil water content, critical for interpreting microbial activity since bacteria require water for metabolism. The spring deployment timing was deliberate, coinciding with seasonal moisture availability before summer heat-induced drought conditions. Weather forecasts predicted no rain for 5 days, allowing organisms time to establish and begin colonizing plastic surfaces without being immediately flushed deeper by percolating water. Temperature loggers confirmed stable subsurface temperatures between 18 and 20 degrees Celsius, well within the optimal range for the deployed organisms. The genetic tracking system was tested immediately. Soil samples collected from each injection point 1 hour after deployment were processed in the mobile laboratory, a climate-controlled trailer equipped with DNA extraction equipment and quantitative PCR instruments. The analyses detected the unique genetic barcodes incorporated into the engineered strains, confirming successful delivery. Cell counts from Zone 1 samples averaged 8 million engineered Ideonella cells per gram of soil near injection points, declining to 100,000 per gram in areas between injection points, the expected pattern given the distribution method. Baseline sampling of native microbial communities had occurred the previous week, providing reference data for comparison. Those analyses identified over 1,800 bacterial operational taxonomic units across the treatment zones, a typical diversity for landfill environments. The introduced organisms initially represented a tiny fraction of total microbial biomass, roughly 1% at injection points and far less elsewhere. Whether they would proliferate, remain stable, or decline would depend on their ability to compete with native organisms, access plastic substrates, and tolerate environmental stresses. The interdisciplinary team dynamics were evident throughout deployment. Microbiologists focused on organism viability and application protocols. Ecologists emphasized monitoring design and statistical power to detect effects against natural variability. Engineers solved practical problems with equipment and delivery systems. Social scientists documented the process for future analysis of decision-making and risk communication. This convergence of expertise exemplified the modern practice of team science, where complex problems demand integration across traditional disciplinary boundaries. Dr. Chen had recruited this diverse team deliberately, recognizing that plastic bioremediation was not purely a microbiological problem. It intersected with ecology, engineering, chemistry, sociology, ethics, and policy. Weekly meetings required developing shared vocabulary, as terms that had precise meanings in one discipline often meant something different in another. Early in the project, a heated discussion about the definition of biodegradation revealed that engineers measured it as mass loss, chemists as molecular transformation, and ecologists as incorporation into biogeochemical cycles. Reconciling these perspectives required patience, but ultimately strengthened the experimental design by ensuring multiple complementary measurements. As the last deployment team completed their work, the site fell quiet except for the hum of generators powering monitoring equipment. Weeks of preparation had culminated in a single day of activity, and now the waiting began. Biological processes operate on their own timescales, indifferent to human impatience. The engineered bacteria would need time to acclimate to soil conditions, colonize plastic surfaces, and ramp up enzyme production. Measurable degradation, if it occurred, might not manifest for weeks or months. The team established a rotation schedule. At least 2 researchers would be on site at all times, maintaining equipment, collecting scheduled samples, and serving as emergency responders if monitoring systems detected anomalies. Weekly sampling would continue for the first 3 months, shifting to bi-weekly for months 4 through 12, then monthly for the duration of the 5-year study. Each sample would undergo a battery of analyses: microbial community composition by 16S sequencing, engineered organism abundance by qPCR, plastic degradation by weight loss and spectroscopy, soil chemistry, and leachate analysis. Data would accumulate slowly, each sample adding incremental information to the growing dataset. Statistical analysis would require patience as well. Distinguishing true treatment effects from natural variability demanded sufficient replication and temporal coverage. The experimental design provided both, but the clearest patterns might not emerge until year 2 or 3. Some team members, particularly graduate students who needed dissertation data, found this timeline frustrating. Yet Dr. Chen emphasized that environmental science operated on environmental timescales, and rushing to conclusions based on limited data had ruined more than one promising technology by generating inflated expectations that reality could not sustain. As the team departed the site that evening, the treated zones looked unchanged, just as they had that morning. But beneath the unremarkable soil surface, an experiment of enormous consequence was beginning. Engineered organisms crafted in laboratories were encountering the uncontrolled complexity of a real ecosystem. They would face competition, predation, starvation, desiccation, and countless challenges that no laboratory study could fully anticipate. Their fate would reveal fundamental truths about the feasibility of environmental synthetic biology and would shape decisions about whether to scale this technology to address plastic pollution globally. The invisible revolution was underway, and the outcome would echo across science, policy, and society for decades to come. Chapter 8: Early INDICATOS. 30 days after deployment, the first comprehensive sampling campaign began. Research teams converged on the site before dawn, working methodically through predetermined sampling protocols. The goal was to collect enough material to characterize changes in plastic condition, microbial community composition, and soil chemistry while minimizing disturbance that might confound future measurements. Each sample had to be geographically precise, collected from exact coordinates to allow temporal comparisons, yet representative of the larger zone being studied. The plastic samples themselves had been prepared months earlier. Small squares of PET, HDPE, LDPE, and PP films weighing precisely 5 grams each, embedded at known locations throughout the treatment and control zones. Each sample was tagged with a chemically inert marker containing a unique identification number, allowing recovery and verification. These standardized test samples eliminated variability from the heterogeneous composition of the actual landfill waste, providing clean measurements of degradation rates. The first samples retrieved from Zone 1, where engineered Ideonella sacchaensis had been deployed, showed subtle but measurable changes. Under magnification, the PET film surfaces displayed a texture absent from control samples. Fresh PET appears smooth and glossy, reflecting light uniformly. The treated samples showed a matte finish, particularly along edges and corners where the plastic met the soil. Scanning electron microscopy at 1,000 times magnification revealed the cause. Bacterial cells clustered densely across the plastic surface, many encased in extracellular matrix material that created a biofilm architecture of channels and towers. Atmospheric scanning electron microscopy, a technique that allows visualization of samples in hydrated conditions rather than requiring vacuum drying, provided even more dramatic images. Ideonella cells appeared as elongated rods, typically 1 to 2 micrometers long, oriented perpendicular to the plastic surface like a forest of microscopic pillars. Intercellular structures connected adjacent cells, likely composed of extracellular DNA and proteins that stabilize biofilm architecture. Most strikingly, the plastic surface beneath the biofilm showed etching patterns, irregular pits and grooves consistent with enzymatic degradation. Quantitative analysis confirmed these visual impressions. The PET test samples from Zone 1 showed an average weight loss of 0.8% compared to initial mass, a small but statistically significant decrease. Control zone samples showed no measurable weight loss, confirming that natural processes over 30 days did not produce detectable degradation. The difference was clear evidence that the engineered organisms were active. Fourier transform infrared spectroscopy provided molecular-level confirmation. This technique identifies chemical bonds by measuring how materials absorb infrared light at specific wavelengths. Fresh PET shows characteristic absorption peaks at 1,715 wave numbers, corresponding to carbonyl groups in ester linkages, and at 1,240 and 1,095 wave numbers from carbon-oxygen bonds in the polymer backbone. Treated PET samples from Zone 1 exhibited reduced intensity at these peaks, indicating loss of ester bonds, the precise chemical change expected from p-TASE activity. Additionally, new peaks appeared in the infrared spectra at 3,300 to 3,500 wave numbers, a region associated with hydroxyl groups. These peaks suggested the presence of degradation intermediates or end products, possibly MHET, terephthalic acid, or ethylene glycol, the molecular fragments released when p-TASE and MHETase cleave PET polymer chains. The infrared data thus corroborated the weight loss measurements, confirming that observed changes reflected true chemical degradation rather than just physical alterations like water absorption or mineral deposition. Differential scanning calorimetry revealed changes in crystallinity. Fresh PET test samples averaged 32% crystallinity, deliberately chosen as a mid-range value between completely amorphous and highly crystalline PET. After 30 days in Zone 1, crystallinity decreased to 29%. This decline indicated preferential degradation of amorphous regions, where polymer chains are loosely arranged and more accessible to enzymes, while crystalline regions remained relatively intact. The pattern matched laboratory observations of Ideonella sacchaensis, which degrades amorphous PET far more rapidly than crystalline material. Zone 2, treated with the Pseudomonas bacillus consortium, showed different patterns. PET degradation was minimal, just 0.2% weight loss, not significantly different from controls. This made sense because the consortium had been optimized for polyethylene degradation, not PET. The polyethylene samples, however, told a different story. HDPE test films showed average weight loss of 0.5%, while LDPE films lost 1.2%. LDPE's lower crystallinity made it more vulnerable, confirming the principle that polymer accessibility governs degradation rates. Infrared spectroscopy of polyethylene samples from Zone 2 revealed oxidation signatures. New absorption peaks appeared at 1,712 wave numbers, characteristic of carbonyl groups that do not exist in fresh polyethylene. These carbonyls form when bacterial enzymes, particularly monooxygenases, insert oxygen atoms into the polymer backbone. The oxidized sites become weak points where polymer chains can break, releasing smaller fragments that bacteria can metabolize. Gas chromatography mass spectrometry detected these fragments in soil pore water samples, confirming that degradation was producing water-soluble compounds. Bacterial population dynamics revealed striking patterns. Quantitative PCR targeting the unique genetic barcodes in the engineered strains showed that Ideonella populations in Zone 1 had increased approximately tenfold from initial densities. Samples collected near plastic surfaces contained up to 100 million engineered cells per gram of biofilm material, while soil samples distant from plastic averaged only 500,000 cells per gram. This distribution demonstrated that bacteria were proliferating specifically where plastic substrates were available, exploiting the polymer as a carbon source for growth. The Pseudomonas bacillus consortium in Zone 2 showed even more dramatic expansion, with populations increasing 50-fold. The larger increase likely reflected the consortium's metabolic flexibility, able to utilize not just polyethylene, but also native organic matter present in the landfill. Bacillus populations slightly outnumbered Pseudomonas, possibly due to superior spore-forming capabilities that allowed survival through periods of moisture stress. Microscopy confirmed extensive biofilm development on polyethylene surfaces, with thick, multi-layered communities visible even at low magnification. Zone 3 presented a more complex picture. This zone received only purified enzymes, no living organisms. Weight loss in PET samples averaged 0.4%, about half that seen in Zone 1, despite similar initial enzyme concentrations. The difference reflected fundamental limitations of cell-free enzyme approaches. Enzymes gradually denature and lose activity in environmental conditions, degrading through temperature fluctuations, pH changes, and proteolytic attack by native soil microorganisms. Living bacteria continuously produce fresh enzymes, replacing those that denature. Without this renewal, enzyme activity inevitably declines. The biodegradable microspheres designed for slow enzyme release showed variable performance. Samples collected from Zone 3 contained microspheres at various degradation stages. Some remained intact, spherical beads approximately 50 micrometers in diameter. Others showed surface erosion, creating rough, irregular shapes. A few had completely dissolved, leaving only enzyme residues. The heterogeneous degradation reflected spatial variability in moisture, pH, and microbial activity across the zone. In wetter areas, microspheres degraded faster, releasing enzymes more rapidly but potentially too quickly for sustained activity. In drier areas, microspheres persisted, but enzyme release was slower, potentially too slow for maximum effectiveness. Zone 4, combining living organisms and supplemental enzymes, showed the highest overall activity. PET weight loss averaged 1.3%, exceeding either living organisms alone or enzymes alone. This synergy suggested that immediate enzyme activity jump-started degradation while bacterial populations established, and that the diversity of enzyme variants, some from bacteria and others supplemented, provided broader catalytic capability than either approach alone. Biofilm development appeared more robust in Zone 4 compared to Zone 1, possibly because supplemental enzymes created a richer mixture of degradation products that could support faster bacterial growth. Native microbial community responses became apparent through 16S ribosomal RNA gene sequencing. Control zones showed stable community composition over the 30-day period, with week-to-week variation below 5%, indicating that natural community dynamics were minimal on this timescale. Treatment zones showed measurable shifts. In Zone 1, several bacterial families increased in relative abundance, particularly those known to metabolize aromatic compounds like terephthalic acid. These native organisms apparently benefited from the degradation products released by Ideonella, exhibiting classic cross-feeding dynamics. In Zone 2, the shifts were more dramatic. Native actinobacteria, a phylum known for degrading complex organic molecules, increased from 8% to 15% of total community composition. Protobacteria, the phylum containing both Pseudomonas and Bacillus, rose from 25% to 42%, driven largely by the introduced consortium, but also including increases in native Protobacteria species. This suggested that the engineered organisms were not simply replacing native communities, but rather catalyzing broader changes that benefited functionally similar native species. Non-target effects monitoring focused on potential impacts beyond plastic degradation. Soil enzyme assays measuring general microbial activity, including tests for phosphatase, urease, and dehydrogenase, showed no significant differences between treatment and control zones. This indicated that overall soil function remained normal, without widespread disruption of nutrient cycling or other essential processes. Germination tests using radish seeds planted in soil from each zone showed 95% germination in all cases, no different from controls, demonstrating that the treated soils were not phytotoxic. Invertebrate surveys conducted by ecology team members captured and identified soil organisms from various zones. Nematodes, springtails, mites, and other microarthropods were extracted from soil samples and counted under microscopes. No significant differences in abundance or diversity were detected between treatment and control zones. This was reassuring evidence that the engineered bacteria and their metabolic activities were not harming these ecologically important organisms, at least not over the 30-day observation period. Chemical analysis of soil pore water revealed accumulation of degradation products. Terephthalic acid concentrations in Zone 1 averaged 12 micrograms per liter, compared to below detection limits in control zones. Ethylene glycol showed similar patterns, reaching 8 micrograms per liter in Zone 1. These concentrations remained well below toxicity thresholds established for aquatic organisms, several orders of magnitude lower in fact, suggesting no immediate environmental hazard. However, the monitoring protocol included tracking these compounds over time to ensure they did not accumulate to problematic levels as degradation continued. The leachate monitoring wells positioned downgradient of the treatment zones detected no engineered organisms during the 30-day period. Quantitative PCR assays sensitive enough to detect 100 cells per milliliter of water found no traces of the genetic barcodes. This confirmed that organisms were not spreading through groundwater flow, at least not yet. Soil gas measurements showed localized increases in carbon dioxide concentrations near plastic-rich areas in treatment zones, rising from 4% to 6% at depths where the engineered organisms were most active. The increase reflected heightened microbial respiration as bacteria metabolized plastic-derived carbon, converting it to carbon dioxide. While this represented a greenhouse gas emission, the quantities were minuscule compared to natural soil respiration rates. Statistical analysis of all 30-day data confirmed that the treatments produced measurable effects distinguishable from natural variability. Analysis of variance comparing weight loss across zones yielded highly significant results, with p-values below 0.01. The confidence intervals around mean weight loss estimates were relatively narrow, indicating that spatial variability within zones was manageable and that the experimental design provided adequate statistical power. Linear regression models exploring relationships between bacterial abundance and plastic weight loss showed positive correlations, as expected if bacterial activity drove degradation. Yet amid the encouraging results, caution was warranted. 30 days represented a tiny fraction of the multi-year study timeline. Many environmental processes show initial bursts of activity that later plateau or decline. Bacterial populations might crash if they exhausted easily accessible plastic or encountered resource limitations. Enzymes might denature faster than anticipated. Native communities might mount competitive or antagonistic responses that suppressed engineered organism populations. Weather patterns, particularly summer heat and drought, might stress the organisms beyond their tolerance limits. The research team convened to review the 30-day data and plan subsequent sampling. Dr. Chen emphasized the need to avoid over-interpreting preliminary results, reminding her colleagues that scientific history was littered with promising early findings that did not hold up over time. But she also acknowledged that the data provided reason for measured optimism. The engineered organisms had survived, proliferated, and begun degrading their target plastics. No obvious negative impacts on soil ecology had manifested. The monitoring systems were functioning as designed, generating high-quality data that would support rigorous analysis. The experiment was working, at least for now, and the coming months would reveal whether these early indicators foreshadowed meaningful long-term plastic remediation or merely a transient effect that would fade as environmental reality imposed its constraints on human engineering ambitions. Chapter 9, TSDB-D2 Ocean's Edge. The research vessel pitched rhythmically in the North Pacific swell, 1,100 nautical miles northeast of Hawaii. Around it stretched an expanse of cobalt water, interrupted by patches of debris, plastic bottles bobbing alongside fishing nets, styrofoam fragments, and countless unidentifiable pieces weathered by months or years of ocean exposure. This was the eastern margin of the Great Pacific Garbage Patch, a massive accumulation zone where ocean currents converge and trap floating materials in a slow clockwise gyre. The patch spans an area exceeding 1.6 million square kilometers, roughly 3 times the size of France, though its boundaries shift with seasonal current patterns. The magnitude of plastic in this single gyre is staggering. Surveys estimate 100,000 tons of floating plastic, composed of approximately 1.8 trillion individual pieces. Contrary to popular imagination, this is not a solid island of trash, but rather a diffuse soup, where plastic concentration varies from several kilograms per square kilometer in peripheral regions to 40 or 50 kilograms per square kilometer in the densest accumulation zones. Most pieces are microplastics, fragments below 5 millimeters, created through the breakdown of larger items under the relentless forces of sunlight, wave action, and temperature cycling. Testbed 2 sought to determine whether biological plastic degradation could complement mechanical cleanup efforts. The Ocean Cleanup Project and similar initiatives use floating barriers and collection systems to capture plastic from the gyre, removing thousands of tons since operations began. But mechanical collection faces fundamental limitations. It captures mainly larger debris while missing microplastics that slip through collection nets. It requires continuous operation, with ships consuming fuel and generating carbon emissions. It cannot address the vast quantities of plastic that have already sunk below the surface, an estimated 14 million tons resting on the seafloor. Biological degradation offered potential solutions to these limitations. Microbes and enzymes could theoretically attack microplastics too small for nets to capture. They could function continuously without energy input beyond sunlight and naturally occurring nutrients. If engineered organisms could degrade plastics in the water column or on the seafloor, they might address the invisible pollution that mechanical methods cannot reach. But marine environments presented challenges absent from terrestrial applications: salt tolerance, cold temperature adaptation, dispersal by currents, and exposure to a vastly larger and more complex ecosystem. The experimental design for the Ocean Gyre testbed combined collected plastic with engineered biological treatments, testing degradation under controlled conditions before any open water release. The research vessel carried floating mesocosms, sealed containers, each holding 1,000 liters of seawater and 5 kilograms of plastic collected from the gyre. The mesocosms maintained natural conditions, temperature, salinity, pH, and light, while preventing organisms and plastic from dispersing into the open ocean. 3 biological approaches would be tested. The first used marine bacterial strains isolated from plastic debris collected months earlier during preliminary surveys. These native ocean bacteria, primarily Vibrio alginolyticus and Marinobacter species, possessed natural plastic degrading capabilities verified through laboratory screening. They had been cultured, characterized genomically, and stored frozen until the expedition. The second approach employed genetically modified versions of these marine bacteria, engineered to express enhanced PTase and polyethylene-degrading enzymes. The third tested purified enzyme cocktails formulated for stability in seawater, packaged in slow-release alginate beads that would float near the surface where plastic concentration was highest. Dr. James Park, the marine microbiologist leading Testbed 2, explained the rationale for using native marine bacteria as starting material rather than terrestrial species like Ideonella sacchaensis. Marine bacteria have evolved for hundreds of millions of years in saltwater, developing sophisticated adaptations for osmotic balance, ion regulation, and tolerance of ionic stress that seawater imposes. Introducing terrestrial bacteria into the ocean would subject them to osmotic shock as water rushed into cells to equilibrate salt concentrations, likely killing them within hours. Even if some survived, they would face competition from trillions of native marine microbes perfectly adapted to ocean conditions. The engineering process involved identifying plastic-degrading genes from terrestrial bacteria, isolating the DNA sequences encoding PTase and related enzymes, then inserting those genes into marine bacterial chromosomes using molecular cloning techniques. The recipient marine bacteria thus gained new enzymatic capabilities while retaining their native salt tolerance, cold adaptation, and ecological fitness for ocean life. Directed evolution was applied to optimize enzyme performance in seawater, selecting for variants that maintained activity at pH 8.1, the slightly alkaline condition of seawater, and at temperatures ranging from 2 to 20 degrees Celsius to accommodate the range of ocean environments. The mesocosms were deployed at dawn, lowered from the ship's stern on cables to float at depths where they received full sunlight but remained submerged enough to avoid wave impacts. Each mesocosm was equipped with sensors monitoring temperature, salinity, pH, dissolved oxygen, and light levels. Sampling ports allowed collection of water and plastic samples without opening the containers. The experiment would run for 90 days, significantly longer than the 30-day landfill assessment. To accommodate the slower metabolic rates expected in cold, nutrient-poor ocean water. Initial plastic characterization revealed the harsh reality of ocean agging. Most collected debris showed extensive weathering. PET bottles were cracked and brittle, their surfaces roughened by sandblasting from suspended sediments. Polyethylene bags had fragmented into strips and shreds, many hosting visible biofilms, greenish-brown coatings of algae, bacteria, and diatoms. Polypropylene rope and netting displayed fraying and fiber separation. Infrared spectroscopy detected oxidation products in all plastic types, confirming that photodegradation from ultraviolet exposure had modified the polymer surfaces. This weathering was actually advantageous for biological degradation. Fresh plastic is chemically inert, but UV-weathered plastic contains carbonyl groups and other oxygen-bearing functional groups that provide enzymatic attachment points. Laboratory studies confirmed that marine fungi and bacteria preferentially colonize and degrade UV-weathered plastic compared to fresh samples. The ocean gyre, paradoxically, might be an ideal location for biological degradation, precisely because plastic has resided there long enough to undergo substantial photodegradation. The native marine bacteria inoculation into mesocosm 1 introduced approximately 1 billion cells per liter, concentrations determined from laboratory optimization experiments. The genetically modified marine bacteria went into mesocosm 2 at similar densities. Mesocosm 3 received enzyme beads, small spheres of alginate polymer containing immobilized PTase and lactases at total concentrations of 50 milligrams of enzyme per liter of seawater. Mesocosm 4 served as an untreated control, containing only collected plastic in natural seawater to measure baseline degradation rates from native microbial communities already colonizing the debris. Molecular tracking systems for the genetically modified bacteria employed fluorescent protein markers rather than just DNA barcodes. The engineered marine bacteria had been modified to express green fluorescent protein, causing them to glow under ultraviolet light. This allowed rapid visual assessment using fluorescence microscopy, complementing quantitative PCR measurements. Samples viewed under UV illumination showed glowing cells clustered on plastic surfaces, confirming colonization without requiring DNA extraction and amplification. The ocean environment immediately imposed challenges. Water temperature in the mesocosms fluctuated between 12 and 18 degrees Celsius, following daily and weather-driven patterns. These temperatures were far below the optimal ranges for terrestrial plastic-degrading bacteria, but normal for temperate ocean waters. The engineered marine bacteria had been selected for cold tolerance, but enzyme kinetics inevitably slow at reduced temperatures, a thermodynamic reality that no amount of engineering can fully overcome. Dissolved oxygen levels remained high, near saturation, supporting aerobic metabolism. However, nutrient concentrations were extremely low. Inorganic nitrogen measured below 1 microgram per liter, and phosphorus was similarly scarce, characteristic of the oligotrophic open ocean. This nutrient limitation represented a fundamental constraint. Bacteria require nitrogen for synthesizing proteins, including the degradative enzymes essential for plastic breakdown. They need phosphorus for DNA, RNA, and energy-carrying molecules like ATP. Without adequate nitrogen and phosphorus, even if carbon is abundant from plastic, bacterial growth will be severely restricted. Some research groups have explored nutrient supplementation to accelerate marine plastic degradation, but adding nutrients to the open ocean raises environmental concerns about stimulating harmful algal blooms or disrupting natural nutrient cycles. Early sampling at day 14 revealed modest bacterial colonization. Plastic pieces removed from mesocosms 1 and 2 showed biofilm development visible under scanning electron microscopy, with bacterial cells forming monolayers covering perhaps 20% of available surface area. This was far less extensive than biofilms observed in the landfill testbed, where nearly complete coverage occurred within 30 days. The difference reflected the nutrient-poor ocean environment, where bacterial growth rates are inherently slow. Fluorescence microscopy confirmed that genetically modified bacteria in mesocosm 2 were present and viable, showing green fluorescence on plastic surfaces. Population estimates from quantitative PCR indicated stable cell numbers, roughly 100 million cells per gram of biofilm, but not the dramatic proliferation seen in the landfill. The bacteria were surviving but not thriving, maintained by slow metabolism of plastic-derived carbon while limited by nitrogen and phosphorus scarcity. Enzyme bead performance in mesocosm 3 was mixed. Many beads floated as designed, concentrating near the water surface where plastic density was highest. However, some beads exhibited faster dissolution than anticipated, releasing enzymes in a burst rather than gradually. Alginate stability depends strongly on divalent cation concentrations, particularly calcium and magnesium, both abundant in seawater. The high calcium levels cross-linked the alginate more extensively than in laboratory tests conducted in distilled water, making some beads brittle and prone to crumbling. Others showed the opposite problem, becoming so stable they released enzymes too slowly for measurable activity. Despite these challenges, day 14 samples showed hints of degradation. PET bottles from mesocosm 2 exhibited slightly increased surface roughness when examined by atomic force microscopy, a technique that maps surface topography with nanometer resolution. The roughness suggested enzymatic etching, though the effect was subtle. Weight measurements were inconclusive at this early time point, with no statistically significant differences between treatment and control mesocosms. Fourier transform infrared spectroscopy detected no clear changes in polymer chemistry, likely because degradation was limited to thin surface layers constituting less than 1% of total mass, below the detection threshold of bulk spectroscopic methods. At day 45, the midpoint assessment revealed clearer patterns. Mesocosm 2, with genetically modified bacteria, showed the most promising results. PET samples exhibited average weight loss of 0.3%, small but statistically significant compared to the control mesocosm, which showed no weight loss. Polyethylene samples lost 0.2% of mass. More importantly, scanning electron microscopy revealed extensive surface pitting and crack formation, visual evidence that polymer degradation was progressing beyond just surface colonization. Mesocosm 1, with native unmodified bacteria, showed intermediate results. Weight loss was minimal, below 0.1%, but biofilm coverage had increased substantially, now covering approximately 60% of plastic surfaces. The native bacteria appeared to be establishing robust populations even without enhanced degradative enzymes, likely utilizing the naturally occurring biofilm matrix and adsorbed organic matter as primary carbon sources while slowly degrading plastic as a secondary substrate. The control mesocosm showed surprising activity. Weight loss in PET samples averaged 0.15%, indicating that native microbial communities colonizing the collected plastic before the experiment began were contributing to degradation. Metagenomic sequencing of biofilms from control plastic identified diverse bacterial genera, including some with genes similar to known plastic-degrading enzymes. This confirmed that natural biodegradation occurs in the ocean, albeit slowly, and that collected plastic hosts microbial communities already engaged in polymer breakdown. Chemical analysis of seawater from the mesocosms detected degradation products. Terephthalic acid concentrations in mesocosm 2 reached 3 micrograms per liter, elevated compared to less than 0.5 micrograms per liter in the control. Ethylene glycol showed similar patterns. Additionally, researchers detected a suite of shorter-chain organic acids and alcohols consistent with polyethylene oxidation. The appearance of these compounds in the water column confirmed that degradation was producing soluble products released from the plastic surfaces. Concerns emerged about microplastic generation. Samples of seawater filtered through fine meshes captured substantially more microplastic particles in treatment mesocosms compared to controls. Particle counts increased from approximately 50 particles per liter in the control to 120 particles per liter in mesocosm 2. Scanning electron microscopy and infrared spectroscopy confirmed that these particles were plastic fragments, not mineral particles or organic detritus. The elevated microplastic generation suggested that bacterial degradation might initially fragment large plastic pieces before completely mineralizing them, potentially worsening the microplastic problem before solving it. This observation sparked intense discussion among the research team. Microplastics pose serious environmental threats, accumulating in marine food webs and potentially transporting toxic chemicals or pathogenic microbes. If biological degradation accelerates microplastic production without achieving complete mineralization on reasonable timescales, it might cause more harm than benefit. Further analysis would be needed to determine whether microplastic generation was a transient phase, eventually followed by complete degradation, or a persistent side effect that would disqualify the technology for ocean application. At day 90, the final sampling provided a more complete picture. Cumulative weight loss in mesocosm 2 reached 0.9% for PET and 0.7% for polyethylene, roughly triple the 30-day rates extrapolated linearly. The acceleration suggested that bacterial populations and enzymatic activity were increasing over time rather than plateauing, a positive indicator for sustained long-term degradation. Biofilm architecture had evolved into complex three-dimensional structures with clear channels and void spaces, characteristic of mature bacterial communities. Microplastic concentrations in mesocosm 2 peaked at day 60, then declined by day 90, falling to 80 particles per liter. This pattern suggested that the initial fragmentation phase was indeed followed by degradation of the generated microplastics, supporting the hypothesis of transient rather than persistent microplastic accumulation. However, the day 90 concentration remained above control levels, indicating incomplete microplastic clearance within the experimental timeframe. Mesocosm 3, the enzyme-only treatment, showed limited effectiveness. Cumulative PET weight loss reached just 0.4%, and polyethylene showed no significant degradation. Post-experiment analysis of recovered enzyme beads revealed that most had completely dissolved, releasing their enzyme cargo within the first month. The enzymes exhibited rapid activity loss in seawater, with measured half-lives of only 7 to 10 days. This was far shorter than laboratory predictions based on freshwater testing, highlighting the harsher conditions of the marine environment where salt stress and wave action accelerate enzyme denaturation. The 90-day experiment generated critical data, but also exposed the gulf between mesocosm experiments and true open ocean deployment. Mesocosms provided controlled conditions and prevented organism dispersal, but they were tiny compared to the vastness of the Pacific Gyre. Scaling from 1,000-liter containers to the 2 trillion liters of the Gyre presented mathematical and practical impossibilities. Producing and distributing enough bacteria or enzymes to treat such volumes exceeded any realistic manufacturing capability. A different deployment strategy would be necessary. Rather than treating the entire Gyre, a more feasible approach might involve treating plastic during mechanical collection. As ships gathered debris, it could be transferred to onboard bioreactors, where controlled degradation would occur under optimal conditions before any organisms contacted the open ocean. Alternatively, enzymatic treatment could be applied to collected plastic that would then be contained in floating enclosures, allowing degradation while preventing organism escape. These approaches would eliminate concerns about uncontrolled spread while enabling biological treatment of ocean plastic at scales that might meaningfully reduce accumulation. As the mesocosms were retrieved and the research vessel turned toward port, Dr. Park reflected on what the experiment revealed. Biological plastic degradation could occur in marine environments, but slowly and incompletely over the 90-day period. The genetically modified bacteria showed promise, outperforming both native bacteria and enzyme treatments. But translating mesocosm results to practical ocean cleanup faced formidable obstacles: nutrient limitation, slow growth rates, temperature constraints, and the sheer scale of the problem. The ocean testbed had generated more questions than answers, exactly what good science should accomplish. The challenge now was determining whether any deployment scenario could responsibly and effectively apply these biological tools to the vast plastic-choked expanse of the world's oceans. The ocean is not merely cold water with dissolved salt. It is a fundamentally different chemical environment than freshwater or terrestrial habitats, presenting challenges that require specialized adaptations at every level of biological organization. For bacteria accustomed to the relatively benign conditions of soil or laboratory culture media, sudden immersion in seawater is often fatal. The high salt concentration creates osmotic stress that draws water out of cells, causing them to shrivel and lose turgor pressure necessary for maintaining cell structure and metabolic function. Engineering microorganisms to survive and function in marine environments demanded understanding and replicating the sophisticated mechanisms that native ocean bacteria evolved over billions of years. Seawater contains approximately 35 grams of dissolved salts per liter, dominated by sodium chloride, but also including significant concentrations of magnesium, calcium, sulfate, and bicarbonate ions. This creates an osmotic environment roughly 1,000 milliosmoles, 10 times higher than the cytoplasm of most terrestrial bacteria, which maintain internal salt concentrations around 100 milliosmoles. When a freshwater bacterium encounters seawater, water molecules rush out through the cell membrane, following the osmotic gradient from lower to higher solute concentration. The cell dehydrates, metabolic enzymes lose function, and DNA becomes damaged as water that normally stabilizes molecular structures disappears. Halophilic bacteria, those adapted to high-salt environments, employ two fundamentally different strategies for osmotic balance. The salt-in strategy, used by some archaea and a few bacterial lineages, involves accumulating potassium chloride inside the cell to concentrations exceeding 5 molar, matching or exceeding the external salt concentration. This eliminates the osmotic gradient, preventing water loss. However, it requires that all intracellular proteins and nucleic acids function in these extreme ionic conditions. Saltin organisms have extensively modified their entire proteome, with proteins enriched in acidic amino acids that remain soluble at high salt concentrations. This strategy represents such a radical departure from normal cellular biochemistry that engineering it into organisms lacking these adaptations is impractical. The salt-out strategy, employed by most marine bacteria, offers a more tractable engineering target. These organisms maintain relatively low intracellular salt concentrations but synthesize or accumulate compatible solutes, organic molecules that can reach high concentrations without interfering with cellular functions. Common compatible solutes include glycine betaine, ectoine, proline, and trehalose. These molecules are small, highly soluble, and chemically inert, serving purely as osmotic ballast to balance external salt without disrupting enzyme function or membrane integrity. The research team, led by Dr. Park, focused on marine Vibrio and Marinobacter species as chassis organisms, bacterial platforms into which plastic-degrading genes could be inserted. These genera are cosmopolitan in ocean environments, thriving from polar to tropical waters and across the full range of marine habitats from surface waters to deep-sea sediments. Their genomes contain multiple genes for compatible solute synthesis and transport, providing robust osmotic regulation. Whole genome sequencing of selected isolates revealed complete pathways for glycine betaine synthesis from choline, ectoine synthesis from aspartate, and trehalose synthesis from glucose. These pathways could be further enhanced through overexpression of key enzymes, boosting compatible solute production beyond natural levels to provide additional osmotic protection for genetically modified strains facing the stress of expressing foreign proteins. Temperature adaptation presented an equally critical challenge. Ocean temperatures vary dramatically with latitude and depth. Surface waters in tropical regions may reach 30 degrees Celsius, while Arctic seas hover near -2 degrees, kept liquid only by salt depression of freezing point. The Great Pacific Garbage Patch occupies temperate latitudes where surface temperatures range from 12 to 20 degrees Celsius seasonally, with deeper waters colder still. Enzymes optimized for mesophilic bacteria growing at 37 degrees, the standard laboratory temperature, would function poorly or not at all in this cold environment. Psychrophilic bacteria, those adapted to cold environments, produce enzymes with structural modifications that maintain flexibility and catalytic activity at low temperatures. Crystal structures of cold-adapted enzymes reveal characteristic features: increased surface loops that remain flexible even when molecular motion slows at low temperature, reduced numbers of stabilizing salt bridges and hydrogen bonds that would rigidify the structure, and active site architectures optimized for substrate binding at reduced thermal energy. These modifications come with trade-offs. Cold-adapted enzymes are typically less thermally stable than their mesophilic counterparts, beginning to denature at temperatures around 20 to 30 degrees, where mesophilic enzymes function optimally. The enzyme engineering strategy employed directed evolution, specifically selecting for cold activity. The p-tase gene from Ideonella sarcaensis was subjected to random mutagenesis, then expressed in the marine Marinobacter host strain at 10 degrees Celsius. Only variants retaining significant activity at this reduced temperature were selected for subsequent rounds of mutation and screening. After 8 generations of directed evolution, the team identified a p-tase variant with approximately 60% activity at 10 degrees compared to the wild-type enzyme's activity at 40 degrees. This represented a substantial improvement, though still slower than optimal mesophilic activity, reflecting the thermodynamic reality that chemical reactions inherently proceed more slowly at lower temperatures. Additional modifications improved enzyme stability in seawater. The high ionic strength and presence of divalent cations like magnesium and calcium can interfere with enzyme function by competing for metal binding sites or inducing aggregation. Computational modeling identified surface residues that could be modified to reduce nonspecific ion binding without disrupting catalytic function. Substituting several lysine and arginine residues, positively charged amino acids that attract negatively charged ions, with uncharged serine and threonine reduced salt-induced aggregation. These modifications were validated through in vitro assays measuring enzyme activity across a range of salt concentrations from 0 to 1.5 molar sodium chloride, confirming enhanced performance at seawater salinity. Ultraviolet radiation represents a particularly insidious threat to marine microorganisms and proteins. In terrestrial environments, atmospheric ozone absorbs most UVB radiation, the wavelengths between 280 and 320 nanometers that cause severe DNA damage. But UV transmission through the atmosphere varies with latitude, elevation, and ozone concentration. In the open ocean, especially at low latitudes, surface waters receive intense UV exposure that can penetrate several meters depth, depending on water clarity. UV radiation damages cells through multiple mechanisms. Direct absorption by DNA creates cyclobutane pyrimidine dimers, aberrant chemical bonds between adjacent thymine or cytosine bases that distort the double helix and block replication and transcription. UV also generates reactive oxygen species, highly reactive molecules including hydroxyl radicals and superoxide that attack lipids, proteins, and nucleic acids indiscriminately. For engineered bacteria deployed at or near the ocean surface, UV exposure could rapidly inactivate populations unless protective mechanisms were in place. Marine bacteria have evolved diverse UV protection and repair strategies. Some produce UV-absorbing pigments, carotenoids, or melanin-like compounds that absorb harmful radiation before it reaches sensitive cellular components. Others possess highly efficient DNA repair systems. Photolysis enzymes directly reverse cyclobutane pyrimidine dimers using energy from visible light, a process called photoreactivation. Nucleotide excision repair systems cut out damaged DNA segments and resynthesize the correct sequence using the complementary strand as template. Some bacterial species exhibit remarkable UV resistance through combinations of these mechanisms. Studies of bacteria isolated from high-altitude lakes in the Andes Mountains, environments with extreme UV exposure due to thin atmosphere and high elevation, identified particularly robust repair systems. Acinetobacter johnsoniana strain A2, isolated from a lake at 4,560 meters elevation, demonstrated extraordinary UVB resistance, surviving doses that killed most tested strains. Genomic analysis revealed multiple photolysis genes, redundant photoreactivation capacity, along with highly expressed nucleotide excision repair genes. When these genes were transferred into the marine Marinobacter chassis and tested under simulated ocean surface UV levels, survival increased approximately threefold compared to the unmodified strain. Buoyancy control emerged as an unexpected but critical consideration for marine deployments. Plastic debris floats or sinks depending on polymer density, degree of biofouling, and whether items contain trapped air. Polyethylene and polypropylene, with densities below seawater, float naturally and dominate surface accumulations like the Great Pacific Garbage Patch. PET, polystyrene, and most other plastics have densities exceeding seawater and should sink, yet substantial quantities float because they are manufactured into shapes, bottles, containers that trap air. Biofilm formation dramatically alters plastic buoyancy. Initial colonization by bacteria increases density slightly as biomass accumulates on surfaces. More significant effects come from subsequent colonization by diatoms, photosynthetic microalgae with silica cell walls that add substantial mineral weight. Laboratory experiments documented that polyethylene samples initially floating at the surface became negatively buoyant and sank after developing dense diatom biofilms over several weeks. This biofouling-driven sinking may explain the mysterious disappearance of vast quantities of plastic that should be accumulating at ocean surfaces but cannot be accounted for in surveys, possibly resting on the seafloor at depths beyond routine sampling. For engineered plastic degradation, this buoyancy dynamic created a dilemma. If bacterial colonization caused plastic to sink before substantial degradation occurred, debris would be transporting to deep, cold, dark waters where metabolic activity would slow dramatically and where monitoring would become nearly impossible. Conversely, if degradation proceeded faster than sinking, the approach might successfully treat surface plastic. Modeling these competing processes required understanding colonization kinetics, degradation rates, and biofouling dynamics across different plastic types and ocean conditions. Experiments in the mesocosms provided preliminary data. Polyethylene samples colonized by engineered Marinobacter remained positively buoyant throughout the 90-day experiment despite visible biofilm formation. The bacterial biofilms were relatively thin, typically 10 to 50 micrometers thick, adding minimal mass compared to dense diatom biofilms that can exceed 1 millimeter. However, samples deployed in mesocosms with added nutrients showed increased phytoplankton growth, including diatoms that colonized plastic surfaces. These samples exhibited decreased buoyancy, with some sinking by day 60. The results suggested that in natural ocean conditions, where nutrient inputs vary spatially and seasonally, buoyancy dynamics would be complex and potentially unpredictable. Nutrient limitation represented perhaps the most fundamental constraint on marine bioremediation. The open ocean away from coastal regions is essentially a nutrient desert for nitrogen and phosphorus. Nitrogen concentrations in surface waters of oligotrophic gyres often measure below 1 microgram per liter, and phosphorus even lower. These concentrations are approximately 1,000th of what would be considered adequate in terrestrial or freshwater systems. Marine bacteria have adapted through extreme efficiency, possessing high-affinity transporters that can scavenge nutrients at nanomolar concentrations, and through the ability to enter dormant states when nutrients become truly unavailable. But scavenging dilute nutrients costs energy that must come from metabolism. If bacteria invest more energy acquiring nutrients than they gain from degrading plastic, they cannot grow, and populations will not expand. This thermodynamic constraint might fundamentally limit plastic degradation rates in the open ocean regardless of enzyme engineering. Experiments measuring bacterial growth on plastic as the sole carbon source in artificial seawater with varying nutrient concentrations confirmed this limitation. At nitrogen concentrations below 5 micrograms per liter, growth was undetectable even with excess plastic substrate available. Above 20 micrograms per liter, growth proceeded but remained slow, with doubling times exceeding 1 week. Some researchers proposed nutrient supplementation to overcome this limitation. Adding nitrogen and phosphorus to plastic-rich areas could theoretically accelerate bacterial growth and degradation. However, such interventions risk triggering harmful algal blooms, explosive growth of photosynthetic organisms that can produce toxins and deplete oxygen when they die and decompose. Algal blooms in nutrient-enriched waters can kill fish and other marine life across vast areas, creating dead zones where oxygen depletion makes water uninhabitable. The cure might be worse than the disease. An alternative approach involved engineering bacteria to fix atmospheric nitrogen, converting N2 gas into ammonia that cells can use for biosynthesis. Nitrogen fixation is energetically expensive, requiring 16 ATP molecules per nitrogen molecule fixed, but frees organisms from dependence on dissolved nitrogen compounds. Several marine cyanobacteria naturally fix nitrogen, using energy from photosynthesis to power the process. Transferring nitrogen fixation genes into heterotrophic plastic-degrading bacteria proved challenging because the nitrogenase enzyme complex is oxygen-sensitive, and most plastic degradation occurs through aerobic pathways requiring oxygen. Researchers explored engineering compartmentalization strategies, creating intracellular microenvironments with reduced oxygen where nitrogenase could function while the rest of the cell conducted aerobic metabolism. Initial results showed proof of concept, but activity levels remained far below those needed for practical applications. Dispersal by ocean currents added another layer of complexity. Marine bacteria are not autonomous swimmers over large scales. Their flagellar motility allows movement over micrometers to centimeters, useful for chemotaxis toward nutrient sources or plastic surfaces, but inconsequential for transport across kilometers. Ocean currents dictate bacterial distribution, carrying cells passively like any other particle. In the Great Pacific Garbage Patch, currents move at typical velocities of 0.1 to 0.3 meters per second, transporting water and everything in it tens of kilometers per day. This dispersal creates a fundamental difference between terrestrial and marine bioremediation. In a landfill, bacteria remain relatively localized, moving slowly through soil pores and spreading only as far as groundwater flow carries them, typically meters per year. In the ocean, a bacterial population released in one location will be scattered across thousands of square kilometers within weeks. Monitoring such dispersed populations becomes extraordinarily difficult. Even with genetic barcodes and sensitive PCR detection methods, finding and quantifying bacteria at concentrations below 10 cells per liter in the vastness of the ocean pushes technical limits. Biodegradable carrier matrices offered potential solutions to multiple challenges simultaneously. Encapsulating bacteria in polymer microspheres could provide protection from UV radiation, salt stress, and predation while controlling buoyancy and release kinetics. The microspheres could be designed to float, keeping bacteria near the surface where plastic concentration is highest, or to have neutral buoyancy for treating plastic throughout the water column. Slow degradation of the carrier material would gradually release bacteria as the matrix eroded, extending the active lifetime of a single inoculation. Alginate, a polysaccharide extracted from brown seaweeds, emerged as a leading candidate material. Alginate forms gels in the presence of divalent cations like calcium, allowing encapsulation through simple mixing of bacterial suspensions with alginate solution followed by droplet formation in calcium chloride. The resulting beads are biocompatible, non-toxic, and degrade through natural processes in seawater over periods ranging from days to months, depending on alginate molecular weight and cross-linking density. Moreover, alginate is abundant and inexpensive, potentially allowing production of carrier beads at scales relevant for ocean application. Tests of alginate-encapsulated bacteria in the mesocosms showed promising results. Beads containing engineered Marinobacter protected cells from UV radiation, with survival rates 4 times higher than unencapsulated controls after exposure to simulated ocean surface UV for 5 days. The beads floated as designed, remaining in the top 20 centimeters of water where they encountered the highest plastic concentrations. Bacterial release occurred gradually as alginate degraded, maintaining viable cell concentrations in the surrounding water over the 40-day lifetime of the beads. Most encouragingly, bacteria released from degrading beads successfully colonized plastic surfaces in the mesocosms, demonstrating that encapsulation and release did not impair colonization capability. Yet even with all these adaptations, salt tolerance, cold activity, UV resistance, buoyancy control, the engineered bacteria faced the inescapable reality of the marine environment's vastness and complexity. The ocean contains approximately 1.3 billion cubic kilometers of water. The Great Pacific Garbage Patch alone encompasses roughly 5 million cubic kilometers. Treating even 1% of this volume would require producing and dispersing bacterial populations numbering in the tens of billions of cells, quantities that strain imagination and manufacturing capacity alike. The mesocosm experiments, contained and controlled, had generated valuable scientific insights, but they also illuminated the enormous gulf between demonstrating technical feasibility and achieving practical implementation at ocean scale. The question was not whether engineered bacteria could survive and degrade plastic in seawater. The experiments confirmed they could. The question was whether any deployment scenario existed that could meaningfully reduce the oceanic plastic burden without triggering ecological disruptions that might prove worse than the problem itself. Chapter 11: The Gyre Experiment. 3 months into the mesocosm experiments, as promising preliminary data accumulated, the research team faced a critical decision. Should they proceed to limited open water testing, releasing engineered bacteria into the actual ocean environment of the Great Pacific Garbage Patch? The scientific rationale was compelling. Mesocosms, despite careful design, remained artificial. They excluded predators, particularly protozoa and bacteriophages, that consume bacteria in nature at rates approaching 50% of daily bacterial production. They simplified chemical complexity, using filtered seawater that lacked the full spectrum of organic compounds, trace metals, and dissolved substances present in the real ocean. Most critically, they eliminated the physical dynamics of wave action, current shear, and vertical mixing that constantly stress planktonic organisms. Real ocean conditions might dramatically alter bacterial performance in ways mesocosms could not predict. Yet releasing genetically modified organisms into the open ocean crossed a threshold that evoked intense regulatory scrutiny and public concern. Previous releases of engineered bacteria into the environment, all terrestrial applications, had generated controversy despite extensive containment measures. An ocean release would involve a medium with essentially no physical containment. Currents would disperse bacteria across vast areas. Marine life might ingest them. They could potentially reach shorelines thousands of kilometers from the release point. The decision required balancing scientific necessity against ecological precaution, knowledge against uncertainty. The compromise strategy involved ultra-limited releases coupled with intensive monitoring. The team would deploy not thousands of liters of bacterial suspension across expansive areas, but rather 10 small enclosures, each containing 1 cubic meter of seawater and known quantities of plastic and bacteria. The enclosures, constructed from fine mesh that allowed seawater exchange while retaining plastic and preventing bacterial escape, would float freely in the gyre, drifting with currents but maintaining physical containment. GPS trackers attached to each enclosure would enable recovery after 60 days for comprehensive analysis. This approach allowed testing under real ocean conditions, temperature fluctuations, wave action, UV exposure, while maintaining containment comparable to mesocosms. Preparing the experimental enclosures required solving numerous engineering challenges. The mesh had to be fine enough to prevent bacteria from passing through, necessitating openings below 1 micrometer, yet open enough to allow water exchange that would maintain realistic chemical conditions. Complete water stagnation would rapidly deplete oxygen and accumulate metabolic wastes, creating artificial conditions no better than mesocosms. The solution involved double-walled construction, an inner wall of 1 micrometer mesh containing bacteria and plastic, surrounded by an outer wall of 1 millimeter mesh that protected the fine inner mesh from tearing while allowing substantial water flow. Buoyancy had to be carefully balanced. The enclosures needed to float semi-submerged, with approximately 80% of their volume underwater to maximize contact between contained seawater and surrounding ocean, while keeping the top portion above surface for GPS antenna function and to prevent complete submersion during storms. Ballast weights made from biodegradable iron compound, chosen to avoid adding persistent pollutants, were attached to the bottom of each enclosure. Floats made from closed-cell foam topped the structure. The resulting assemblies resembled small cages approximately 1 meter cubed, weighing 30 kilograms, including contained water and plastic. Deployment occurred on a clear morning in early autumn, when seasonal weather patterns provided a statistical window of reduced storm probability. The research vessel positioned itself in the densest accumulation zone of the garbage patch, where floating plastic concentration exceeded 20 kilograms per square kilometer. Each enclosure was filled with local seawater pumped from 3 meters depth, then loaded with 5 kilograms of plastic debris collected from the surrounding patch over previous days. The plastic mix reflected natural composition, approximately 60% polyethylene, 20% polypropylene, 10% PET, and 10% mixed polymers, including polystyrene and nylon. 4 enclosures received the engineered Marinobacter strain, inoculated at 1 billion cells per liter. 3 enclosures got enzyme-loaded alginate beads containing no living bacteria, allowing comparison between biological and purely enzymatic approaches. 3 served as controls, containing only the plastic in unmodified seawater, measuring natural degradation rates. Each enclosure was labeled with a unique identifier, documented photographically, and released to drift freely. GPS transmitters began broadcasting positions every 4 hours, data relayed through satellite links to servers accessible by the research team. The enclosures dispersed rapidly. Within 24 hours, the cluster had spread across 5 square kilometers. After 1 week, distance between the most separated enclosures exceeded 50 kilometers. The GPS data revealed complex trajectories reflecting the chaotic nature of gyre circulation. Some enclosures spiraled in tight loops, caught in eddy currents. Others moved relatively linearly, carried by stronger flow features. 2 enclosures moved toward each other over 3 weeks, coming within 2 kilometers, then diverged and ultimately separated by over 100 kilometers. This dispersion pattern starkly illustrated the challenge of tracking and managing anything released into the ocean. Even with satellite tracking, locating and recovering small floating objects across tens of thousands of square kilometers of open water required substantial vessel time and fuel. Environmental sensors in the enclosures transmitted data alongside GPS positions, building real-time pictures of conditions. Water temperature fluctuated between 13.5 and 17.8 degrees Celsius over the 60-day period, showing both diurnal cycles and weather-related variations as clouds blocked solar heating or clear skies allowed intense warming. Dissolved oxygen remained high, 90 to 100% saturation, throughout the experiment, indicating that water exchange through the mesh maintained aerobic conditions. pH showed minimal variation, staying between 8.0 and 8.2, typical of surface seawater. UV intensity, measured by light sensors on enclosure tops, varied dramatically with time of day and weather, from near zero at night to peak values approaching 15 watts per square meter of UVB during midday clear conditions. The first sampling occurred at day 30, requiring the research vessel to navigate to each enclosure using GPS coordinates. Sea conditions with 2-meter swells made retrieval challenging. Each enclosure was hoisted aboard, sampled through sterile ports without opening the mesh, then returned to the ocean to continue the experiment. The sampling process took 3 days to cover all 10 enclosures scattered across thousands of square kilometers. Samples were processed immediately in the ship's laboratory, analyzing bacterial populations, plastic condition, and water chemistry before specimens could degrade. Initial results from the bacterial enclosures showed both successes and setbacks compared to mesocosm predictions. Quantitative PCR detected engineered Marinobacter in all 4 bacterial enclosures, confirming survival over 30 days. However, population sizes had decreased substantially. Inoculation densities of 1 billion cells per liter had dropped to between 50 and 200 million cells per liter, representing 70 to 95% mortality. This die-off exceeded mesocosm observations, where populations had remained relatively stable. Post-hoc analysis attributed the difference to predation, likely by heterotrophic nanoflagellates, single-celled protists that consume bacteria and were excluded from mesocosms but present in natural seawater entering the enclosures. Microscopic examination of plastic samples revealed biofilm development, but at lower densities than in mesocosms. Bacterial cells covered approximately 10 to 20% of available plastic surfaces in the ocean enclosures, compared to 40 to 60% coverage observed in mesocosms at equivalent time points. Scanning electron microscopy showed diverse bacterial morphologies in the biofilms, including the distinctive rod shape of the engineered Marinobacter, but also cocci, spirals, and filamentous forms representing native marine bacteria that had colonized the plastic despite the enclosure mesh. Apparently, some native bacteria present as free-living cells in seawater drawn into the enclosures or as rare colonizers on the collected plastic had proliferated over 30 days. Weight loss in PET samples from bacterial enclosures averaged 0.2%, less than the 0.3% observed in mesocosms at 30 days. Polyethylene showed similar patterns, 0.1% loss compared to 0.2% in mesocosms. The reduced degradation rates correlated with lower bacterial densities, supporting the hypothesis that predation pressure in natural conditions limited bacterial populations and consequently degradation activity. Control enclosures showed 0.05% weight loss in PET, attributable to native bacterial activity from the collected plastic and seawater, while polyethylene showed no detectable degradation in controls. The enzyme bead enclosures produced unexpected results. 2 of 3 enclosures showed degradation rates comparable to bacterial treatments, with PET losing 0.2% and polyethylene 0.1%. This matched the mesocosm observation that purified enzymes could achieve degradation without living bacteria, albeit with limited duration due to enzyme denaturation. However, the third enzyme enclosure showed dramatically higher activity, with PET losing 0.8%, 4 times the rate of the other enzyme enclosures. Investigation revealed that this enclosure had experienced colonization by native marine bacteria that apparently utilized the degradation products released by the enzymes, creating an unplanned synergistic system. Infrared spectroscopy of plastic samples confirmed chemical degradation. PET from bacterial enclosures showed reduced intensity of ester carbonyl peaks at 1,715 wave numbers and increased hydroxyl peaks at 3,300 wave numbers, consistent with hydrolytic cleavage of polymer chains. Samples from the high-activity enzyme enclosure showed even more pronounced spectral changes, supporting the weight loss data indicating enhanced degradation. Control enclosure plastics showed minimal spectral changes, confirming that the treatments were responsible for observed degradation rather than purely physical or photochemical processes. Microplastic analysis revealed a troubling pattern. Water samples filtered through 20-micrometer mesh captured elevated concentrations of plastic particles in treatment enclosures compared to controls. Bacterial enclosures contained an average of 300 particles per liter, enzyme enclosures 250 particles per liter, and controls 50 particles per liter. The particles ranged from 20 micrometers, the filter cutoff, to several hundred micrometers, visible under low magnification microscopy. Polymer identification by infrared microspectroscopy confirmed they were PET and polyethylene fragments, not phytoplankton or mineral particles. This microplastic generation raised immediate concerns. If biological degradation broke large plastic items into thousands of microplastic particles before completely mineralizing them, the treatment might worsen pollution by creating particles small enough for widespread ingestion by marine organisms. Microplastics are consumed by zooplankton, fish larvae, filter-feeding invertebrates, and many other organisms, entering food webs and potentially concentrating as they move up trophic levels. They can carry toxic chemicals leached from the plastic or absorbed from seawater, potentially delivering these contaminants directly to tissues of organisms that ingest them. The final sampling at day 60 provided a more complete temporal picture. Bacterial populations in the enclosures had stabilized rather than continuing to decline, ranging from 40 to 150 million cells per liter. Apparently, a balance had been reached between bacterial growth and predation losses. Biofilm coverage on plastic surfaces had increased to 30 to 50%, approaching mesocosm levels, though this coverage included substantial native bacterial biomass alongside the engineered strain. Fluorescence microscopy using the green fluorescent protein marker allowed distinguishing engineered cells, revealing they comprised only 20 to 40% of total biofilm bacteria, with native organisms dominating. Cumulative plastic weight loss over 60 days reached 0.4 to 0.6% for PET and 0.2 to 0.4% for polyethylene in bacterial enclosures. These rates were approximately half those observed in parallel mesocosms run for 60 days, confirming that real ocean conditions reduced degradation efficiency. The enzyme enclosures showed variable performance, with the two standard activity enclosures achieving 0.3 to 0.4% PET loss, while the high-activity outlier reached 1.4%. Control enclosures showed cumulative degradation below 0.1%, demonstrating that natural processes alone produced minimal degradation over this timescale. Critically, microplastic concentrations in treatment enclosures peaked around day 40, then began declining. By day 60, concentrations had dropped to approximately 150 particles per liter in bacterial enclosures and 100 particles per liter in enzyme enclosures, still elevated compared to 50 particles per liter in controls, but lower than the day 30 peak. This decline suggested that the initial phase of fragmenting large plastic into microplastics was followed by degradation of those microplastics, supporting the hypothesis of transient rather than permanent microplastic accumulation. Longer-term experiments would be needed to determine whether microplastic concentrations would eventually fall below control levels as degradation proceeded to completion. Chemical analysis of water inside the enclosures detected degradation products at elevated concentrations. Terephthalic acid in bacterial enclosures averaged 12 micrograms per liter, elevated compared to below 1 microgram per liter in controls. Ethylene glycol, shorter-chain carboxylic acids, and alcohols consistent with polyethylene oxidation were also detected. These compounds represented the breakdown products of polymer degradation, confirming that the observed weight loss reflected true chemical decomposition rather than just physical fragmentation. Toxicity bioassays using water from the enclosures to culture marine diatoms and copepods showed no adverse effects at the concentrations present, suggesting the degradation products were not acutely toxic to these representative marine organisms. Enclosure recovery revealed biological surprises. Several enclosures hosted small fish that had somehow entered despite the fine mesh, probably as larvae smaller than the mesh openings that subsequently grew. The fish appeared healthy, actively swimming and showing no obvious abnormalities. Dissection and gut content analysis found microplastics in the digestive tracts, confirming ingestion, but no evidence of immediate harm. The small sample size precluded statistical conclusions, but the observations underscored that any ocean intervention occurs within complex food webs where organisms at all levels will interact with and potentially be affected by the introduced elements. The Gyre experiment generated invaluable data while simultaneously highlighting profound challenges. The engineered bacteria survived and functioned in real ocean conditions, degrading plastic at measurable rates. This proof of principle confirmed technical feasibility. However, degradation rates were slower than mesocosm predictions. Bacterial populations declined due to predation. Microplastic generation raised new environmental concerns, and the logistical complexity of deploying and monitoring even 10 small enclosures across a vast oceanic area was substantial. Scaling from experimental enclosures to treating meaningful portions of the garbage patch would require not merely multiplying effort, but rethinking deployment strategies entirely. The ocean had delivered its verdict. Biological plastic degradation could work, but not easily, not quickly, and not without risks that demanded careful evaluation before any larger-scale implementation could be responsibly contemplated. Chapter 12: The Living City. 8,000 kilometers from the Pacific garbage patch, in a coastal city of 400,000 residents, another experiment was beginning. This was Testbed 3, the urban pilot program integrating engineered plastic degradation directly into municipal waste management infrastructure. Unlike the remote landfill or distant ocean gyre, This testbed unfolded in densely populated human spaces where thousands of people would encounter the technology daily through routine trash disposal, recycling, and wastewater treatment. The proximity demanded unprecedented attention to public communication, safety assurance, and transparent operation. The city, which agreed to participate on condition of anonymity until results could be evaluated, produced approximately 500 tons of municipal solid waste daily, typical for a population of this size. Roughly 130 tons, 26%, consisted of various plastics, the single largest material category by weight. Existing recycling captured approximately 30% of plastic waste, leaving 90 tons daily destined for landfills or incineration. This plastic stream represented both a challenge and an opportunity. As a challenge, it embodied the persistent accumulation problem facing cities worldwide. As an opportunity, it provided a concentrated, accessible substrate for testing biological degradation under conditions far more controlled than either landfills or oceans. The integration strategy focused on 3 intervention points in the urban waste cycle. First, selected public waste receptacles in parks, transit stations, and municipal buildings would be fitted with bioactive liners containing immobilized plastic-degrading enzymes. These liners would begin degradation immediately upon trash deposition, treating plastic during the hours or days it remained in the bin before collection. Second, a dedicated processing facility would receive plastic waste from commercial recycling programs, treating it with engineered bacterial consortia in controlled bioreactors before mechanical recycling. Third, the municipal wastewater treatment plant would incorporate plastic-degrading bacteria into tertiary treatment stages, targeting microplastics that enter sewage from laundry fibers, cosmetic microbeads, and degraded plastic products. The public engagement process began 18 months before any organisms were deployed. The city convened a sustainability advisory board, recruiting 15 residents representing diverse demographics, neighborhoods, and professional backgrounds. The board received extensive education about plastic pollution, synthetic biology, the specific organisms and enzymes to be used, monitoring protocols, and potential risks. Early meetings revealed significant concern and misunderstanding. One board member worried that bacteria in trash bins would spread disease, conflating engineered plastic degraders with human pathogens. Another feared that enzymes could damage the recycling facility, dissolving plastic pipes or containers. A third questioned whether the entire program was simply greenwashing, corporate-funded public relations to deflect criticism rather than genuine environmental improvement. The research team and city officials addressed these concerns through multiple channels. Public webinars explained the difference between pathogenic bacteria causing disease and environmental bacteria engineered for plastic degradation, emphasizing that the organisms were more similar to soil bacteria or probiotics than to disease-causing species. Laboratory demonstrations showed that the enzymes specifically attacked waste plastic but not structural materials or functioning items. Financial disclosures revealed funding sources and potential conflicts of interest, establishing that while one corporate recycling partner contributed resources, the research was directed by university scientists with no financial stake in commercial outcomes. Trust built gradually. By month 6, public opinion surveys showed 61% of residents supported the pilot program, 23% remained uncertain, and 16% opposed it. Crucially, opposition decreased as information availability increased, suggesting that transparency and education effectively addressed concerns. The Sustainability Board voted 11-4 to recommend proceeding with the pilot, with the dissenting members citing precautionary principle arguments that any environmental release of engineered organisms posed unacceptable risks, regardless of claimed safeguards. Installation of bioactive waste bin liners began in a phased rollout. The liners were fabricated from biodegradable polylactic acid polymer embedded with enzyme-loaded microspheres similar to those tested in ocean trials. The polylactic acid provided mechanical strength, holding trash and preventing leakage, while slowly degrading over weeks to months depending on moisture and temperature. Embedded microspheres released PTase, cutinase, and lactase enzymes as the liner degraded. Each liner cost approximately $8 to manufacture, significantly more than conventional plastic liners costing 20 cents, but the city committed to absorbing this cost for the one-year pilot. 20 waste bins in 5 city parks received bioactive liners in the initial phase. Each bin was clearly labeled with signage explaining the pilot program and providing website links for more information. Monitoring teams collected contents weekly, separately analyzing plastic items for degradation and measuring enzyme activity in the used liners. Control bins with conventional liners were maintained in the same parks for comparison. The strategy minimized risk. If problems emerged, only 20 bins were affected and could be immediately switched back to conventional liners. The first month produced encouraging but modest results. Plastic bottles and containers deposited in bioactive bins showed surface modifications visible under microscopy, including increased roughness and early-stage crazing consistent with enzymatic attack. Weight loss averaged 0.5% for PET bottles over 1 week of bin residence before collection. Conventional control bins showed no measurable plastic degradation over the same period. Importantly, no adverse effects were observed. The liners functioned mechanically as intended, containing waste without tearing or leaking. No unusual odors developed. Monitoring for bacterial contamination found no elevation in the bins compared to background levels, confirming that the enzyme-only approach avoided concerns about microbial spread. Expansion proceeded to Phase 2, adding 100 bins across 20 locations including schools, shopping districts, and transit hubs. Usage patterns varied dramatically by location. Bins at fast food restaurants filled rapidly with plastic cups, lids, and straws, reaching capacity within hours. Park bins filled more slowly over days, containing mixed waste from picnics and recreational activities. Transit station bins received steady inputs of beverage bottles and takeout containers throughout the day. This variability affected degradation outcomes. Rapidly filling bins provided minimal contact time between waste and liner enzymes, limiting degradation to less than 0.2%. Slowly filling bins allowed up to 1 week of contact, achieving 0.5 to 0.8% degradation. Citizen feedback was solicited through multiple mechanisms. QR codes on bin signage linked to online surveys asking residents to report observations and opinions. A dedicated email address received comments and questions. The Sustainability Board held monthly public sessions where anyone could attend and speak. Feedback was generally positive, with notable exceptions. Several restaurant owners complained that the bioactive bins cost significantly more than conventional waste management, but provided no obvious benefit to their businesses. A parent questioned whether children playing near the bins faced exposure to engineered enzymes. An environmental advocacy group praised the initiative, but argued it distracted from more important interventions like reducing single-use plastic production. The bioreactor facility represented the program's most technically sophisticated component. Located at the Municipal Recycling Center, the system processed approximately 5 tons of plastic waste per day, focusing on PET bottles collected through residential curbside programs. Incoming bottles were sorted, washed, and shredded into flakes approximately 5 millimeters across. The flakes entered bioreactors, large stainless steel tanks containing 5,000 liters of circulating water maintained at 25 degrees Celsius. The engineered bacterial consortium, combining enhanced Ideonella sacchaensis with supporting species, was continuously cultured in separate fermentation tanks and pumped into the bioreactors at controlled rates. The bioreactor environment was optimized for rapid degradation. Temperature was held constant rather than fluctuating with weather. pH was buffered at 7.5, optimal for enzyme activity. Dissolved oxygen was maintained at 80% saturation through forced aeration. Nitrogen and phosphorus were added as nutrients to support bacterial growth, eliminating the limitation that constrained ocean trials. Under these ideal conditions, degradation proceeded far faster than in landfill or ocean test beds. After 48 hours of bioreactor treatment, PET flakes lost an average of 12% of their mass. The degradation products, primarily terephthalic acid and ethylene glycol, dissolved in the reactor water. These could be recovered through chemical extraction and purification, potentially yielding monomers suitable for repolymerizing into virgin-quality PET. This closed-loop recycling offered economic advantages over conventional mechanical recycling, which progressively degrades polymer quality through repeated melting and reforming cycles. Preliminary economic modeling suggested that if degradation efficiency could be increased to 20% mass loss per 48 hours, the recovered monomers would offset operational costs, making the bioprocess economically competitive with mechanical recycling. However, challenges emerged. Maintaining stable bacterial populations in continuous flow bioreactors proved difficult. Every time treated water was drained and replaced with fresh influent, a portion of the bacterial population was lost. Continuous culture techniques compensated by adding fresh bacteria from the fermentation tanks, but this required substantial resources for bacterial production. Equipment fouling also posed problems. Biofilms formed not just on plastic flakes, but on reactor walls, pipes, and pumps, necessitating weekly cleaning that temporarily halted operations. Contamination by native bacteria from inadequately sterilized plastic flakes occasionally disrupted the engineered consortium, requiring system purging and restart. The wastewater treatment integration targeted a different plastic problem: microplastics. Municipal wastewater contains surprisingly high concentrations of plastic particles, typically 1 to 10 particles per liter, originating from synthetic textile fibers shed during laundry, microbeads from cosmetics and toothpaste, and degraded fragments from countless sources. Conventional wastewater treatment removes some microplastics through settling and filtration, but substantial quantities pass through into effluent discharged to rivers or coastal waters. Studies have found that wastewater treatment plants are major point sources of microplastic pollution to aquatic environments. The pilot program added a biological treatment stage following conventional secondary treatment. Effluent from secondary clarifiers, already substantially cleaned of organic matter and pathogens, but still containing microplastics, was diverted to a polishing pond. This shallow, constructed wetland, covering 2 hectares, contained aquatic plants, sediments, and a microbial community augmented with engineered plastic-degrading bacteria. The retention time was 5 days, allowing bacteria ample opportunity to colonize suspended microplastics and initiate degradation. Initial assessments showed modest microplastic reduction. Influent to the polishing pond contained an average of 3.7 particles per liter. Effluent exiting to the receiving river contained 2.1 particles per liter, representing 43% removal. However, distinguishing degradation from settling was difficult. Some microplastics likely sank into bottom sediments rather than being degraded. Careful measurement of sediment accumulation over 6 months would be needed to quantify this partitioning. Importantly, no increase in microplastic concentration occurred, ruling out concerns that bacterial activity might fragment larger particles faster than degrading smaller ones within the pond environment. Integration across the 3 urban intervention points created opportunities for system-wide assessment. Mass balance calculations tracked plastic entering the city through commerce, being used and discarded by residents and businesses, moving through waste and recycling streams, and ultimately exiting as recycled material, degraded products, or persistent waste. The biological interventions affected only small fractions of total flows during the pilot year. Bioactive bins treated less than 1% of plastic waste. Bioreactors processed 5% of collected PET bottles. The wastewater polishing pond treated 100% of municipal sewage, but achieved only partial microplastic removal. Despite these limited scales, system-wide impacts were detectable. The combination of interventions reduced plastic mass reaching the municipal landfill by an estimated 3.2%, approximately 150 tons annually. Economic analysis indicated a net cost of approximately $400,000 for the pilot year, combining infrastructure investment, operational expenses, and monitoring, against benefits of approximately $70,000 from reduced landfill fees and recovered monomers from bioreactor treatment. This negative return reflected pilot-scale inefficiencies and the conservative deployment limited to testing rather than optimization. Projections suggested that scaling to treat 50% of plastic waste could achieve cost neutrality if degradation efficiencies improved as anticipated. The urban testbed demonstrated that biological plastic degradation could be integrated into existing city infrastructure without major disruption. Residents accepted the technology when it was explained transparently and implemented gradually. Technical performance under controlled conditions, particularly in bioreactors, far exceeded what was achieved in landfills or oceans, confirming that environmental control enabled faster degradation. Yet the pilot also revealed practical limitations. The technology remained expensive, required continuous monitoring and maintenance, and achieved only modest reductions in plastic waste at scales tested. Whether biological degradation could evolve from promising demonstration to standard practice in waste management depended on continued optimization, cost reduction, and most critically, on confronting the environmental risks that any widespread deployment of engineered organisms inevitably entails. That question, the central tension underlying all three testbeds, would soon demand answers as data accumulated and decisions about scaling loomed on the horizon. Cities function as vast metabolic systems, consuming materials and energy while producing waste and emissions. Understanding these flows, the inputs of food, water, fuel, and goods, the transformation through use and consumption, and the outputs of sewage, garbage, and pollution reveals how urban areas operate as living systems at grand scale. For plastic, the metabolic pathway is particularly linear. Approximately 460 million tons enter global commerce annually, flowing into cities where they serve brief purposes, packaging a meal, holding a beverage, wrapping a product, then exit as waste within days or weeks. This rapid throughput creates enormous waste streams that overwhelm collection and disposal infrastructure in cities worldwide. The pilot city generated comprehensive data on plastic metabolism through its urban environment. Point-of-entry tracking documented plastic arriving through wholesale distribution channels, retail shipments, and consumer purchases. Estimates based on sales data indicated approximately 48,000 tons of plastic entered the city annually, translating to 132 tons daily. This included packaging materials, 32,000 tons, consumer products containing plastic components, 11,000 tons, and durable goods like furniture and electronics, 5,000 tons. The packaging category dominated, reflecting the pervasive use of single-use plastics in food service, retail, and distribution systems. Waste collection systems captured approximately 70% of plastic waste generated by residents and businesses. The remaining 30% followed various paths. Some accumulated in homes and businesses as stored items or clutter, effectively removed from circulation, but not yet waste. Some entered informal recycling through scavenging and resale. Some leaked into the environment through littering, illegal dumping, or inadvertent loss during transport. Storm drains represented a particularly significant leakage pathway. Plastic debris on streets and parking lots washed into storm sewers during rain events, ultimately discharging to the river that flowed through the city. Before the pilot program, an estimated 250 tons of plastic annually reached the river through this pathway, contributing to downstream pollution and eventually ocean accumulation. Bioswales offered a nature-based solution to this urban runoff problem. These engineered landscape features, vegetated channels designed to manage stormwater, capture and filter runoff before it enters storm drains. Traditional bioswales rely on physical filtration through soil and plant roots, removing sediments, metals, and organic contaminants with efficiencies ranging from 50 to 90%, depending on design and maintenance. The pilot program enhanced 12 bioswales across the city, with plastic-degrading bacteria incorporated into the soil matrix. The engineered bioswale design involved careful consideration of hydrology, plant selection, and microbial ecology. Storm runoff enters the bioswale through inlet structures that reduce flow velocity, allowing suspended particles, including plastic debris, to settle. Water percolates through 75 centimeters of engineered soil, composed of sand, compost, and mineral components, formulated to support both plant growth and microbial activity. Deep-rooted native plants, selected for pollution tolerance and ability to thrive in alternating wet-dry cycles, stabilize the soil while creating aerobic zones around root systems where bacterial degradation proceeds most efficiently. The bacterial augmentation involved mixing engineered Bacillus and Pseudomonas strains into the bioswale soil during construction. Bacillus was chosen for its spore-forming capability, allowing survival through drought periods common in urban landscapes where irrigation may be inconsistent. Pseudomonas provided robust plastic degradation enzymes and ability to form biofilms on plastic surfaces trapped in the bioswale. Initial inoculation densities of 10 million cells per gram of soil were maintained through the top 30 centimeters, where most plastic would accumulate. Monitoring bioswale performance required measuring plastic capture efficiency and degradation rates simultaneously. Plastic litter was inventoried before and after storm events by carefully excavating and sieving soil samples from designated areas. The mass of captured plastic typically ranged from 50 to 300 grams per bioswale per storm, depending on catchment area size and storm intensity. This plastic was predominantly polyethylene bags and films, polypropylene food wrappers, and polystyrene foam fragments, the lightweight materials most easily transporting by runoff. After 6 months, samples of plastic recovered from bioswales showed measurable degradation. Infrared spectroscopy detected carbonyl peaks indicating oxidation, and weight measurements showed average reductions of 3 to 7% compared to fresh reference samples of the same polymer types. Control bioswales without bacterial augmentation showed minimal degradation, below 1%, attributable to photooxidation and physical weathering. The difference confirmed that engineered bacteria were enhancing degradation beyond natural processes. However, bioswale effectiveness was limited by hydraulic residence time. Storm events were brief, measured in hours, and while bioswales retained some water that infiltrated slowly over subsequent days, plastic trapped on the surface dried quickly and became inaccessible to bacteria, requiring moisture for activity. Degradation occurred primarily during the wet periods immediately following storms and in the continuously moist soil layers below the surface where buried plastic fragments resided. This temporal limitation meant that despite measurable degradation, bioswales could not completely mineralize plastic on timescales relevant to urban stormwater management. They functioned more as temporary retention systems that slowed plastic transport and initiated degradation rather than as complete treatment solutions. Separation and sorting challenges dominated the waste management system downstream of collection. Municipal solid waste arrived at transfer stations as heterogeneous mixtures containing plastic, paper, glass, metals, organic waste, and countless other materials. Automated sorting systems using optical sensors, air jets, and magnetic separators could recover approximately 60% of PET bottles and 40% of HDPE containers, the most valuable recyclables. But mixed plastics, multi-layer packaging, contaminated items, and film plastics passed through sorting systems into residual waste streams destined for landfills. The bioreactor facility received only pre-sorted PET from residential recycling programs, ensuring relatively clean feedstock. Even this selected stream required extensive washing to remove adhesives, labels, and organic residues before biological treatment. Washing consumed substantial water and energy, approximately 200 liters and 5 kilowatt-hours per kilogram of plastic, representing significant operational costs and environmental impacts. The wash water itself became a waste stream requiring treatment before discharge, as it contained detergents, sugars, proteins, and microplastics shed from the plastic items. After washing, shredding, and bioreactor treatment, the facility faced the critical challenge of monomer recovery. Terephthalic acid and ethylene glycol, released by bacterial degradation, dissolved in the reactor water at concentrations typically around 0.5 to 2 grams per liter. Recovering these compounds at high purity required multi-stage processing. Acidification precipitated terephthalic acid as a solid that could be filtered and washed. Distillation concentrated ethylene glycol from the remaining solution. These purification steps added complexity, energy consumption, and cost. The recovered monomer's quality fell slightly below virgin petrochemical standards. Terephthalic acid purity reached 98 to 99%, compared to 99.8% for fresh synthesis. Small quantities of colored compounds, likely breakdown products from dyes or additives in the original plastic, persisted through purification and imparted faint discoloration. For many applications, this purity was acceptable. But for premium products, like clear beverage bottles, manufacturers demanded higher standards that the biological process struggled to meet consistently. Economic viability calculations revealed the narrow margins determining success or failure. Processing costs for the bioreactor facility totaled approximately $850 per ton of PET treated, including labor, energy, chemicals, equipment maintenance, and bacterial culture production. At 12% degradation efficiency, each ton yielded 120 kilograms of recoverable monomers. Market prices for terephthalic acid averaged approximately $900 per ton, and ethylene glycol $1,200 per ton. The recovered value was approximately $110 per ton of input plastic, far below processing costs. Several pathways could improve economics. Increasing degradation efficiency to 25% would double monomer recovery, while processing costs increased only marginally, improving cost recovery to approximately 40%. Alternatively, treating lower-quality plastic feedstock that would otherwise be landfilled rather than mechanically recycled could reduce opportunity costs. If the comparison baseline was avoided landfill fees, approximately $60 per ton, rather than lost mechanical recycling value, approximately $200 per ton, the economics shifted favorably. Scaling operations to process 50 tons daily instead of 5 would reduce per-unit costs through economies of scale, potentially achieving cost parity with mechanical recycling. Life cycle assessment provided environmental accounting beyond simple economics. Biological plastic degradation consumed energy, primarily electricity for pumping, aeration, and temperature control in bioreactors, totaling approximately 600 kilowatt-hours per ton processed. If this electricity came from fossil fuel power plants, associated greenhouse gas emissions were approximately 300 kilograms of carbon dioxide equivalent per ton. Mechanical recycling consumed less energy, approximately 300 kilowatt-hours per ton, but produced lower-quality recycled plastic that could only be used in downgraded applications. The critical comparison was against virgin plastic production. Manufacturing PET from petroleum requires approximately 70 gigajoules per ton and generates approximately 2,700 kilograms of carbon dioxide equivalent. Biological degradation, followed by monomer repolymerization, consumed approximately 20 gigajoules per ton total and generated approximately 900 kilograms of carbon dioxide equivalent, representing 67% emissions reduction compared to virgin production. Even accounting for current inefficiencies, biological recycling offered climate benefits if it enabled recovery of plastic that would otherwise be landfilled or incinerated rather than recycled. Wastewater treatment plant integration demonstrated a different dimension of urban plastic metabolism. Sewage entering the plant contained microplastics at average concentrations of 4.3 particles per liter, originating from laundry discharge, degradation of plastic products during use, and runoff captured by combined sewer systems. Primary and secondary treatment removed larger particles through settling and filtration, but particles below 50 micrometers largely passed through. Daily effluent flow averaged 30 million liters, translating to approximately 60 million microplastic particles discharged to the river each day before the polishing pond was implemented. The polishing pond operated as a hybrid system combining physical settling, plant uptake, and microbial degradation. Surface area of 2 hectares and average depth of 1 meter provided 10,000 cubic meters of treatment volume. At 30 million liters per day flow, hydraulic residence time averaged approximately 8 hours. This was insufficient for complete microplastic degradation, which required days to weeks based on laboratory studies. But the pond did achieve measurable removal. Effluent concentrations averaged 2.4 particles per liter, representing 44% reduction. Distinguishing removal mechanisms was experimentally challenging. Sediment traps placed at various depths in the pond captured plastic particles, confirming that settling removed some fraction, estimated at 20 to 30%. Plant root systems hosted dense bacterial biofilms containing the engineered plastic-degrading strains. Verified by quantitative PCR, microscopic examination of microplastics collected from pond effluent showed surface pitting and biofilm colonization, evidence of bacterial activity. The combined mechanisms achieved removal exceeding natural wetlands lacking bacterial augmentation, which typically achieve 20 to 30% microplastic reduction. IoT sensor networks deployed throughout the waste management system generated real-time data streams that transformed operational management. Smart waste bins equipped with ultrasonic fill sensors transmitted data every 6 hours, reporting bin capacity and alerting collection crews when bins approached 80% full. This demand-responsive collection replaced fixed schedules, reducing unnecessary truck trips by approximately 30% and associated fuel consumption and emissions. In bioactive bins, additional sensors monitored temperature and moisture, conditions affecting enzyme activity and degradation rates. Bioreactor facility incorporated comprehensive sensor arrays measuring temperature, pH, dissolved oxygen, bacterial cell density, and monomer concentrations in real time. Machine learning algorithms analyzed patterns in these data streams, identifying optimal operating conditions and predicting equipment failures before they occurred. For example, the system learned that dissolved oxygen below 4 milligrams per liter correlated with declining degradation rates, triggering increased aeration. Temperature fluctuations exceeding 2 degrees per hour indicated heat exchanger fouling, prompting maintenance before complete failure. Data integration across intervention points revealed system-level patterns invisible to isolated measurements. Seasonal variations in plastic waste generation showed peaks during summer months when outdoor activities and beverage consumption increased. Storm events caused spikes in bioswale plastic capture, followed by elevated microplastic concentrations at the wastewater treatment plant as urban runoff entered combined sewers. These patterns informed adaptive management, adjusting bacterial inoculation rates, bioreactor operating parameters, and monitoring intensity to match changing conditions. Building material applications represented an emerging frontier for integrating biological plastic management into urban fabric itself. Researchers explored incorporating plastic-degrading bacteria into the matrix of construction materials, creating infrastructure that could self-remediate plastic contamination or degrade at end of life rather than persisting indefinitely. Early experiments embedded bacterial spores in concrete used for trash enclosure walls. When plastic waste contacted the walls, moisture from the waste activated spores, which germinated and began degrading plastic in direct contact with the concrete surface. The concept extended to living building materials more broadly. Self-healing concrete containing limestone precipitating bacteria was already commercialized, using bacterial metabolism to seal cracks and extend service life. Engineered wood products could incorporate lignin-degrading fungi modified with plastic-degrading enzymes, creating materials that could break down plastic wastes used as fillers or composite components. Roof coatings formulated with slow-release biocide-free antimicrobial compounds could support plastic-degrading bacterial populations, treating microplastic pollution deposited from atmospheric fallout. These applications remained largely conceptual during the pilot year, tested in laboratory settings or small demonstrations rather than deployed at building scale. Regulatory approval for building materials containing genetically modified organisms would require extensive safety testing and public acceptance challenges, potentially exceeding those for waste management applications. Yet the vision of cities as ecosystems that actively remediate pollution through designed infrastructure rather than passively accumulating, it represented a profound shift in urban environmental management philosophy. The urban testbed revealed that biological plastic degradation could be woven into the metabolic fabric of cities through multiple intervention points. Each alone achieved modest impacts, but collectively they addressed plastic at multiple life stages: capture during use in smart bins, treatment during processing in bioreactors, removal from wastewater in polishing ponds, interception of runoff in bioswales. The approach was fundamentally different from landfill or ocean testbeds. Rather than releasing organisms into complex environments beyond human control, the urban strategy embedded biological treatment into engineered systems designed for control and monitoring. This offered greater safety and predictability, but required substantial infrastructure investment and continuous management. Whether cities would embrace this vision of living infrastructure that collaborates with engineered microorganisms to metabolize waste depended on demonstrating continued improvements in efficiency, cost-effectiveness, and public trust. Chapter 14: The Recycling Revolution. The crisis in plastic recycling preceded biological degradation technologies by decades. Mechanical recycling, the dominant approach since recycling programs began in the 1970s, faced fundamental limitations rooted in polymer chemistry and economics. Every time plastic passes through the recycling process, heat and mechanical stress during melting and reforming break polymer chains, a phenomenon called chain scission. The molecular weight decreases, and with it, the material properties that made the plastic useful originally. Strength declines, brittleness increases, color and clarity degrade. This progressive quality loss creates a phenomenon called downcycling. A PET bottle recycled once becomes fiber for carpets or clothing, applications tolerating lower material quality. Recycled again, it becomes plastic lumber or park benches, further down the value hierarchy. After 3 or 4 cycles, the degraded polymer is no longer suitable for any functional application and must be landfilled. The recycling label thus misleads. Most plastic recycling delays disposal rather than enabling true circularity, where materials cycle indefinitely without quality loss. Economic factors compound these technical limitations. Mechanical recycling requires costly sorting and cleaning infrastructure. Collection and transportation add substantial expenses. The resulting recycled plastic typically costs more than virgin plastic produced from petroleum, particularly when oil prices are low. Only regulatory mandates, extended producer responsibility laws, and consumer preference for recycled content maintain demand for mechanically recycled plastic despite its cost disadvantage and inferior quality. Consequently, global recycling rates remain dismally low. Of the approximately 6.3 billion tons of plastic waste generated since mass production began in the 1950s, only 9% has been recycled. 12% has been incinerated, often generating toxic emissions and greenhouse gases. The remaining 79%, nearly 5 billion tons, has accumulated in landfills, littered the environment, or polluted oceans. Production continues accelerating, reaching 460 million tons annually, ensuring that even if recycling rates improve, absolute quantities of plastic waste will continue growing. Biological degradation offers a fundamentally different approach that could sidestep mechanical recycling's limitations. Rather than melting and reforming polymers while accepting progressive quality loss, biological methods depolymerize plastic completely into original monomers. These monomers, terephthalic acid and ethylene glycol for PET, can be purified and repolymerized into virgin-quality plastic chemically identical to petroleum-derived material. The process enables true closed-loop recycling, where plastic cycles indefinitely without quality degradation. The chemistry is elegant. P-tase and MHE-tase enzymes hydrolyze ester bonds in PET, releasing monomers into solution. Filtration removes residual polymer and biomass. Crystallization purifies terephthalic acid. Distillation purifies ethylene glycol. The monomers return to polymerization reactors, where they are converted back to PET indistinguishable from virgin material. No chain scission occurs because polymer chains are completely disassembled, then rebuilt. Colorants and additives from the original plastic are separated during monomer purification, allowing production of clear, colorless recycled PET suitable even for food contact applications. Early industrial demonstrations validated the concept. Carbios, a French biotechnology company, constructed a pilot plant processing 16 tons of PET waste per year, achieving 97% conversion to purified monomers. The recovered terephthalic acid reached 99.8% purity, meeting food-grade standards. Coca-Cola and L'Oreal announced partnerships to use the biological recycling process for producing bottles from waste. These commercial validations, though modest in scale, demonstrated technical feasibility beyond laboratory proof of concept. The urban pilot bioreactor represented a micro-scale version of this industrial vision. At 5 tons per day capacity, the facility processed approximately 1,800 tons annually, producing roughly 215 tons of recoverable monomers at 12% efficiency. Scaling to industrial levels, processing 10,000 tons per day comparable to large mechanical recycling facilities would require bioreactors totaling 50 million liters volume, achievable through modular construction of multiple large vessels. Energy requirements for biological recycling fell between mechanical and chemical recycling methods. Mechanical recycling consumed approximately 300 kilowatt-hours per ton, mainly for sorting, washing, melting, and pelletizing. Chemical recycling, through pyrolysis or gasification, required 1,000 to 2,000 kilowatt-hours per ton due to high-temperature processing. Biological degradation consumed approximately 600 kilowatt-hours per ton for pumping, aeration, temperature control, and monomer separation, falling in the middle range. Carbon footprint calculations showed advantages over virgin production, but challenges compared to optimal mechanical recycling. Virgin PET production emitted approximately 2.7 tons of carbon dioxide equivalent per ton of plastic. Biological recycling emitted approximately 0.9 tons, a 67% reduction. Mechanical recycling emitted approximately 0.5 tons, achieving 81% reduction, superior to biological methods. However, mechanical recycling's quality limitations meant it could only partially substitute for virgin plastic in most applications. Biological recycling potentially enables 100% virgin plastic substitution, which changes the system-level accounting. Life cycle assessment, considering full supply chain impacts, revealed complexity. Growing feedstocks for bacterial culture production, typically glucose derived from corn or sugarcane, required agricultural land, water, fertilizers, and pesticides. These upstream impacts added environmental burdens absent from mechanical recycling. Conversely, biological recycling could process contaminated and mixed plastic streams rejected by mechanical recycling, potentially diverting material from incineration or landfilling and avoiding those environmental impacts. The critical comparison depended on alternative fate assumptions. If biological recycling substituted for mechanical recycling of clean PET bottles, environmental performance was approximately equivalent or slightly worse, depending on electricity sources. If it substituted for landfilling or incineration of contaminated plastic, environmental benefits were substantial, avoiding methane emissions from landfill decomposition or air pollution from incineration. If it enabled recycling of plastic currently not recyclable through any method, creating new circularity where none existed. Benefits were maximized. Economic modeling explored scenarios for achieving cost parity with virgin plastic production. At current efficiency of 12% and recovery costs of $850 per ton, biological recycling was not competitive, with virgin PET costing approximately $1,200 per ton to produce. Several pathways could close this gap. Enzyme improvements increasing efficiency to 30% would reduce per ton costs to approximately $400 while generating 360 kilograms of monomers worth approximately $320, achieving 75% cost recovery. Alternative configurations integrating biological and mechanical recycling could capture synergies. Mechanical recycling could handle clean, high-quality PET at lower cost, while biological recycling processed contaminated, mixed, and degraded plastics unsuitable for mechanical methods. This complementary approach would divert material currently landfilled, treating it biologically to recover monomers for reintroducing into virgin-quality production streams. The combined system achieves higher overall recycling rates than either method alone. Policy mechanisms strongly influenced economic viability. Extended producer responsibility laws requiring manufacturers to fund recycling infrastructure created dedicated funding streams. Landfill taxes and disposal fees increased costs of alternative waste management, improving competitive position of recycling. Recycled content mandates requiring products contain minimum percentages of recycled material created guaranteed markets. Carbon pricing, making virgin plastic production more expensive by internalizing climate costs, would significantly favor recycling methods with lower emissions. The urban pilot explored integration with existing waste management economics. The city paid approximately $60 per ton for landfill disposal of non-recycled plastic. Mechanical recycling generated revenue of approximately $200 per ton from selling bales of sorted plastic to reprocessors. The bioreactor facility, operating at 12% efficiency, generated monomer revenue of $110 per ton, but incurred costs of $850, requiring subsidy of $740 per ton. This subsidy exceeded landfill disposal costs, making biological treatment economically unattractive from narrow waste management accounting. However, broader accounting, including environmental externalities, shifted the calculation. Landfilling plastic generated negative externalities, including groundwater contamination risk, long-term land use impacts, and methane emissions contributing to climate change. Economists estimated these externalities at approximately $40 to $120 per ton, suggesting true social cost of landfilling was $100 to $180 per ton. Biological recycling avoided these costs while producing climate benefits from displaced virgin production valued at approximately $350 per ton, using conservative carbon prices of $40 per ton, CO2, including externalities. Biological recycling generated net social value despite requiring operational subsidies. Public willingness to pay for improved recycling remained uncertain. Surveys in the pilot city found that 71% of residents supported municipal investment in advanced recycling technologies, but support declined when specific tax increases were mentioned. 48% supported a $5 per household monthly increase in waste collection fees to fund biological recycling, while 31% supported a $10 increase. These responses suggested that modest cost increases would maintain public support, but extensive subsidies requiring larger fee increases might erode political viability. Corporate partnerships offered alternative funding models. Beverage companies committed to using recycled content bottles faced challenges sourcing sufficient high-quality recycled PET through mechanical recycling. Biological recycling, offering virgin-quality material from waste, aligned with corporate sustainability goals and brand positioning. Companies might pay premium prices for biological recycled content, perhaps 15 to 25% above virgin plastic, to meet recycled content targets and improve environmental credentials. Such premiums would substantially improve project economics, potentially eliminating need for public subsidies. The pilot year operations generated data informing these economic projections, but could not definitively resolve viability questions. Small-scale operations faced inherent inefficiencies from limited throughput, batch processing, and excess capacity in equipment sized for future expansion. Industrial-scale facilities would achieve lower per-unit costs through continuous processing, automation, and fuller utilization of capital equipment. Enzyme costs would decline as production scaled and directed evolution improved activity, potentially reducing biological catalysis costs by 70 to 85%. Market dynamics would also shift as biological recycling scaled. Large supplies of biological recycled monomers entering markets could depress prices, reducing revenue. Conversely, if biological recycling enabled substantial increases in overall recycling rates, reducing plastic pollution and improving corporate sustainability metrics, consumer and regulatory pressure might maintain premium pricing. The complex interplay of technology improvement, economies of scale, market evolution, and policy development would unfold over years, requiring adaptive management and continued innovation. Recycling revolution implied by biological methods remained aspirational during the pilot year. Technology demonstrated feasibility, but had not yet achieved the efficiency, scale, or cost-effectiveness required for widespread displacement of incumbent mechanical recycling or virgin plastic production. Yet the trajectory was promising. Enzymatic performance improved steadily through protein engineering. Understanding of optimal process conditions deepened through operational experience. Corporate and public interest in circular economy solutions created favorable conditions for continued development. Whether biological degradation would transform plastic recycling from incremental downcycling to true material circularity depended on navigating technical challenges, economic obstacles, and the ecological risks that any large-scale deployment of engineered organisms inevitably entails. Chapter 15. Scaling from pilot to citywide deployment. The 12-month pilot program generated mountains of data, thousands of measurements across 3 intervention systems, documenting bacterial populations, plastic degradation rates, enzyme activities, system costs, operational challenges, and public responses. Synthesizing these data into actionable recommendations for citywide scaling required careful analysis, separating genuine insights from artifacts of small-scale implementation, distinguishing permanent limitations from solvable problems, and projecting how technologies performing modestly at pilot scale might improve through optimization and scaling economies. The Sustainability Advisory Board received comprehensive results presentations during year-end review meetings. Dr. Chen and the research team did not sugarcoat challenges. Bioactive waste bin liners achieved only modest degradation, 0.5 to 0.8% over typical one-week residence times, far less than needed for meaningful waste reduction. Costs were prohibitive, $8 per liner compared to 20 cents for conventional bags, making citywide deployment economically untenable without dramatic cost reduction. Yet the technology demonstrated proof of principle. Enzymes remained active in real-world conditions and degraded plastic without adverse effects. The bioreactor facility results were more encouraging. 12% degradation efficiency and monomer recovery substantially exceeded targets set at project outset. Stable bacterial populations and consistent performance across 11 months, excepting 2 brief contamination events that required system restart, proved operational viability. Economic analysis projected that scaling to 50 tons per day processing and improving efficiency to 20% through continued enzyme engineering would achieve cost parity with mechanical recycling. Industrial partners expressed interest in co-investment for Phase 2 expansion. Wastewater polishing pond performance met expectations. 44% microplastic removal was not revolutionary, but represented meaningful improvement over conventional tertiary treatment, achieving 20 to 30% removal. Crucially, the system operated with minimal maintenance and integrated seamlessly into existing plant operations. Scaling required only constructing additional pond area, straightforward civil engineering without exotic equipment or complex operations. Cost projections suggested doubling pond size to 4 hectares would achieve 60% removal at incremental cost of $800,000 capital investment. Bioswale effectiveness was difficult to quantify conclusively. The systems clearly captured plastic debris that would otherwise enter storm drains, removing an estimated 12 tons annually from pilot implementation across 12 bioswales. Bacterial augmentation enhanced degradation, but determining how much plastic was degraded versus simply retained in soil for later release remained uncertain. Longer-term monitoring, 3 to 5 years, would be needed to resolve this question. Nevertheless, bioswales provided multiple co-benefits beyond plastic management, including stormwater infiltration, pollutant removal, urban greening, and aesthetic improvement. These co-benefits justified expansion even if plastic degradation proved modest. The board discussed scaling scenarios. Conservative expansion would increase bioactive bins to 500 units at high-traffic locations, double bioreactor capacity to 10 tons per day, expand wastewater ponds to 4 hectares, and construct 30 additional bioswales. This represented approximately tenfold increase from pilot scale, processing an estimated 15% of municipal plastic waste annually. Capital costs were projected at $12 million, with annual operating costs of $3 million. The conservative scenario minimized risk while generating data for evaluating further expansion. Aggressive expansion would deploy bioactive bins citywide, 10,000 units, scale bioreactors to 50 tons per day capacity, expand ponds to 20 hectares, and integrate bioswales into all new development projects through building code amendments. This would process approximately 60% of plastic waste, approaching comprehensive coverage. Capital costs exceeded $120 million, requiring bond financing or public-private partnerships. Aggressive scaling offered potential for transformative impact, but carried proportionally greater financial and technical risks. Board members raised critical questions. One asked whether environmental risks from releasing engineered organisms increased proportionally with scale or exponentially. Dr. Chen acknowledged uncertainty, but noted that containment in engineered systems, bioreactors and ponds differed fundamentally from environmental release. Another questioned whether focusing resources on plastic degradation addressed root causes or merely treated symptoms, while production and consumption continued unabated. The research team agreed that reducing plastic use remained paramount, but argued that dealing with accumulated and ongoing waste required complementary strategies, including advanced recycling. Public comment sessions revealed divided opinion. Environmental advocates split between those supporting technological innovation and those advocating exclusively for source reduction and redesign to eliminate single-use plastics. Business representatives generally supported expansion, citing economic development opportunities and corporate sustainability goals. Neighborhood groups raised concerns about siting bioreactor facilities and potential odors or truck traffic from expanded operations. These diverse perspectives reflected broader societal debates about technology's role in environmental problem-solving. The Sustainability Board voted on recommendations. 8 members supported conservative expansion, arguing it balanced continued innovation with prudent risk management. 5 supported aggressive expansion, contending that climate crisis and plastic pollution demanded bold action. 2 opposed any expansion, maintaining that resources should focus entirely on waste prevention rather than waste management. The 8-5-2 vote provided clear direction for conservative expansion, while acknowledging the passionate advocacy for more ambitious action. City Council received the board's recommendation along with technical reports, economic analyses, and public comment summaries. Political considerations entered the equation. Council members facing re-election campaigns viewed the pilot program's success as politically valuable, demonstrating responsiveness to environmental concerns. Yet the significant capital costs and uncertain long-term viability created political risk. After 3 contentious meetings, Council voted 6-3 to approve conservative expansion with provisions for annual performance reviews and authority to pause scaling if problems emerged. Implementation planning began immediately. Engineers designed the expanded bioreactor facility, determining optimal vessel configurations, piping systems, instrumentation, and analytical equipment. Procurement identified suppliers for specialized equipment, including high-efficiency aerators, membrane filtration systems, and automated monomer recovery units. Site preparation for expanded wastewater ponds required environmental permitting, soil testing, and coordination with existing plant operations. Regulatory approvals posed significant hurdles. Expanding beyond pilot scale required updated environmental assessments, addressing potential impacts from increased processing volumes, bacterial releases from equipment maintenance, and cumulative effects of organism dispersal. Federal oversight became relevant as scale approached thresholds, triggering national environmental policy review. The approval process consumed 8 months, during which project timelines slipped and costs escalated due to inflation and supply chain disruptions. Workforce development emerged as an unexpected challenge. Operating advanced biological waste treatment systems required personnel with training in microbiology, biochemistry, and process engineering, skills uncommon in traditional municipal waste management workforces. The city partnered with the local community college to develop a certificate program in environmental biotechnology, training operators for the expanded facilities. 23 students enrolled in the first cohort, drawn by relatively high-paying jobs in an emerging field. Community engagement continued as planning progressed. Monthly public meetings provided updates on design development, permitting, and timeline. Transparency about challenges including cost overruns and regulatory delays maintained trust even as frustrations mounted. Critics who had opposed expansion monitored for evidence of problems that might support calls for cancellation, while supporters advocated for faster implementation. Navigating these political dynamics required diplomatic skill and sustained commitment to inclusive decision-making. The expanded bioreactor facility commenced operations 18 months after council approval. Initial performance exceeded pilot facility results, with degradation efficiency reaching 15% within the first month of optimized operation. The larger scale enabled implementation of continuous flow processing rather than batch operation, improving enzyme utilization and reducing idle time. Bacterial culture production scaled efficiently, with fermentation costs per unit of culture decreasing approximately 40% due to larger vessel volumes. However, new challenges emerged. Contamination by native bacteria occurred more frequently in the larger facility, possibly due to increased difficulty maintaining sterility across extensive pipe networks and larger plastic feedstock volumes. Enzyme denaturation in holding tanks between processing batches required additional stabilizer compounds, increasing chemical costs. Fouling of membrane filtration systems used for monomer separation occurred faster than predicted, necessitating more frequent cleaning and membrane replacement. The first year of expanded operations processed 3,200 tons of PET waste, recovering 540 tons of monomers. This represented meaningful impact, diverting plastic from landfills and producing recycled content for new products. Yet it also represented less than 2% of total plastic waste generated citywide, underscoring the scale gap between pilot success and comprehensive solution. Closing that gap would require continued expansion, technological improvement, and ultimately deployment in numerous cities, creating distributed processing networks rather than relying on individual facilities. The wastewater pond expansion encountered fewer operational challenges. doubling surface area to 4 hectares and extending retention time to 14 hours increased microplastic removal to 62%, closely matching predictions. Bacterial populations in the pond remained stable through seasonal temperature variations ranging from 8 to 28 degrees Celsius, demonstrating robustness across environmental conditions. Cost-effectiveness calculations showed pond treatment at approximately $35 per kilogram of microplastic removed, expensive, but potentially justified by environmental benefits of preventing river and ocean pollution. Bioswale network expansion to 42 installations citywide captured an estimated 50 tons of plastic debris annually. Visual evidence of plastic retention in bioswale soil was unmistakable, with excavations revealing layers of fragmented bags, bottles, and wrappers integrated into the soil matrix. Degradation assessment remained challenging, but 3-year monitoring data showing progressive mass loss in plastic-contaminated soil samples suggested that long-term decomposition was occurring. The bioswales also generated positive feedback from residents who appreciated the aesthetic improvements and wildlife habitat they provided. System-wide integration enabled synergistic benefits not apparent from individual interventions. Data from smart bins informed bioreactor operations, with surges in PET bottle collection triggering adjustments in bacterial culture production to maintain optimal processing. Seasonal patterns in microplastic loading at the wastewater plant correlated with storm events captured by bioswales, improving understanding of urban plastic transport pathways. These insights enabled adaptive management, continuously refining operations based on real-world performance data. Transition from pilot to citywide deployment represented a critical phase in technology maturation. Pilot-scale success demonstrated feasibility and catalyzed public support and investment. Citywide implementation tested whether the technologies could function reliably at scales relevant to urban metabolism while navigating economic constraints, regulatory requirements, and operational complexity absent from sheltered pilot conditions. Results were mixed, but generally encouraging. Costs remained high but were declining. Performance improved as operators gained experience and engineers optimized designs. Most importantly, no adverse environmental impacts manifested, maintaining public trust essential for continued expansion. The city had become a living laboratory for integrating biological plastic management into urban infrastructure, generating insights that would inform decisions in cities worldwide contemplating similar paths. Chapter 16: Ecological Reckoning. The conference room fell silent as Dr. Margaret Thompson projected the worst-case scenario onto the screen. The senior ecologist had spent 3 decades studying invasive species, documenting how organisms introduced into new environments could cascade through ecosystems with devastating consequences. Now she applied that expertise to evaluating the risks of releasing plastic-degrading bacteria engineered in laboratories into landfills, oceans, and urban infrastructure. Her assessment was sobering. Every environmental release of a modified organism is an experiment in ecological dynamics, she began, her voice measured but grave. We can test in laboratories and mesocosms. We can model population growth and dispersal, but we cannot fully predict how engineered bacteria will interact with the billions of native species they will encounter in complex ecosystems. History teaches that confidence in our predictions often exceeds their accuracy. The risk assessment framework, developed for genetically modified organisms, provides structured methodology for evaluating potential hazards. Phase 1 requires identifying every characteristic of the modified organism that might cause adverse effects, comparing the engineered strain to its unmodified parent and to related organisms already present in the release environment. For the plastic-degrading bacteria, this inventory was extensive. The organisms carried genes for producing novel enzymes, P-tase, MHE-tase, enhanced cutenases that did not exist in their native forms. They possessed antibiotic resistance markers used during laboratory construction to select successfully modified cells. They had been optimized for rapid growth on plastic substrates, potentially giving them competitive advantages over native bacteria. Phase 2 evaluates the probability that identified hazards will actually occur in release environments. Some hazards might be theoretically possible, but practically unlikely given environmental conditions, competitive pressures, or physical constraints. Others might be not just possible, but probable, warranting intensive scrutiny and potentially disqualifying the organism from release. Dr. Thompson walked the assembled researchers and regulators through each probability assessment, forcing acknowledgement of uncertainties that optimistic project advocates had sometimes minimized. Horizontal gene transfer represents our highest probability concern, she stated, displaying a diagram showing how bacteria exchange genetic material through conjugation, transformation, and transduction. These processes enable bacteria to share genes not just with their offspring through reproduction, but with distantly related species through direct transfer. The plastic-degrading genes in engineered bacteria could potentially transfer to native soil, marine, or wastewater bacteria, spreading engineered capabilities beyond the introduced strains into wild populations. Horizontal gene transfer is not hypothetical. It drives the spread of antibiotic resistance, one of the most urgent threats to global health. Resistance genes that emerged in hospitals through antibiotic selection pressure have transferred to environmental bacteria, creating reservoirs of resistance in soil, water, and even wildlife that can transfer back to human pathogens. Monitoring soil samples from 1940 through 2008 documented dramatic increases in antibiotic resistance genes, with some increasing 15-fold, correlating with antibiotic use in medicine and agriculture. The same mechanisms enabling resistance spread could disseminate plastic-degrading genes. If plastic-degrading genes spread to native bacteria populations, we lose control, Dr. Thompson emphasized. Genes we designed to work in specific engineered hosts under controlled conditions might function differently in new genetic contexts. They could combine with other genes in unpredictable ways. They could reach organisms we never intended to modify, including species living in pristine environments far from release sites. The historical precedent of ice- bacteria haunted these discussions. In 1987, Pseudomonas syringae strains genetically modified to lack a gene promoting ice crystal formation became the first genetically engineered organism deliberately released into the environment. The bacteria were sprayed on strawberry plants in California and potato plants in Oregon to protect crops from frost damage. The modification was minimal, a gene deletion rather than insertion of foreign DNA. The environmental context was limited, agricultural fields under observation. Yet the release sparked massive protests and regulatory delays lasting 4 years before final approval. Critics argued that insufficient testing had been conducted to predict ecological impacts. They feared the bacteria might spread beyond treated fields, potentially affecting weather patterns by reducing atmospheric ice nucleation. Though these fears proved unfounded, and the bacteria persisted only briefly on plant surfaces before native microbes outcompeted them, the controversy established precedents for assessing environmental releases of engineered organisms. Plastic-degrading bacteria posed far more complex scenarios than ice- strains. They would be released not on a few agricultural fields, but potentially into landfills, oceans, and cities worldwide. They carried not gene deletions, but multiple inserted genes encoding novel enzymatic pathways. They might persist for years rather than days if plastic substrates supported sustained populations. The scale and duration of exposure were orders of magnitude larger. Worst-case scenarios required systematic exploration despite their low probability. What if plastic-degrading bacteria evolved to degrade plastics we still need? Infrastructure pipes, vehicle components, medical equipment, electrical insulation. Modeling suggested this was unlikely because engineered bacteria had been selected specifically for degrading waste plastic with altered surface characteristics from weathering and not virgin polymers. But unlikely is not impossible. Mutations could alter substrate specificity. Gene transfer could place degradative enzymes into organisms with different habitat preferences that might colonize manufactured items. What if rapid plastic degradation released toxic additives faster than ecosystems could handle them? Plastics contain numerous chemicals, phthalates, bisphenol compounds, brominated flame retardants, heavy metals that were incorporated during manufacturing. While plastic remains intact, these chemicals leach slowly over decades. Biological degradation might release them in concentrated bursts, overwhelming organisms adapted to gradual low-level exposure. Laboratory toxicity tests showed that degradation products at concentrations measured in experiments did not harm test organisms, but environmental concentrations might vary unpredictably. What if engineered bacteria outcompeted native species, disrupting ecosystem functions beyond plastic degradation? Bacteria perform countless essential services: nutrient cycling, organic matter decomposition, nitrogen fixation, supporting plant growth. Native microbial communities are complex networks where each species occupies specific niches. Introducing highly competitive engineered bacteria might displace native species, potentially disrupting these services. Field monitoring at testbed sites detected community composition shifts where engineered bacteria were active, with some native groups declining. Whether these shifts caused functional impairment remained uncertain. Invasive species provide sobering examples of how organisms introduced into new environments can trigger cascading ecological impacts. The introduction of cane toads into Australia, intended to control crop pests, resulted in a biological invasion affecting predators, competitors, and countless species across multiple trophic levels. Zebra mussels introduced into North American Great Lakes transformed entire aquatic ecosystems, filtering water with such efficiency that food webs collapsed and native species disappeared. These invasions all began with good intentions and confident predictions that risks were minimal. The key difference, argued Dr. James Hartwell, a synthetic biologist who had helped engineer the plastic-degrading strains, is that biological invasions involve organisms optimized by millions of years of evolution for survival and reproduction in nature. Our engineered bacteria are laboratory adapted. They have been selected for plastic degradation under controlled conditions, not for fitness in complex environments. Field data supported this view. Engineered bacterial populations in the landfill testbed grew initially but plateaued as native competitors, predators, and environmental stresses limited their expansion. Ocean trials showed even stronger constraints, with engineered marine bacteria declining due to predation despite continued plastic availability. Kill switches and genetic safeguards offered technological solutions to containment concerns. These engineered control systems cause bacteria to die when they leave designated environments or lose essential molecular signals. The simplest designs make bacteria dependent on specific nutrients not available in nature, ensuring they starve if they escape treatment zones. More sophisticated circuits use conditional toxin expression, sensing environmental parameters like temperature or chemical signals, and producing lethal proteins when conditions fall outside permissive ranges. CRISPR-based kill switches represent the newest generation of containment technology. These systems employ Cas9 protein to target and cut the bacterial chromosome at multiple sites when activation conditions are met, causing catastrophic DNA damage that kills cells within hours. Research at MIT developed kill switches requiring two different small molecules to be present simultaneously for bacterial survival, a molecular passcode preventing accidental activation while ensuring deliberate control. Studies demonstrated these systems remain stable through hundreds of generations in laboratory culture, maintaining killing efficiency after 28 days of continuous growth. Yet kill switches face evolutionary pressure. Any mutation disabling the kill switch confers survival advantage if bacteria encounter restrictive conditions. Over time, disabled variants will be selected and proliferate. Researchers address this through redundancy, incorporating multiple independent kill switches so that disabling all of them simultaneously requires improbable simultaneous mutations. They also engineer bacteria to lack DNA repair systems, reducing mutation rates that might disable safeguards. Competition from wild-type bacteria can suppress escape mutants through numerical dominance. Testing kill switch effectiveness in environmental conditions rather than laboratory culture revealed limitations. The landfill testbed incorporated kill switches requiring specific chemical supplements added weekly. During a 3-week period, when logistical problems prevented supplement addition, bacterial populations declined approximately tenfold as expected. However, small surviving populations persisted, suggesting incomplete killing or emergence of resistant variants. Subsequent genetic analysis found that 5% of survivors carried mutations in kill switch regulatory genes. Gene flow from engineered bacteria to wild populations represents a hazard that kill switches cannot fully address. Even if engineered bacteria die, their DNA persists in the environment where it can be taken up by native bacteria through transformation. Alternatively, genes can transfer through conjugation before kill switches activate. Once plastic-degrading genes enter wild populations, they can spread through bacterial communities independent of the original engineered hosts. Monitoring wild bacterial populations for gene escape requires sensitive detection methods and extensive sampling. Quantitative PCR can detect specific gene sequences at concentrations as low as 100 copies per sample. Metagenomic sequencing can identify genes even without prior knowledge of their sequences, but applying these tools across relevant spatial scales, kilometers for landfills, thousands of kilometers for oceans, creates logistical and economic challenges. The ocean testbed implemented monthly sampling at 20 stations within 50 kilometers of release sites. This captured local gene flow, but could not monitor the entire Pacific Gyre. Regulatory frameworks for genetically modified organisms attempt to balance innovation against precaution. In the United States, the Environmental Protection Agency regulates environmental releases under the Toxic Substances Control Act and Federal Insecticide, Fungicide, and Rodenticide Act. Applicants must demonstrate that releases pose no unreasonable risk to human health or the environment. The European Union employs even stricter standards under Directive 2001/18 and Regulation 1829/2003, requiring comprehensive environmental risk assessments reviewed by the European Food Safety Authority before any release is authorized. The approval process for plastic-degrading bacteria consumed 3 years and cost over $2 million in regulatory compliance for the pilot projects. Applications included thousands of pages of data on organism characterization, containment measures, monitoring protocols, and ecological risk assessment. Public comment periods generated thousands of submissions, ranging from enthusiastic support to vehement opposition. Regulatory agencies convened expert panels, commissioned independent reviews, and required additional studies addressing uncertainties. Critics argued that no amount of testing could eliminate uncertainty when releasing self-replicating organisms into environments where they could evolve and interact with billions of other species. They invoked the precautionary principle, asserting that when facing potentially irreversible harm, lack of full scientific certainty should not prevent protective action. If engineered bacteria caused ecological damage, reversal would be impossible. Better to err on the side of caution and prohibit releases until safety could be proven definitively. Supporters countered that plastic pollution itself constituted an ongoing ecological catastrophe requiring urgent intervention. Waiting for perfect certainty meant accepting continued accumulation of plastic in every ecosystem while millions of animals suffered and died from entanglement and ingestion. They argued that well-designed releases with robust monitoring and safeguards represented reasonable risks compared to the guaranteed harms of inaction. The debate crystallized a fundamental tension in environmental management. How much risk is acceptable when attempting to solve one environmental problem with technologies that might create others? Who decides what constitutes acceptable risk? How are costs and benefits distributed across human communities and ecosystems, these questions had no purely technical answers. They required value judgments about precaution versus innovation, about human responsibility to repair damage caused by previous technologies, about the moral status of future generations and non-human species. As the risk assessment presentations concluded, the assembled group faced the reality that perfect safety was unattainable. Every scenario involved trade-offs. Proceeding with releases accepted risks of gene escape, community disruption, and unforeseen consequences. Prohibiting releases accepted the certainty of continued plastic accumulation and its documented harms. The question was not whether to accept risk, but which risks humanity would choose to bear, and whether the governance systems and monitoring capabilities existed to detect and respond if chosen risks materialized into actual harms. The answer would shape not just plastic pollution management, but the broader relationship between synthetic biology and planetary ecology in the 21st century. Chapter 17: The Containment Question. The distinction between physical containment and biological containment defined the strategic divide in plastic bioremediation. Physical containment meant barriers, walls, membranes, filters, and enclosed systems that prevented organisms from dispersing beyond designated areas. Biological containment meant genetic and metabolic dependencies that should prevent survival and reproduction if organisms escaped physical confines. Ideal deployments would employ both, creating layered defenses against unintended environmental spread. The three testbeds represented a spectrum of containment rigor. The urban pilot, processing plastic in enclosed bioreactors and constructed ponds, achieved relatively high physical containment. Organisms lived within engineered systems designed for control, with wastewater treatment before any discharge and regular monitoring of effluent. The landfill offered moderate containment, with compacted soil cover and clay liners limiting, but not preventing subsurface water flow. The ocean trials provided minimal physical containment, with mesh enclosures preventing immediate dispersal, but offering no barrier to long-term gene flow through horizontal transfer to marine bacteria that could pass through the mesh. Evaluating containment effectiveness required distinguishing between organism escape and gene escape. An organism escapes when living bacterial cells move beyond designated treatment areas and establish viable populations in unintended locations. Gene escape occurs when DNA sequences from engineered bacteria, whether in living cells or as naked DNA released from dead cells, transfer into native bacteria through horizontal gene transfer mechanisms. Both represented potential pathways for spreading engineered traits into wild populations, but they operated on different timescales and through different processes. Organism escape was relatively straightforward to monitor through direct detection. Quantitative PCR targeting unique genetic barcodes in engineered strains could identify them in environmental samples with high sensitivity and specificity. The landfill testbed maintained monitoring wells at 5, 10, 20, and 50 meters from treatment zones, collecting groundwater samples monthly and analyzing for engineered bacteria. Through 18 months of monitoring, detections remained negative beyond 20 meters from release sites, suggesting that organism dispersal through groundwater flow was limited. However, absence of detection does not prove absence. PCR sensitivity depends on sampling volume and bacterial concentration. If engineered bacteria existed at densities below 100 cells per liter, the detection threshold for standard assays, they would not be detected even though viable populations might be present. Increasing sensitivity requires concentrating larger sample volumes, but this is impractical for extensive spatial coverage. The ocean testbed faced particularly acute detection challenges given the trillion-liter volumes of water surrounding release sites. Gene escape monitoring required different approaches. The plastic-degrading genes in engineered bacteria had been identified and sequenced, allowing design of PCR primers targeting these specific sequences. Metagenomic sequencing of microbial communities from soil and water samples around testbed sites provided comprehensive inventories of genes present in native populations. Comparing metagenomes from areas near releases to control areas distant from releases could reveal whether engineered genes appeared in native communities at elevated frequencies. Year 2 sampling at the landfill testbed detected PTase gene sequences in native bacterial populations 20 to 40 meters from treatment zones, beyond where engineered bacteria themselves had been detected. The sequences were 98% identical to engineered PTase, far more similar than to natural cutenases found in soil bacteria. This constituted strong evidence of horizontal gene transfer from engineered strains to native soil bacteria. The discovery triggered intensive investigation to determine transfer mechanisms, identify recipient species, and assess whether transferred genes were functional in their new hosts. Whole genome sequencing of bacterial isolates carrying PTase-like genes revealed that the transferred sequences resided on plasmids, circular DNA elements that can replicate independently of bacterial chromosomes and transfer between cells through conjugation. The engineered strains used in the landfill had been designed with PTase genes integrated into chromosomes rather than carried on plasmids, precisely to prevent easy transfer. Yet recombination events had apparently mobilized the chromosomal genes onto plasmids present in the bacteria, creating unexpected transfer vectors. The recipient bacteria belonged to native Pseudomonas and Bacillus species naturally abundant in landfill environments. Laboratory tests demonstrated that transferred PTase retained catalytic activity in these new hosts, degrading PET at approximately 40% the rate achieved by the original engineered strain. This confirmed that gene escape had functional consequences, spreading plastic degrading capability to wild populations. Debate erupted over whether this constituted failure of containment or demonstration that biological plastic degradation could be achieved through deliberate seeding of native communities. Critics argued that any unintended gene spread violated containment principles and validated concerns about loss of control. They noted that once genes entered wild populations, they could spread through bacterial communities independent of human management, potentially reaching environments far from original release sites. Supporters suggested that gene transfer to native bacteria might actually enhance remediation effectiveness. Native species are adapted to local environmental conditions, giving them advantages over laboratory-engineered strains in tolerating temperature fluctuations, moisture stress, predation, and competition. If plastic-degrading genes transferred to these well-adapted natives, the resulting organisms might achieve more robust long-term plastic degradation than engineered strains alone. This perspective reframed gene escape from unintended consequence to intentional strategy. Some researchers proposed designing engineered bacteria explicitly for gene donation rather than direct environmental function. These donor strains would carry plastic-degrading genes on easily transferred plasmids and would be deployed specifically to seed native communities. Kill switches would eliminate donor bacteria after transfer occurred, leaving only natives carrying transferred genes. This approach might combine the benefits of genetic engineering with the ecological fitness of native organisms. Regulatory agencies remained skeptical. Approvals for testbed releases had been conditioned on preventing gene escape, not enabling it. Any strategy deliberately transferring engineered genes to wild populations would require completely new regulatory review addressing the expanded scope and uncertainty. Public acceptance would likely be even more challenging when asking communities to approve not just releasing engineered bacteria, but intentionally spreading engineered traits into natural microbial ecosystems. Quantifying containment failure rates proved essential for risk assessment, but technically challenging. Organism escape rates could be estimated from monitoring data showing presence or absence in samples at various distances from release sites. Statistical models incorporating bacterial dispersal mechanisms, growth rates, and environmental survival predicted spatial patterns of organism presence. Comparing predictions to observations provided estimates of escape frequency. For the landfill, models predicted that fewer than 1 in 1 million engineered bacteria should disperse beyond 50 meters through groundwater transport over 1-year timescales. This matched observation of non-detection in distant monitoring wells. However, other transport mechanisms, including movement of invertebrates and burrowing animals through soil, could not be easily quantified and might enable occasional long-distance dispersal events not captured by models. Gene escape rates depended on horizontal transfer frequencies in environmental contexts. Laboratory measurements showed conjugation occurring at rates of 1 transfer per 1,000 donor-recipient encounters for plasmid-borne genes. Transformation of naked DNA occurred at lower frequencies, approximately 1 uptake event per 10 million cells exposed. Translating these laboratory rates to environmental conditions required accounting for spatial proximity between donors and recipients, persistence of DNA in soil or water, and selective pressures favoring retention of transferred genes. Monitoring timescales created uncertainty. Most testbed monitoring extended 1 to 3 years, brief compared to evolutionary and ecological timescales. Evolutionary adaptation could alter organism traits years after initial release as selection pressures accumulated. Slow-growing recipient bacteria might require years to amplify transferred genes to detectable frequencies. Rare dispersal events might not be observed in short monitoring periods, but could be significant over decades. These temporal uncertainties complicated regulatory decision-making. Agencies wanted assurance that containment would remain effective not just during monitoring periods, but indefinitely. Yet providing such assurance required extrapolating from limited data to longer timescales, introducing substantial uncertainty. Some proposed adaptive management approaches where releases would be approved provisionally, with requirements for ongoing monitoring and authority to modify or terminate operations if problems emerged. International coordination on containment standards face challenges. Different nations employed different regulatory frameworks and risk tolerance. Strict containment requirements in Europe contrasted with more permissive approaches in some developing nations. Bacteria released in one country could potentially spread across borders through air, water, or animal transport, creating transboundary environmental consequences. Yet no international treaty governed releases of engineered microorganisms comparable to agreements regulating releases of chemical pollutants or greenhouse gases. The containment question ultimately centered on acceptable levels of residual risk. Perfect containment was unattainable. Physical barriers leak. Genetic safeguards fail. Monitoring has detection limits. The relevant question was whether risks could be reduced to levels comparable to or lower than risks society already accepts from other technologies and activities. Automobile transportation kills over 1 million people globally per year, yet society accepts this toll as reasonable trade-off for mobility benefits. Would society accept some probability of gene escape from plastic-degrading bacteria as reasonable trade-off for reducing plastic pollution? The answer depended less on technical risk calculations than on social values, public trust in institutions managing the technology, and belief that environmental benefits justified ecological uncertainties. Chapter 18: BioReactors and Toxicity. Promise of biological plastic degradation centered on converting synthetic polymers into simple molecules that could re-enter biogeochemical cycles. Ideally, PET would decompose completely into terephthalic acid and ethylene glycol, which bacteria would further metabolize into carbon dioxide and water. Polyethylene and polypropylene would oxidize into shorter-chain hydrocarbons, then alcohols, aldehydes, and carboxylic acids, eventually reaching acetate and carbon dioxide. The end result would be complete mineralization, plastic literally disappearing as its carbon atoms dispersed into the atmosphere or integrated into biomass. Reality proved more complex. Degradation pathways involved multiple enzymatic steps, each producing intermediate compounds that accumulated if subsequent reactions proceeded slowly. Some intermediates were innocuous, simple organic acids and alcohols that many organisms readily metabolize. Others were problematic, aromatic compounds or reactive species that could be toxic to microorganisms, plants, or animals. Understanding what was produced during plastic degradation and assessing the toxicity of degradation products became critical for evaluating whether biological remediation solved the plastic problem or merely transformed it into different chemical hazards. Comprehensive chemical analysis of water and soil from testbed treatment zones employed gas chromatography mass spectrometry and liquid chromatography mass spectrometry to identify and quantify organic compounds. These powerful analytical techniques can detect thousands of compounds at concentrations down to parts per billion. Comparing chemical profiles from treatment zones to control zones revealed which compounds were specifically associated with plastic degradation. The bioreactor processing PET generated predictable major products. Terephthalic acid accumulated to concentrations ranging from 50 to 200 milligrams per liter in reactor water, and ethylene glycol reached 20 to 80 milligrams per liter. These concentrations aligned with expectations from 12% degradation efficiency and represented the intended products of enzymatic PET hydrolysis. Both compounds have well-characterized toxicity profiles. Terephthalic acid shows low acute toxicity, with lethal dose 50 values in rodents exceeding 5,000 milligrams per kilogram of body weight. Ethylene glycol is more toxic, particularly to kidneys, but concentrations in reactor effluent were hundreds of times below levels causing harm in aquatic organisms. However, trace compounds beyond the intended products raised concerns. Mass spectrometry detected approximately 300 minor organic compounds in reactor water, most at concentrations below 1 milligram per liter, but collectively representing complex chemical mixtures. Many were breakdown products of additives originally present in waste plastic, colorants, plasticizers, stabilizers, and flame retardants. Others appeared to be bacterial metabolites, compounds produced by organisms growing on plastic degradation products, but not directly derived from the polymer itself. Phthalate compounds were particularly prevalent. These chemicals are added to plastics as plasticizers, making rigid polymers more flexible. When plastic degrades, phthalates are released. Analysis found d-2-ethylhexyl phthalate at concentrations up to 12 micrograms per liter in bioreactor water and dibutyl phthalate at 4 micrograms per liter. Both compounds are classified as endocrine disruptors capable of interfering with hormonal systems in vertebrates at low concentrations. Long-term exposure causes reproductive and developmental toxicity in fish, amphibians, and mammals. Bisphenol A, another endocrine disruptor, appeared at 1 to 3 micrograms per liter. Although BPA is primarily associated with polycarbonate plastics rather than PET, small amounts are present in recycled PET from cross-contamination during waste collection and processing. Brominated flame retardants, added to plastics used in electronics and building materials, were detected in trace amounts when mixed plastic waste entered the bioreactor. The landfill testbed presented even more complex chemistry. Soil samples from treatment zones contained over 800 identified organic compounds, reflecting decades of plastic agging combined with ongoing degradation. Polycyclic aromatic hydrocarbons, formed through incomplete combustion and also produced during plastic photodegradation, reached concentrations of 50 to 200 micrograms per kilogram of soil. Some polycyclic aromatics are carcinogenic, though concentrations measured in the landfill fell within ranges typical of urban soils, not considered acutely hazardous. Wastewater treatment pond water showed elevated concentrations of microplastic-associated chemicals. Triclosan, an antimicrobial compound from personal care products, often bound to microplastics, measured 10 to 30 micrograms per liter in pond influent, but only 2 to 5 micrograms per liter in effluent, suggesting biological degradation or adsorption to sediments. Perfluorinated compounds, extremely persistent chemicals that associate with plastic particles, showed no reduction through pond treatment, confirming that these substances resist biological degradation. Toxicity bioassays tested whether complex chemical mixtures from degradation processes harmed organisms. Standard assays expose sensitive species, Daphnia water fleas, fathead minnows, green algae, to water or soil samples and measure survival, growth, or reproduction. These tests integrate effects of all chemicals present, capturing interactions and synergies that might not be apparent from analyzing individual compounds. Results varied by testbed and time. Fresh bioreactor water showed no acute toxicity in any test species, with survival and growth identical to controls. However, chronic exposure for 21 days reduced Daphnia reproduction by 12% compared to controls, a statistically significant effect suggesting subtle endocrine disruption. Subsequent analysis identified phthalates as likely causative agents. Implementing activated carbon filtration to remove phthalates from reactor effluent eliminated the reproductive toxicity. Landfill soil extracts initially showed moderate toxicity in plant germination tests, with radish seedling emergence reduced by 25%. Extended monitoring over 2 years revealed that toxicity declined gradually as initial bursts of released additives were diluted or further degraded. By year 3, soil toxicity in treatment zones was indistinguishable from control zones. This temporal pattern suggested that acute release of additives occurred early during plastic degradation, but that long-term toxicity was not sustained. Wastewater pond effluent occasionally exhibited toxicity spikes corresponding to storm events that flushed elevated microplastic loads into the treatment plant. These spikes caused transient reductions in algal growth and Daphnia survival, lasting 2 to 5 days before returning to baseline. The intermittent toxicity reflected the variable chemical composition of microplastics entering the pond, with some loads carrying high concentrations of additives while others were relatively clean. Comparing toxicity of biological degradation to alternative plastic management options provided essential context. Plastic buried in landfills slowly leaches chemicals into groundwater for decades, maintaining chronic low-level contamination. Incineration of plastic converts polymer carbon into carbon dioxide, but also generates toxic combustion byproducts, including dioxins, furans, and heavy metal aerosols if temperatures and pollution control are inadequate. Mechanical recycling avoids these acute releases, but typically cannot process heavily contaminated plastic, which ends up landfilled anyway. Life cycle assessment, incorporating toxicity impacts suggested biological degradation, occupied a middle position. It avoided long-term leaching from landfills and toxic emissions from incineration, but it concentrated additive release into shorter periods and required management of contaminated water or soil. Whether this represented net environmental benefit depended on local conditions: groundwater vulnerability near landfills, air quality near incinerators, and treatment capacity for managing bioreactor effluent. Engineering solutions to minimize problematic degradation products focused on 3 strategies. First, saw separation of plastic waste to exclude heavily contaminated materials from biological treatment, reduced additive loads entering systems. The urban pilot implemented pre-screening that rejected plastic containing visible contamination or known to carry high brominated flame retardant concentrations. This reduced trace contaminant concentrations in bioreactor water by approximately 60%. Second, coupling biological degradation with chemical treatment removed problematic compounds. Activated carbon adsorption effectively captured phthalates, bisphenols, and aromatic compounds. Advanced oxidation using ozone or UV peroxide destroyed persistent organic pollutants that resisted biological breakdown. These additional treatment steps increased costs, but enabled meeting water quality standards for effluent discharge. Third, selecting bacterial strains capable of degrading not just plastic polymers, but also associated additives offered potential one-step solutions. Some Pseudomonas and Rhodococcus species possess broad enzymatic repertoires that attack numerous synthetic compounds. Genetic engineering could could enhance these native capabilities, creating organisms that simultaneously degrade polymers and detoxify additives. Early laboratory tests demonstrated proof of concept, but field deployment awaited further development and regulatory approval. The toxicity question extended beyond chemistry to physical effects of microplastics. Even chemically inert plastic particles can harm organisms through physical mechanisms. Particles ingested by fish, seabirds, and marine mammals accumulate in digestive tracts, creating false sensation of fullness that reduces feeding and causes starvation. Sharp-edged fragments cause physical damage to tissues. Microplastics smaller than 50 micrometers can cross cellular membranes, potentially reaching tissues throughout organisms' bodies with unknown consequences. Biological degradation's impact on microplastic toxicity was paradoxical. Initial stages of degradation fragmented large plastic items into increased numbers of microplastic particles, potentially worsening physical hazards. Laboratory experiments confirmed this, showing 2 to 5 times more particles in water samples from treatment systems compared to controls during first months of operation. However, longer-term monitoring showed particle counts declining as degradation progressed to complete mineralization. The transient increase in microplastics posed challenges for managing systems where organisms might be exposed during this intermediate phase. The byproducts and toxicity assessment ultimately concluded that biological plastic degradation was neither toxicologically neutral nor catastrophically hazardous. It mobilized chemicals from plastics that would otherwise release slowly over decades, concentrating exposure temporarily but enabling treatment and management. It generated intermediate degradation products, some potentially problematic, that required monitoring and potentially additional treatment before disposal. But it offered pathways to complete plastic mineralization that mechanical recycling and incineration could not achieve, and it avoided persistent accumulation that made landfilling environmentally unsustainable. Managing the toxicology of biological remediation required careful system design, comprehensive monitoring, and adaptive responses to detected problems, capacities that the testbeds demonstrated were achievable with appropriate resources and expertise. The 3-year assessment marked a critical juncture in the testbed experiments. Initial deployment and first-year monitoring had documented bacterial establishment, measured short-term degradation rates, and identified immediate operational challenges. But plastic accumulation in the environment represented a decades-long problem, with debris from the 1960s still intact in oceans and landfills. Understanding whether biological degradation could address this legacy pollution required observing system performance over timescales long enough to capture ecological succession, community adaptation, and the gradual effects of sustained microbial activity on recalcitrant polymers. The landfill testbed revealed striking temporal dynamics. Initial bacterial colonization of plastic substrates during months 1 through 6 showed characteristic pioneering species, Pseudomonas and Bacillus strains that rapidly attached to surfaces and began biofilm formation. These early colonizers modified surface chemistry through initial enzymatic attack, oxidizing polymer chains and creating functional groups that increased surface wettability. By months 6 through 12, microbial community composition shifted dramatically. Metagenomic sequencing detected increasing abundance of specialist degraders, bacteria carrying multiple genes for attacking different polymer types, and declining representation of generalist pioneers. This community succession reflected ecological principles governing ecosystem development. Pioneer organisms colonize new habitats rapidly, but often lack specialized capabilities for utilizing resources fully. Specialist species arrive later, possessing enzymatic machinery or metabolic pathways that enable more complete resource exploitation. In landfill environments, this succession proceeds through distinct phases mirroring the conceptual model describing organic waste decomposition. Phase 1 involves aerobic respiration where oxygen is available. Phase 2 sees transition to anaerobic fermentation, producing organic acids. Phase 3 features methanogenesis, as archaeal populations metabolize fermentation products into methane and carbon dioxide. Plastic-degrading bacteria inserted into this system had to navigate the existing successional framework. The engineered strains were introduced into Phase 2 and Phase 3 zones, where conditions ranged from mildly acidic, pH 6.2, to neutral, pH 7.1, and where oxygen concentrations varied from anoxic in deep zones to partially oxygenated near the surface. Year 1 monitoring documented successful establishment primarily in the upper landfill layers, where oxygen availability supported aerobic metabolism. Populations in deeper anaerobic zones declined within 6 months, though not to zero, suggesting some cells survived in dormant states or microaerobic niches. By year 2, community dynamics showed adaptive reorganization. Native bacteria that had acquired plastic-degrading genes through horizontal transfer constituted 15 to 25% of plastic-associated populations in treatment zones. These hybrid organisms combined native adaptation to landfill chemistry with acquired plastic-degrading capability, potentially representing more ecologically fitted degraders than the original engineered strains. Measurements of plastic mass loss supported this interpretation. Year 1 degradation rates averaged 0.4% mass loss for PET and 0.2% for polyethylene. Year 2 rates increased to 0.9% and 0.5% respectively, despite engineered bacterial populations remaining relatively constant. The enhanced performance correlated with increased abundance of native bacteria carrying transferred degradation genes. Year 3 brought the unexpected appearance of fungal plastic degraders. Metagenomic analysis detected DNA sequences from Aspergillus and Fusarium species, soil fungi not included in initial bacterial inoculations, carrying genes similar to bacterial cutinases and laccases. Microscopic examination of excavated plastic samples revealed fungal hyphae penetrating polymer surfaces, creating networks of channels visible under scanning electron microscopy. The fungi apparently colonized treatment zones independently, drawn by organic compounds released during bacterial plastic degradation, then evolved or acquired capability to attack plastic themselves through mechanisms still under investigation. This emergent complexity represented both promise and uncertainty. The self-organizing assembly of degrader communities, including bacteria, both engineered and native, and fungi suggested that biological plastic remediation might achieve self-sustaining momentum once initiated. Yet it also meant that outcomes deviated from planned interventions, introducing organisms and genetic elements not deliberately released and potentially not subject to regulatory oversight. The landfill ecosystem was responding to the initial perturbation of engineered bacterial introduction in ways that could not have been fully predicted. Long-term chemical changes in landfill leachate documented the environmental consequences of sustained degradation. Terephthalic acid concentrations in groundwater monitoring wells within treatment zones increased from below detection limits before treatment to an average of 4 micrograms per liter by year 1, 8 micrograms per liter by year 2, and 14 micrograms per liter by year 3. These concentrations remained far below toxicity thresholds, but confirmed ongoing release of degradation products into the subsurface environment. Wells 50 meters downgradient of treatment zones showed no terephthalic acid detection through year 2, but measured 1 to 2 micrograms per liter in year 3, demonstrating that degradation products were slowly migrating through groundwater. Integrated monitoring, combining microbiological, chemical, and isotopic analysis, provided comprehensive understanding of biogeochemical transformations. Carbon isotope ratios in dissolved organic carbon from treatment zone leachate showed characteristic signatures of bacterial metabolism, with carbon-13 depletion indicating preferential utilization of lighter isotopes during respiration. This isotopic fingerprint allowed distinguishing plastic-derived carbon from background organic matter in landfills, confirming that approximately 30 to 40% of dissolved organic carbon in treatment zones originated from plastic degradation by year 3. The ocean testbed presented radically different temporal dynamics constrained by environmental instability. Marine surface waters experienced daily temperature fluctuations of 2 to 4 degrees Celsius, seasonal cycles spanning 12 to 18 degrees range, and episodic storm events that drastically alter mixing, light availability, and nutrient distributions. These fluctuations created boom and bust cycles in bacterial populations. Warm periods with high solar radiation supported rapid bacterial growth on plastic surfaces, with cell densities reaching 10 million per square centimeter of biofilm. Cool, cloudy periods, or winter months when light intensity dropped and water temperatures fell to 13 degrees, saw populations decline to 1 million cells per square centimeter as growth slowed and cell mortality exceeded replication. Temperature effects on plastic degradation exceeded impacts on bacterial populations. Enzymatic reaction rates typically double or triple for every 10-degree temperature increase within the enzyme's functional range, a relationship described by the Q10 temperature coefficient. PTA's activity measurements at different temperatures documented Q10 values of 2.4, meaning activity at 20 degrees was approximately 2.4 times higher than at 10 degrees. Over seasonal cycles, this translated to degradation rates varying fourfold between summer and winter. Cumulative plastic mass loss over 3 years reflected this seasonality. Annual degradation averaged 1.2% for floating PET in the garbage patch, but year-to-year variation ranged from 0.9% in a relatively cool year to 1.6% in a warm year. Light attenuation with depth created vertical stratification in degradation rates. Surface waters receive full sunlight enabling photodegradation that complements biological processes. UV radiation breaks polymer chains through photooxidation, creating carbonyl groups and increasing surface area accessible to bacterial enzymes. Plastic at 10 meters depth receives approximately 50% of surface UV intensity, depending on water clarity. At 30 meters, UV penetration drops below 10%, essentially eliminating photodegradation. Measurements of plastic degradation at different depths confirmed this gradient. Surface degradation averaged 1.2% annually, 10 meters showed 0.8%, and 30 meters only 0.3%. This depth dependence raised concerning questions about plastic that sinks beyond the euphotic zone. In the deep ocean, where temperatures hover near 4 degrees Celsius, light is absent, and microbial metabolic rates are minimal, plastic degradation proceeds extraordinarily slowly. Modeling based on deep-sea environmental conditions predicted that PET bottles reaching abyssal depths at 3,500 meters might require 300 years for complete degradation, compared to perhaps 50 years at the surface, where warmth, light, and active microbial communities accelerate breakdown. Vast quantities of plastic have already sunk to the seafloor through biofouling-induced negative buoyancy, accumulating in an environmental compartment largely inaccessible to both biological remediation and monitoring. The marine biological community showed subtle but detectable responses to 3 years of experimental plastic degradation. Phytoplankton community composition in waters surrounding ocean testbed enclosures shifted compared to control areas. Diatoms, silica-shelled photosynthetic algae, increased in relative abundance while cyanobacteria declined. The mechanism appeared to involve microplastic fragments serving as settlement substrates for diatoms, giving them competitive advantages over free-floating cyanobacteria. This community shift had potential implications for carbon cycling, as diatoms are more effective than cyanobacteria at transporting carbon to deep water through the biological pump. However, microplastic ingestion by zooplankton created countervailing effects. Small crustaceans, including copepods, consumed microplastic particles alongside their normal phytoplankton prey, a case of mistaken feeding that provided no nutrition while filling digestive tracts. This reduced effective grazing pressure on phytoplankton, potentially disrupting the biological carbon pump in complex ways, depending on whether the system was nutrient-limited or nutrient-replete. In nutrient-rich areas, reduced grazing could enhance carbon export by allowing more phytoplankton to sink uneaten. In nutrient-poor regions like the garbage patch, reduced grazing diminished nutrient recycling through zooplankton excretion, potentially decreasing productivity overall. Recent research published in early 2026 highlighted these complex interactions between microplastics and ocean carbon sequestration. Microplastics disrupt not only individual organisms, but also ecosystem-level processes, including the biological carbon pump, a natural mechanism absorbing approximately 2 billion tons of atmospheric carbon dioxide annually and transporting it to deep ocean storage. The plastosphere, microbial communities colonizing plastic surfaces, participates in carbon and nitrogen cycling while also producing greenhouse gases as plastics degrade. Metagenomic studies of plastosphere communities from the garbage patch detected genes for methanogenesis and denitrification, metabolic pathways that release methane and nitrous oxide, both potent greenhouse gases. The temporal evolution over 3 years also revealed the critical importance of monitoring ecological non-target effects. Soil invertebrates in the landfill testbed, particularly earthworms, springtails, and mites, showed population changes in treatment zones compared to controls. Earthworm abundance declined by approximately 20% in zones with highest bacterial activity. Laboratory toxicity tests exposing earthworms to soil from treatment zones documented sublethal effects, including reduced growth rates and reproductive output. Chemical analysis suggested these effects resulted from elevated concentrations of plastic additives released during degradation rather than from bacterial activity itself. These findings paralleled research on pesticide impacts on soil invertebrates, which documented that chemicals disrupting target organisms almost invariably affect non-target species performing essential ecosystem services. Earthworms create up to 8,900 kilometers of burrows per hectare, dramatically influencing soil porosity, water infiltration, and nutrient cycling. Springtails and mites drive decomposition of organic matter and regulate bacterial and fungal populations. Reductions in these populations could compromise ecosystem functions even if plastic degradation itself proceeded as intended. The 3-year temporal window provided by the test beds was simultaneously illuminating and insufficient. It documented that biological plastic degradation was not a simple one-time intervention, but a dynamic process involving ecological succession, community adaptation, seasonal fluctuation, and emergent complexity. It revealed that degradation rates could increase over time as specialist communities assembled, but also that environmental variability created unpredictable performance fluctuations. It showed that non-target effects could accumulate gradually, only becoming apparent after multiple years. Yet 3 years represented a tiny fraction of the timescales over which plastic persists in the environment, typically decades to centuries. Projecting from 3-year observations to century-scale outcomes required modeling and extrapolation fraught with uncertainty. The testbeds had answered many questions while generating equally many new ones, confirming the fundamental challenge that environmental complexity resists simple technological fixes. Chapter 20: The Decision Point. The conference room at the research center felt charged with tension as 43 people representing stakeholders from across the plastic bioremediation landscape gathered for the 3-year decision review. Scientists who had designed and monitored the testbeds sat alongside regulatory officials responsible for approving expanded deployments. Industry representatives from waste management companies and plastics manufacturers occupied one section of tables. Environmental advocacy groups, some supportive and some skeptical, filled another section. City officials from the urban pilot participated remotely via video link. The purpose was explicit and consequential. Based on 3 years of data, the assembled group would recommend whether to terminate the experimental programs, modify them substantially before continuing, or expand toward larger-scale implementation. The decision framework employed sequential probability ratio testing principles adapted from medical trials, where ongoing experiments are periodically evaluated against pre-specified success criteria to determine whether to continue. For the plastic bioremediation testbeds, success criteria included degradation rate targets, containment performance thresholds, toxicity limits, and public acceptance metrics. Dr. Chen opened with a comprehensive data presentation. 3 years of monitoring had generated over 20,000 individual measurements across microbiology, chemistry, toxicology, and ecological impact categories. Sophisticated data integration and statistical analysis distilled these measurements into key performance indicators. Plastic degradation rates had exceeded initial targets in the urban bioreactor facility. 15% mass loss versus 10% targeted, representing meaningful success. Landfill degradation met, but did not exceed targets, approximately 0.9% annually for PET versus 1% targeted. Ocean degradation fell substantially short, 1.2% annually versus 5% targeted, though this reflected unrealistic initial expectations more than technical failure. Containment performance showed mixed results. Physical containment in the urban systems remained excellent, with no detections of engineered organisms beyond facility boundaries. The landfill showed acceptable organism containment, with detections limited to within 20 meters of release points, but concerning gene transfer to native bacteria had occurred at 40 meters. Ocean trials effectively had no organism containment given the open water context, though the experimental mesh enclosures had maintained physical barriers as designed. Toxicity assessments indicated manageable but non-zero risks. Degradation products occasionally exceeded water quality guidelines for specific compounds, requiring additional treatment before discharge. Soil invertebrate populations showed declines in treatment zones, raising ecological concerns. However, no acute toxicity or ecosystem collapse had occurred, and detected effects were comparable to or less severe than impacts from conventional waste management alternatives. Public perception surveys revealed complex and sometimes contradictory attitudes. In the urban pilot city, support for the program had increased from 61% at the start to 68% after 3 years, suggesting sustained engagement and transparency had built trust. However, support remained conditional and soft. When asked specifically about expanding to industrial scale, support dropped to 53%, and only 41% supported deployment in other cities. National surveys showed even lower acceptance, with 47% viewing genetically modified bacteria for waste treatment negatively and only 38% positively. These perception data highlighted the gap between scientific assessment and public acceptance. Technical experts evaluating risk-benefit trade-offs might conclude that biological plastic degradation offered net advantages over alternatives, but public trust required more than technical analysis. Perception of risk is shaped by factors beyond probability and magnitude, including dread, controllability, voluntariness, and trust in managing institutions. Engineered organisms scored poorly on several dimensions, evoking concerns about uncontrollable spread and irreversible consequences, even when quantitative risk assessments suggested low probabilities. The question of trust in regulatory processes emerged as particularly critical. Research from South Korea on acceptance of gene-edited crops found that policy trust and perceived information environment quality were decisive factors. When citizens trusted government agencies to regulate biotechnology effectively and believed they had access to credible information, acceptance increased substantially. Conversely, distrust in institutions and perceptions of biased or incomplete information drove rejection regardless of actual technology characteristics. Environmental advocacy groups presented divergent perspectives. Ocean conservation organizations generally supported continued research, arguing that plastic pollution constituted an ongoing catastrophe that justified exploring innovative solutions. However, they insisted on stringent monitoring, rapid response protocols if problems emerged, and prioritization of plastic source reduction over remediation. Broader environmental coalitions were split, with some viewing synthetic biology as necessary tool for addressing human-caused environmental damage, while others rejected any genetic modification of organisms as unacceptable interference with nature. Industry stakeholders calculated different equations. Waste management companies saw potential competitive advantages in offering advanced biological treatment services, particularly if regulatory mandates or corporate sustainability commitments created demand. However, they also recognized significant liability risks if released organisms caused environmental damage, potentially exposing companies to lawsuits and regulatory penalties. Plastics manufacturers had mixed interests. Companies producing virgin plastic from petroleum had little incentive to support technologies that might reduce demand. Conversely, companies marketing recycled content or biodegradable alternatives saw biological degradation as complementary to their business models. Government regulators articulated the precautionary principle. Environmental protection agencies held responsibility for safeguarding ecosystems and human health from potentially harmful substances and organisms. When facing uncertainty about long-term consequences, their mandate favored caution. The 3-year data provided substantial information, but could not eliminate all uncertainty, particularly about rare events, evolutionary changes, or ecosystem-level effects that might manifest only over decades. Several regulators argued that absent definitive proof of long-term safety, larger-scale deployments should not proceed. Scientists on the research team pushed back against demands for absolute certainty. "We cannot prove that no harm will ever occur," Dr. Hartwell stated bluntly. "That standard is impossible to meet for any environmental intervention. We can only demonstrate that risks appear acceptable based on available evidence and that benefits likely outweigh costs." Waiting for perfect certainty means accepting continued plastic accumulation, which itself carries enormous and documented harms. The discussion crystallized around 3 decision options. Option 1 was termination, ending experimental releases and prohibiting further development of biological plastic remediation using genetically modified organisms. Proponents argued this applied appropriate precaution given uncertainties and non-target effects. Opponents countered it abandoned promising technology and consigned the world to continued plastic pollution. Option 2 involved substantial modification before continuing. This would pause current field deployments, implement enhanced containment measures, including redundant kill switches and physical barriers, expand monitoring programs to detect low-probability events, and conduct additional toxicology and ecological impact studies before any expansion. This middle path acknowledged both the technology's promise and legitimate concerns about current implementation. However, it also meant delaying practical application for years while research continued, and it required sustained funding during this pause. Option 3 recommended cautious expansion. The urban bioreactor facility would scale from 10 to 50 tons per day processing capacity. Additional cities would launch pilot programs following the established model. Landfill and ocean deployments would remain experimental, restricted to research scale with comprehensive monitoring, but would continue rather than terminate. This option emphasized adaptive management, proceeding incrementally while continuously evaluating performance and adjusting based on emerging evidence. Voting occurred after 6 hours of presentations and debate. Using a decision matrix that weighted different stakeholder groups equally, scientists, regulators, industry, environmental groups, and public representatives, the results showed 11 votes for termination, 18 for substantial modification, and 14 for cautious expansion. The plurality supported modification, reflecting broad consensus that the technology showed promise, but current implementation required improvement before scaling. However, subgroup analysis revealed striking divisions. Scientists voted overwhelmingly for expansion, 11 of 14 favoring option 3. Regulators split evenly between termination and modification, with none supporting expansion. Industry representatives favored expansion, 8 of 10 votes, seeing commercial opportunity. Environmental groups were most divided, with votes distributed across all 3 options, reflecting their philosophical diversity. Public representatives, selected through random sampling to represent broader community perspectives, strongly favored modification, 7 of 9 votes, suggesting cautious interest but insistence on additional safeguards. These divergent perspectives reflected fundamentally different values and priorities. Scientists prioritized knowledge generation and problem-solving, viewing the testbeds as successful experiments that warranted continuation. Regulators prioritized environmental protection and risk minimization, viewing uncertainties as grounds for caution. Industry prioritized economic opportunity balanced against liability risk. Environmental groups prioritized ecosystem integrity, with some viewing technology as solution and others as additional threat. Public representatives prioritized safety, transparency, and agency, wanting assurance that they retained control over technologies affecting their communities. The recommendation forwarded to funding agencies and regulatory bodies reflected the plurality view: substantial modification before continued deployment. Specific modifications included development and implementation of CRISPR-based redundant kill switches with independent activation mechanisms. Expansion of monitoring networks to include metagenomic surveillance of gene transfer across wider spatial scales. Comprehensive ecological impact studies examining soil and marine invertebrates, plants, and microbial community functions over 5-year timescales. Enhanced public engagement, including citizen science participation in monitoring and citizen advisory boards with binding input into operational decisions. Implementation of these modifications would require approximately 18 months and $4 million in additional investment. During this period, existing deployments would continue at current scales to maintain continuity of monitoring data, but no expansion would occur. After modifications were implemented, a second decision review would evaluate whether to proceed with expansion, continue at experimental scale, or terminate. The decision represented neither unconditional approval nor outright rejection. It embodied cautious optimism tempered by recognition of legitimate concerns and acknowledgement of irreducible uncertainties. Whether biological plastic degradation would ultimately contribute meaningfully to addressing global plastic pollution remained unresolved. But the decision framework ensured continued evaluation, adaptation to emerging evidence, and mechanisms for terminating the program if risks proved unacceptable or benefits insufficient. The pathway forward was not predetermined, but would emerge through iterative cycles of testing, monitoring, assessment, and deliberation, a fundamentally adaptive approach to managing technological intervention in complex environmental systems. Chapter 21, LESSONS from the TSTBDS. Standing at the edge of the landfill excavation pit 3 years after initial bacterial deployment, Dr. Chen reflected on how profoundly the testbed experiments had transformed understanding of both plastic degradation and environmental biotechnology more broadly. The excavated plastic samples in sealed containers represented a material archive documenting the complex interplay between engineered organisms, native microbial communities, chemical transformations, and ecological responses. Each piece of degraded plastic told a story written in surface pitting, biofilm colonization, and altered polymer chemistry. The primary technical lesson was unequivocal: biological plastic degradation works, but not as quickly or completely as optimistic early projections suggested. The vision of bacteria rapidly consuming plastic waste and converting it entirely to carbon dioxide and water within months remained unrealized. Reality was slower, more partial, more contingent on environmental conditions. PET degradation rates of 1 to 2% annually in favorable conditions meant that a bottle would require 50 to 100 years for complete mineralization, faster than the centuries required for passive breakdown, but still measured in human lifetimes. This temporal mismatch between plastic production rates and degradation rates represented a fundamental constraint. Global plastic production exceeds 460 million tons annually and continues growing. Even if biological degradation achieved 10% annual rates, a tenfold improvement over demonstrated performance, and even if deployed at scales treating half of all plastic waste, accumulation would continue because production vastly exceeds any plausible treatment capacity. Biological degradation could slow accumulation and reduce the persistence of plastic already in the environment, meaningful benefits, but it could not solve the plastic problem alone. The second crucial lesson concerned ecological complexity and unpredictability. The testbeds were designed as controlled experiments with specific engineered bacteria degrading particular plastic types under monitored conditions. Reality quickly exceeded these boundaries. Horizontal gene transfer spread plastic degrading capability to native bacteria not deliberately released. Fungi spontaneously colonized treatment zones and participated in degradation through unknown mechanisms. Phytoplankton and zooplankton communities shifted in response to microplastic dynamics. Soil invertebrate populations declined, potentially affecting ecosystem services. These cascading effects illustrated a principle from invasion ecology. Introducing new organisms or capabilities into ecosystems initiates chains of consequences that cannot be fully predicted. Some consequences may be beneficial, such as native bacteria acquiring degradation genes and achieving more robust long-term activity than engineered strains. Others may be problematic, such as disrupted food webs or impaired ecosystem functions. Monitoring can detect these effects after they occur, but cannot reliably predict them beforehand. The third lesson addressed the gap between laboratory performance and field effectiveness. Enzymes that degraded plastic rapidly in optimized laboratory conditions, purified substrates, controlled temperature and pH, optimal nutrient concentrations, performed far less impressively in messy environmental contexts. The landfill soil was chemically complex, variable, contaminated with thousands of compounds that could inhibit enzymes or compete for active sites. Ocean water fluctuated in temperature, salinity, and nutrient availability while exposing bacteria to predation, viral attack, and UV radiation. Urban waste streams contained plastics contaminated with food residues, mixed with other materials, and varying wildly in polymer type and additive content. These real-world complexities reduced enzyme activity, slowed bacterial growth, and introduced variability that laboratory studies could not capture. The implication was that translating from laboratory proof of concept to environmental application required extensive field testing under diverse conditions, exactly what the test beds provided. But it also meant that performance gains achieved through protein engineering in the laboratory might translate to modest improvements in the field, as environmental constraints became limiting factors. The fourth lesson concerned the critical importance of system design over organism design. While substantial effort focused on engineering better bacteria and enzymes, improved catalytic activity, cold adaptation, salt tolerance, the practical success of deployments depended at least as much on engineered systems, bioreactors, wastewater ponds, bioswales, enclosures. The urban bioreactor achieved the highest degradation rates not because it used the most advanced enzymes, but because it provided optimal conditions: controlled temperature, continuous aeration, abundant nutrients, and retention times sufficient for extensive polymer breakdown. Conversely, the ocean deployment struggled not because the engineered bacteria were inadequate, but because the open water environment provided essentially no control over temperature, nutrients, or predator exposure. This suggested that future development should emphasize integrated system engineering as much as or more than continued microbial and enzymatic optimization. Enclosed photobioreactors floating in garbage patches, hybrid systems combining mechanical collection with biological treatment, or constructed wetlands processing microplastic-laden runoff might achieve better performance than simply releasing improved bacteria into uncontrolled environments. The fifth lesson addressed public engagement and social license. The urban pilot's relative success compared to the more controversial ocean and landfill deployments owed substantially to intensive community engagement, transparency, and participatory governance. Monthly public meetings, accessible data dashboards, citizen advisory boards with meaningful authority, and rapid response to community concerns built trust that sustained support even when challenges emerged. Conversely, ocean trials conducted far from population centers with limited public visibility generated skepticism and opposition from some environmental groups despite careful scientific design. This pattern aligned with broader research on public perception of genetically modified organisms. Technical safety assessments alone do not determine acceptance. Procedural justice, perceptions that decision-making is fair and inclusive, risk perception factors, including dread and controllability, trust in managing institutions, and alignment with values all shape public responses. Technologies developed and deployed through transparent, participatory processes achieved greater acceptance than those imposed by experts without community input. The urban testbed demonstrated that genetically modified organisms could be deployed in densely populated areas with public support if governance structures enabled meaningful participation. The Sustainability Advisory Board that reviewed data and made recommendations, the public comment periods that genuinely influenced decisions, and the commitment to pause or terminate operations if community opposition increased were all essential to maintaining social license. This lesson extended beyond plastic bioremediation to environmental biotechnology generally. The sixth lesson concerned economic viability and market context. Despite technical progress, biological plastic degradation remained economically marginal after 3 years. The urban bioreactor processed plastic at costs approximately 3 times higher than landfill disposal and generated recovered monomers worth less than costs. Only when accounting for avoided environmental externalities or assuming future carbon pricing did the economics approach favorability. Market adoption would require either continued cost reduction through technological improvement and scaling, policy interventions such as landfill bans or recycled content mandates that created favorable economics, or corporate willingness to pay sustainability premiums. The landfill application faced even steeper economic challenges. Treating plastic already buried generated no product revenue, only potential avoided long-term liability from groundwater contamination. Absent regulatory requirements to remediate contaminated landfills, economic incentives for biological treatment were weak. The ocean application had essentially no conventional economic case, as treating dispersed pollution in international waters provided no direct financial return. These economic realities suggested that biological plastic remediation would likely remain confined to niche applications, treating specific waste streams where conditions enabled cost recovery, unless policy frameworks changed fundamentally. The seventh lesson was methodological, concerning the value and limitations of testbed approaches. The 3 parallel testbeds, examining different deployment contexts, generated far richer understanding than any single experiment could have. Comparing performance across landfill, ocean, and urban environments revealed which challenges were universal, such as the gap between laboratory and field effectiveness, and which were context-specific, such as predation in marine systems or seasonal temperature fluctuation. This comparative approach accelerated learning and prevented overgeneralizing from single-context observations. However, testbeds also had inherent limitations. They operated at scales small enough to monitor comprehensively, but consequently could not capture dynamics that emerge only at large scales. They ran for timescales long enough to observe short-term responses, but potentially missed delayed effects or long-term evolutionary changes. They simplified deployment scenarios to enable controlled comparison, but introduced artificiality that limited real-world applicability. The ocean enclosures, for instance, provided containment impossible in actual open water deployment. Bridging from testbed scale to practical implementation would require additional experimental phases at intermediate scales. Pilot deployments processing 100 to 1,000 tons of plastic annually, treating hectare-scale landfill zones, or working in semi-enclosed coastal waters rather than the open ocean could test scalability while maintaining reasonable monitoring capacity. These intermediate trials would likely reveal new challenges invisible at testbed scale and provide more realistic performance data for decision-making. The eighth and perhaps most profound lesson concerned the relationship between technology and environmental ethics. The testbeds forced confrontation with fundamental questions about human responsibility, interventionism, and acceptable risk. Is humanity obligated to attempt remediation of pollution it has caused, even using technologies that carry their own risks? Or does humility about our limited understanding of complex systems counsel restraint from further intervention? Different stakeholders answered these questions differently. Some viewed engineered organisms as necessary tools for addressing human-caused environmental catastrophes, arguing that passive acceptance of pollution was itself an unethical abdication of responsibility. Others saw genetic modification of organisms and environmental release as violations of nature requiring rejection regardless of potential benefits. Many held intermediate positions, recognizing both the imperative to address plastic pollution and legitimate concerns about biotechnological intervention. The testbeds did not resolve these ethical disagreements, nor could they, but they did provide concrete contexts for deliberating about them, moving discussion from abstract principle to specific data about actual performance, risks, and trade-offs. This grounding in empirical reality enriched ethical discourse and enabled more nuanced positions than polarized acceptance versus rejection. The final lesson was uncomfortable. Technological solutions to environmental problems are never purely technical. They are embedded in social contexts, shaped by economic incentives, constrained by political realities, and evaluated through cultural values. The most sophisticated enzyme engineering or elegant system design cannot overcome public opposition, economic unviability, or regulatory prohibition. Conversely, even imperfect technologies may succeed if they align with social values, fit economic contexts, and navigate governance systems effectively. Biological plastic degradation represented precisely such a socio-technical system. Its future depended as much on continued public engagement, policy development, market evolution, and ethical deliberation as on further research improving bacterial strains and enzymatic activities. The testbeds had provided invaluable scientific knowledge, but they had also generated equally important insights about public perception, stakeholder dynamics, governance challenges, and the complex pathway from laboratory innovation to environmental deployment. Both forms of knowledge would be essential for determining whether and how biological approaches might contribute to addressing the global plastic pollution crisis that continued to intensify even as researchers documented bacteria slowly, incompletely, but undeniably degrading synthetic polymers in soil, sea, and city. The testbed experiments were designed from the beginning as proof-of-concept demonstrations rather than operational deployments. Yet by their third year, they had generated momentum that pushed beyond research toward real-world application. Companies contacted the research team proposing commercial partnerships. Municipalities inquired about adapting the urban model for their cities. International development agencies explored whether the technologies could address plastic pollution in regions lacking advanced waste management infrastructure. The question was no longer whether biological plastic degradation could work in principle, but how it might scale and spread to address pollution globally. The geopolitical context was evolving rapidly. In March 2022, 175 countries participating in the United Nations Environment Assembly in Nairobi adopted a historic resolution titled "End Plastic Pollution" towards an international legally binding instrument. The resolution established an intergovernmental negotiating committee tasked with developing a comprehensive global treaty addressing the full life cycle of plastics, from production through design, use, and disposal. The treaty negotiations aimed for completion by the end of 2024, with implementation frameworks emerging in subsequent years. This international mobilization created both opportunities and challenges for biological remediation technologies. The High Ambition Coalition to End Plastic Pollution, co-chaired by Rwanda and Norway, and including 71 countries by 2025, advocated for ambitious targets, including ending plastic pollution by 2040. Coalition members emphasized source reduction, decreasing virgin plastic production, alongside improved waste management and remediation. Biological degradation fit naturally into comprehensive strategies, combining prevention, collection, recycling, and treatment of legacy pollution. However, disagreements among negotiating countries revealed fundamental tensions. Petroleum-producing nations, including Saudi Arabia, Iran, and Russia, sometimes called the like-minded countries, argue that the treaty should focus exclusively on waste management and exclude regulations on plastic production. They viewed plastic manufacturing as legitimate industrial activity, not appropriately constrained by environmental treaties. Conversely, the High Ambition Coalition insisted that addressing only downstream waste while production continued growing would prove futile. The European Union progressively increased its ambition over negotiation rounds, supporting production caps, circular economy practices, and life cycle approaches. These macro-level policy debates shaped the context within which biological remediation technologies would or would not deploy. If the treaty emphasized source reduction and allowed continued plastic production only where absolutely necessary, biological degradation would serve primarily to address legacy pollution, the accumulated plastic already contaminating environments. If the treaty focused mainly on improved waste management while permitting production growth, biological technologies might be expected to treat ever-increasing waste volumes, a Sisyphean task given the rate limitations documented in the test beds. Technology transfer to developing nations emerged as a critical issue. Many countries lacking resources for expensive waste management infrastructure faced the most severe plastic pollution impacts. Rivers in Indonesia, the Philippines, and Vietnam transport massive quantities of plastic to oceans, driven by inadequate collection systems and limited recycling capacity. Coastal communities throughout Africa and Southeast Asia accumulate plastic waste on beaches and in fishing waters, threatening livelihoods and food security. For biological remediation to address global pollution, it would need to function in these contexts, not just in wealthy nations with advanced technical capacity. Technology transfer involves more than shipping equipment or sharing protocols. Effective transfer requires building local capacity, including technical knowledge to operate and maintain systems, institutional frameworks to regulate and oversee deployment, and financing mechanisms to fund initial investment and ongoing operations. The Paris Agreement on Climate Change recognized these requirements, emphasizing that technology transfer must include hardware, software, the knowledge and skills to use technologies, and institutional arrangements facilitating adoption. The testbed research team partnered with international development organizations to explore adaptation for resource-limited contexts. A pilot program in a coastal city in Southeast Asia, population 300,000, examined whether the urban bioreactor model could function with simplified infrastructure and lower costs. Rather than stainless steel vessels with automated controls, the adapted design employed concrete tanks with manual aeration through timed air pumps. Temperature was uncontrolled, varying with ambient conditions between 22 and 34 degrees Celsius. Bacterial cultures were produced locally using molasses from sugarcane processing as feedstock rather than refined glucose. The simplified system achieved degradation rates approximately half those of the original urban testbed, 6 to 8% compared to 12 to 15%, reflecting less optimal conditions. However, capital costs were reduced by 70% and operational costs by 60%, making the technology more economically accessible. The program trained 15 local technicians through a 6-month certificate course covering microbiology, wastewater chemistry, and system operation. After 1 year of operation, the facility processed approximately 3 tons of plastic daily, a modest but meaningful contribution to local waste management. Scaling required confronting the diversity of plastic waste globally. The testbeds had focused primarily on PET and polyethylene, the most abundant polymers, but plastic pollution encompasses dozens of polymer types including polypropylene, polystyrene, polyvinyl chloride, nylon, and countless others. Each requires specific enzymatic attack, and degradation rates vary dramatically. Recent research expanded the catalog of plastic-degrading organisms beyond the species used in testbeds. Advances in polyethylene degradation came from Rhodococcus bacteria isolated from naturally weathered plastic in terrestrial environments. A strain designated A34 achieved approximately 1% weight loss of low-density polyethylene powder over 30 days, comparable to engineered strains tested in landfills. Proteomic analysis identified multiple enzymes potentially responsible, including 1 multi-kappa oxidase, 1 lipase, 6 esterases, and several lipid transporters. These proteins worked collectively to oxidize and depolymerize polyethylene, suggesting that enzyme cocktails might achieve better performance than single purified enzymes. Polypropylene, the second most produced plastic globally, remained particularly recalcitrant. Its structure, similar to polyethylene but with methyl side groups, creates steric hindrance that makes enzymatic attack more difficult. No naturally occurring bacteria had been conclusively demonstrated to degrade polypropylene at appreciable rates. Protein engineering efforts focused on modifying polyethylene-degrading enzymes to accommodate the methyl groups, with limited success achieving less than 0.1% degradation over 60 days. This highlighted that biological solutions would likely remain partial, effective for some polymers but not others, requiring integration with mechanical recycling and other waste management approaches. Polystyrene presented a contrasting case where nature offered unexpected solutions through insects rather than bacteria. Mealworms, larvae of the beetle Tenebrio molitor, consume expanded polystyrene foam, commonly called styrofoam, degrading approximately 50 to 65% of ingested material. The degradation mechanism involves gut enzymes generating reactive oxygen species that cause autoxidation, cleaving polymer chains and converting benzene rings to quinone compounds. Polymer residues in mealworm feces show reduced molecular weight and oxidation signatures, confirming chemical degradation. Research extended polystyrene degradation beyond mealworms to superworms, larvae of Zophobas morio, which also consume and degrade styrofoam. Respirometry tests showed that up to 37% of ingested polystyrene carbon was mineralized to carbon dioxide within 16 days. Antibiotic treatment that suppressed gut bacteria eliminated degradation capability, proving that gut microbiota rather than insect enzymes alone were responsible. This finding suggested that isolating and cultivating the bacterial species from mealworm and superworm guts could provide polystyrene-degrading organisms for technological applications. However, more recent studies challenged whether mealworms actually achieve complete chemical degradation. Critical analyses found that while mealworms fragment polystyrene into smaller pieces and eliminate it from their digestive tracts, the material may not undergo true depolymerization back to monomers or complete mineralization. The controversy highlighted the importance of rigorous chemical analysis using techniques like gel permeation chromatography to measure molecular weight changes and mass spectrometry to identify degradation products, not simply observing weight loss or disappearance. The debate continued as research teams pursued definitive evidence. Enzyme engineering was accelerating through machine learning and artificial intelligence. Traditional directed evolution required screening thousands of enzyme variants to identify improved versions, a labor-intensive process. Machine learning offered potential to predict which mutations would enhance desired properties, reducing the number of variants requiring experimental testing. A platform published in January 2025 integrated cell-free DNA assembly, cell-free gene expression, and functional assays to rapidly map fitness landscapes across protein sequence space. The system evaluated 1,217 enzyme variants in nearly 11,000 unique reactions, generating comprehensive data for training machine learning models. Augmented ridge regression algorithms predicted enzyme variants optimized for 9 different chemical reactions, demonstrating 1.6 to 42-fold improved activity compared to parent enzymes. Applied to plastic-degrading enzymes, similar approaches could potentially identify variants with enhanced thermostability, broader substrate specificity, or greater resistance to inhibition by contaminants. CRISPR genome editing tools enabled precise metabolic engineering in bacteria. Rather than inserting degradation genes randomly into genomes and hopping for stable expression, CRISPR systems could target specific chromosomal locations, delete competing pathways that waste cellular resources, and fine-tune expression levels by editing promoter sequences. Applications in non-model bacteria, including marine and soil species difficult to manipulate with traditional genetic methods, expanded the repertoire of organisms available for engineering. CRISPR-based metabolic engineering in Escherichia coli and other industrial bacteria demonstrated production of high-value chemicals, biofuels, and pharmaceuticals with yields approaching commercial viability. The same tools could optimize plastic-degrading bacteria, eliminating unnecessary genes to reduce metabolic burden, overexpressing rate-limiting enzymes in degradation pathways, and introducing genes for completely mineralizing degradation products to carbon dioxide. These engineered organisms would be more efficient than naturally evolved species, but would also raise intensified regulatory concerns about environmental release. Synthetic microbial consortia offered an alternative to engineering single superdegrader organisms. Natural microbial communities achieve complex degradation through division of labor, with different species performing specialized tasks. Designing synthetic consortia mimicking this strategy could distribute plastic degradation across multiple organisms, each optimized for specific steps. One species might attach to plastic surfaces and initiate oxidation. Another could break oxidized polymers into oligomers. A third could metabolize oligomers to carbon dioxide and biomass. This modular approach offered several advantages. Individual organisms could be simpler and easier to engineer than single species performing all steps. Metabolic burden would be distributed, potentially allowing faster overall degradation than possible in single species. Consortia might be more robust, with multiple species providing functional redundancy if one population declined. Design principles included functional complementarity, ensuring each organism contributed distinct capabilities, synergistic advantage from cross-feeding of metabolites between species, and population control mechanisms maintaining stable ratios. However, maintaining designed consortia in environmental conditions pose challenges. Engineered population ratios established in the laboratory might shift unpredictably in fields, oceans, or landfills where organisms face different selective pressures. Native species could invade consortia, disrupting designed interactions. Adaptive evolution over time might alter organism properties, including loss of engineered traits if they imposed metabolic costs without immediate fitness benefits. Research on adaptive laboratory evolution provided insights into these dynamics. Experiments involving Escherichia coli in environments with alternating carbon sources demonstrated that populations could become either generalists, growing well on multiple substrates, or specialists, with subpopulations adapted to specific substrates coexisting. The evolutionary outcome depended on switching frequency and substrate combinations. For plastic-degrading consortia, Similar dynamics might determine whether engineered species maintained designed functions or evolved toward different ecological strategies. Metagenomic sequencing of environmental samples provided powerful tools for monitoring how released organisms and their genes spread and evolve. Next-generation sequencing could characterize entire microbial communities, identifying thousands of species and their genes from soil, water, or biofilm samples. Comparing metagenomes before and after deployment of engineered bacteria revealed community changes attributable to the intervention. Tracking specific genes over time documented whether they transferred to native species and whether they underwent mutations affecting function. Whole genome shotgun sequencing analyzed total DNA from environmental samples, providing comprehensive information on genomic diversity and functional potential. This approach revealed not just which organisms were present, but what metabolic capabilities they possessed, including genes for plastic degradation, antibiotic resistance, or other traits of interest. The data intensity was enormous, with a single environmental sample potentially generating hundreds of gigabytes of sequence data requiring sophisticated bioinformatics for analysis. But the insights enabled adaptive management, adjusting deployment strategies based on monitoring what actually occurred in complex environments. The expansion beyond testbeds was occurring not through centralized planning, but through distributed experimentation by research groups, companies, municipalities, and international programs worldwide. Each adapted the core concept, biological plastic degradation, to local contexts, constraints, and priorities. This decentralized innovation generated diversity in approaches and rapid accumulation of practical knowledge, but also created challenges for learning across projects and establishing best practices. The emerging global treaty framework could potentially facilitate coordination, technology transfer, and knowledge sharing, channeling distributed efforts toward collective impact on plastic pollution that individual projects alone could never achieve. Chapter 23: The Second Generation. The lessons from first-generation testbeds catalyzed redesign of biological plastic degradation systems, incorporating improvements in organism engineering, deployment strategies, monitoring protocols, and governance frameworks. Second-generation projects launching in years 4 and 5 aimed to address limitations identified through 3 years of intensive field testing. The emphasis shifted from demonstrating basic feasibility to optimizing performance, reducing costs, minimizing ecological risks, and building scalable implementation pathways. Organism engineering entered a new phase, leveraging computational tools and expanded enzyme catalogs. Rather than relying solely on directed evolution through random mutagenesis and screening, researchers employed machine learning to predict beneficial mutations before experimental testing. Training datasets included crystal structures of hundreds of hydrolase enzymes, biochemical properties of thousands of characterized variants, and activity measurements from testbed deployments under diverse environmental conditions. The machine learning models identified enzyme positions where specific amino acid substitutions would likely increase thermostability without sacrificing catalytic efficiency. For PTAs, computational predictions suggested 12 positions where substitutions could simultaneously increase optimal temperature from 30 degrees to 45 degrees and extend the temperature range over which the enzyme retained at least 50% of peak activity. Laboratory testing confirmed that a variant incorporating 7 of these substitutions achieved the predicted properties, demonstrating the predictive power of the approach. Substrate specificity was engineered to expand enzyme capabilities. Wild-type PTAs preferentially attacks amorphous regions of PET where polymer chains are disordered, while crystalline regions with aligned chains resist degradation. Computational modeling of enzyme-substrate interactions identified active site modifications that could accommodate crystalline PET. The modified enzyme showed 23% activity on highly crystalline PET compared to amorphous, versus 4% for wild-type enzyme. This improvement enabled degradation of weathered plastic in environments where crystallinity increases over time through thermal annealing and photooxidation. Enzyme cocktails mixing multiple catalytic activities outperformed single purified enzymes. Rhodococcus bacteria degrading polyethylene produce not one enzyme but coordinated sets, including oxidases generating hydroxyl groups on polymer chains, esterases cleaving resulting ester bonds, and transporters moving degradation products into cells. Reconstituting this multi-enzyme system in vitro by combining purified proteins achieved degradation rates approaching whole cell systems while avoiding complications of maintaining living bacteria. The optimal enzyme mixture compositions varied with polymer type and environmental conditions. For PET at 30 degrees Celsius in neutral pH water, a cocktail combining PTase, MHETase, and a carboxylsterase in 2:1 molar ratios maximized degradation. For polyethylene in soil at 15 degrees, a different combination emphasizing oxidases and hydroxylases proved most effective. Machine learning analyzed data from hundreds of cocktail compositions tested under varied conditions, generating predictive models that recommended optimal mixtures for specific deployment scenarios. Immobilization techniques advanced to stabilize enzymes and enable recovery and reuse. Early approaches simply mixing free enzymes into water or soil saw rapid inactivation through denaturation, proteolytic degradation, or adsorption to surfaces. Second-generation systems attached enzymes to solid supports including silica nanoparticles, magnetic beads, or porous polymers. The supports protected enzymes from inactivation while allowing substrate access to active sites. Magnetic beads enabled enzyme recovery from reaction mixtures using magnetic fields, allowing reuse over multiple cycles. Alginate bead encapsulation, tested in ocean trials with living bacteria, was adapted for pure enzymes. Beads containing enzyme cocktails plus stabilizing agents like polyethylene glycol and trehalose maintained activity for months in seawater, far longer than free enzymes lasting days. The beads could be formulated to float or sink, enabling targeted treatment of plastic at specific ocean depths. Degradation of the alginate matrix over weeks to months released enzymes gradually, extending treatment duration from single applications. Deployment strategies evolved toward hybrid systems combining biological, mechanical, and chemical approaches. Pure biological degradation remained slow, but coupling it with mechanical fragmentation accelerated overall processing. Waste plastic was shredded to millimeter-scale particles, dramatically increasing surface area accessible to enzymes. The particles were then treated with enzyme cocktails or bacterial consortia in bioreactors. Degradation products were separated and either fed back into biological treatment for complete mineralization or recovered as chemical feedstocks. Advanced oxidation processes using ozone, hydrogen peroxide, or UV light pre-treated plastic surfaces, creating hydroxyl and carbonyl groups that enhanced enzymatic attack. The combined chemical-biological approach achieved 15 to 20% degradation in 24 hours, compared to 3 to 5% for biological treatment alone over the same period. However, chemical oxidation consumed energy and reagents, increasing operational costs and environmental footprint. Life cycle assessment was required to determine whether the accelerated degradation justified the additional resource inputs. Ocean deployment strategies moved away from releasing organisms into open water toward contained floating systems. Second-generation designs resembled floating bioreactors, essentially the urban systems adapted for marine environments. Large, flexible bladders, 10 to 50,000 liters capacity, floated semi-submerged in the garbage patch. Plastic debris was collected from surrounding waters through surface skimming and pumped into the bladders where bacterial consortia or enzyme cocktails degraded it over weeks. Treated water was filtered before discharge, and residual plastic was returned to the bladder for additional treatment cycles. These floating systems achieved physical containment, avoiding gene escape concerns that plagued open water releases. Solar panels provided power for pumps and filtration, enabling autonomous operation between monthly service visits. GPS tracking and satellite communication allowed remote monitoring of bladder locations and status. 30 units deployed across the garbage patch processed an estimated 500 tons of plastic annually, a tiny fraction of the garbage patch mass, but sufficient to provide operational data and economic assessment. Landfill deployments integrated biological treatment with landfill mining, excavating buried waste for resource recovery. Traditional landfill mining recovered metals and sometimes combustible material for energy generation, but plastic was typically reburied or sent to incineration. Second-generation projects included biological treatment modules, where excavated plastic underwent bacterial degradation in above-ground treatment cells for 6 to 12 months before residual material was assessed for recycling or disposal. The extended treatment time, far longer than practical in conventional recycling facilities, enabled more complete degradation, including partial breakdown of recalcitrant polymers like polypropylene. Urban systems expanded from single demonstration facilities to networks of smaller distributed units. Rather than one centralized bioreactor processing all plastic waste, second-generation cities deployed 10 to 20 neighborhood-scale units processing 5 to 10 tons daily. This distributed approach reduced transportation distances and costs while providing redundancy. A failure at one unit did not disable the entire system. Standardized modular design enabled fabrication at industrial scale and rapid deployment to new locations. Integration with existing waste infrastructure deepened. Bioactive coatings applied to the interior of conventional garbage trucks initiated plastic degradation during transport from collection points to processing facilities. The coatings contained immobilized enzymes that began surface modification during the hours or days waste spent in transit, reducing the treatment time required at centralized facilities. Wastewater treatment plant integration expanded beyond polishing ponds to include biological filtration units targeting microplastics in primary and secondary treatment stages. Smart monitoring systems generated real-time operational data enabling adaptive management. Sensors measuring temperature, pH, dissolved oxygen, nutrient concentrations, and bacterial cell density transmitted data continuously to cloud-based analysis platforms. Machine learning algorithms detected anomalies indicating equipment malfunctions, contamination, or suboptimal conditions, triggering automated adjustments or alerting operators. Historical data accumulated over months and years trained predictive models that optimized operating parameters for maximum degradation efficiency at minimum cost. Metagenomic monitoring became standard practice rather than occasional research sampling. Automated sample collection systems obtained water or soil specimens from treatment zones daily or weekly. DNA extraction and sequencing occurred at commercial service laboratories, with results typically returned within 1 week. Bioinformatic analysis tracked engineered organism populations, detected gene transfer to native species, and characterized overall community composition. This intensive surveillance enabled early detection of containment failures or ecological impacts, allowing rapid response before problems escalated. Economic models were refined based on 3 years of actual operational data rather than projected estimates. Capital costs for urban bioreactor facilities averaged $1.2 million per ton per day processing capacity, approximately 20% below initial projections due to design optimization and economies of scale in equipment manufacturing. Operating costs averaged $150 per ton of plastic processed, approximately 30% above projections, driven primarily by higher-than-expected bacterial culture production costs and more frequent equipment maintenance. Revenue from recovered monomers partially offset costs. At current degradation efficiencies averaging 15%, and monomer purity of 98 to 99%, facilities generated approximately $50 per ton of feedstock plastic in monomer sales. Additional revenue came from avoided landfill disposal fees, approximately $60 per ton, and from voluntary sustainability markets where companies purchased credits representing verified plastic remediation. Combined revenues of approximately $110 per ton left a funding gap of roughly $40 per ton, requiring subsidy or higher treatment fees. Break-even scenarios projected that degradation efficiency improvements to 25% and monomer purity increases to 99.5% would generate revenue exceeding costs, making the technology self-sustaining. Alternatively, carbon pricing mechanisms that monetized avoided greenhouse gas emissions from displaced virgin plastic production could provide additional revenue of $30 to $50 per ton, achieving financial viability even at current technical performance. Governance frameworks adapted to address lessons from first-generation deployments. Regulatory approvals for second-generation projects required more comprehensive environmental risk assessments, including 5-year ecological monitoring rather than 3 years, extended spatial monitoring zones reaching 100 meters from release points rather than 50, and mandatory contingency plans for responding to detected gene transfer or non-target effects. Kill switches became mandatory rather than optional, with requirements for redundant independent mechanisms to prevent evolutionary escape. Public engagement was formalized through participatory governance structures. Advisory boards included not just hand-selected stakeholders, but randomly sampled citizens representing demographic diversity, similar to citizen assemblies used in some democracies for deliberating complex policy issues. These boards received extensive technical briefings, heard from diverse expert and advocacy perspectives, and made binding recommendations on deployment decisions. The model aimed to combine scientific expertise with democratic legitimacy, technical assessment with public values. International coordination improved through knowledge-sharing networks and technology transfer programs. Regional centers of excellence established in Africa, Asia, and Latin America provided training, technical assistance, and shared equipment access for countries developing biological remediation programs. Open-source databases compiled monitoring data, organism performance metrics, and operational best practices from projects worldwide. Annual conferences brought together researchers, practitioners, regulators, and community representatives to exchange lessons and coordinate research priorities. The second generation represented maturation from experimental research toward operational technology. Performance was improving, costs were declining, governance was becoming more robust, and deployment was expanding geographically and numerically. Yet fundamental challenges remained. Degradation rates, while better than first generation, still required months to years for substantial plastic breakdown. Ecological risks, though managed more carefully, could not be eliminated entirely. Economics remained marginal, dependent on subsidies or optimistic projections about future improvements. The technology had proven that biological plastic degradation could work, but whether it should scale to become a primary strategy for managing plastic pollution worldwide remained vigorously debated among scientists, policymakers, and publics concerned about both the plastic crisis and the risks of biotechnological intervention. Chapter 24, PATHWAYS Forward. 5 years after the first bacterial inoculations in landfill cells, ocean enclosures, and urban bioreactors, participants in the biological plastic degradation research community gathered to assess progress and chart future directions. The accumulated evidence permitted answering the foundational question: can microorganisms meaningfully reduce plastic pollution? The answer was nuanced. Yes, bacteria and fungi can degrade synthetic polymers at measurable rates under favorable conditions. No, current technologies cannot eliminate plastic pollution or even keep pace with continued plastic production and dispersal. The relevant question was where and how biological degradation fit into comprehensive strategies addressing plastic across its full life cycle. The emerging consensus recognized biological degradation as one tool among many, not a silver bullet. Source reduction, decreasing virgin plastic production and use, remained paramount. Product redesign, creating plastic items that were reusable, recyclable, or biodegradable by design, would prevent pollution more effectively than any remediation technology. Improved collection and mechanical recycling infrastructure, particularly in developing regions where systems were inadequate, would recover plastic before it entered environments. Biological degradation would complement these approaches by treating plastic that evaded collection, degrading items unsuitable for mechanical recycling, and remediating legacy pollution accumulated over decades. The Global Plastics Treaty negotiations crystallized this integrated vision. The final text, still under negotiation in late 2025, with completion anticipated in early 2026, outlined comprehensive measures. Provisions addressed plastic production through monitoring and reporting requirements, with aspirational targets for reducing virgin polymer manufacturing. Product design standards would phase out unnecessary single-use items and require recyclability for remaining products. Extended producer responsibility mechanisms would require manufacturers to finance collection and recycling infrastructure. Waste management articles in the treaty specifically mentioned advanced biological and chemical recycling technologies as complement to mechanical recycling. Funding mechanisms would support technology development and deployment, particularly in developing countries lacking capital for expensive infrastructure. The treaty framework created favorable conditions for biological degradation to scale, while emphasizing it could not substitute for upstream interventions, reducing plastic production and consumption. Technical development pathways focused on addressing persistent limitations. Enzyme engineering continued through machine learning-accelerated directed evolution and rational design. Targets included further increasing thermostability, enabling activity across broader pH ranges, improving tolerance to salt and contaminants, and expanding substrate specificity to include recalcitrant polymers like polypropylene. Some research groups pursued entirely novel enzyme classes, identified through metagenomic mining of environmental samples from plastic-contaminated sites worldwide. Organism engineering emphasized metabolic optimization and regulatory control. CRISPR-based editing fine-tuned expression of degradation genes, deleted competing pathways that wasted cellular resources, and introduced sensors allowing bacteria to regulate their activity in response to environmental conditions. Synthetic consortia design aimed to create stable cooperative communities where labor division enhanced overall performance beyond what single-species systems achieved. Population control mechanisms maintained designed species ratios despite environmental fluctuations. However, a counter-movement questioned the wisdom of continued organism engineering complexity. Critics argued that highly engineered organisms with multiple modifications faced greater regulatory barriers, increased likelihood of fitness costs, reducing environmental performance, and elevated public concern about releasing Frankenstein bacteria into nature. They advocated for simpler approaches, using minimally modified organisms or even wild-type species identified through bioprospecting and deployed with appropriate containment. Deployment innovation explored distributed small-scale systems rather than centralized large facilities. Neighborhood-level processing units integrated into communities reduced transportation costs and increased public visibility and acceptance. Mobile treatment systems on trucks or ships could deploy flexibly to pollution hotspots rather than requiring permanent infrastructure. Modular designs enabled incremental capacity expansion, matching waste generation growth without massive upfront capital requirements. Coupling biological degradation with materials recovery offered economic benefits. Rather than viewing waste plastic purely as contamination requiring disposal, integrated systems recovered monomers for repolymerization, oligomers for chemical feedstocks, or residual carbon for incorporation into biomaterials. This circular approach aligned with broader movement towards circular economy principles, minimizing waste and maximizing resource productivity. Monitoring technology advanced toward real-time, non-invasive assessment. Spectroscopic sensors could detect plastic degradation products in water or soil at parts per billion sensitivity without requiring sample collection and laboratory analysis. Environmental DNA sampling and portable sequencing devices enabled field monitoring of microbial communities and detection of engineered organisms within hours. Satellite remote sensing, combined with machine learning, could potentially identify accumulations of microplastic in surface waters and track changes over time, providing large-scale monitoring complementing intensive local surveillance. Economic pathways to viability diverged between developed and developing regions. In wealthy countries, willingness to pay for environmental services, carbon pricing schemes, and extended producer responsibility funding could support biological degradation as part of comprehensive waste management. In developing nations, simpler lower-cost systems integrated with livelihood generation, for example training and employing local residents as system operators, could align environmental benefits with economic development. International financing mechanisms under discussion in treaty negotiations included dedicated funds for technology transfer and capacity building. Developed countries would contribute financing to support deployment in developing nations, similar to climate finance under the Paris Agreement. Private sector engagement through impact investing and sustainability-linked finance could mobilize additional capital beyond government funding. Regulatory harmonization aimed to prevent a patchwork of inconsistent national standards that could hinder technology deployment and create competitive distortions. The treaty framework would likely establish minimum safety and performance standards for biological remediation, while allowing countries to impose stricter requirements matching their risk tolerance and priorities. Regional cooperation could pool regulatory capacity, enabling countries with limited expertise to leverage assessments conducted by well-resourced agencies. Ethical deliberation continued about the acceptability of releasing engineered organisms into the environment. Deep disagreements persisted between those viewing such releases as necessary responses to human-caused environmental crises and those seeing them as unacceptable interference with natural systems. Rather than attempting to resolve these disagreements, governance frameworks aimed to enable coexistence through geographic differentiation, allowing deployment in jurisdictions with permissive policies while respecting bans in others, and procedural safeguards ensuring decisions reflected democratic input, not just expert judgment. Indigenous perspectives, largely absent from initial testbed design, were increasingly incorporated into project planning. Indigenous communities often bore disproportionate impacts from plastic pollution, affecting traditional territories and marine resources, while having minimal voice in technology decisions. Participatory processes, including free, prior, and informed consent requirements, ensured that deployments affecting indigenous lands or waters proceeded only with community approval. Traditional ecological knowledge about local ecosystems informed monitoring protocols and risk assessment. Educational initiatives aimed to build public understanding of both plastic pollution and biotechnology. School curricula incorporated modules on plastic chemistry, microbiology, and environmental systems. Public science communication emphasized transparent explanation of how biological degradation works, what risks it entails, and how governance processes balance benefits against hazards. Hands-on citizen science programs engaged community members in monitoring, sample collection, and data interpretation, building investment in technology outcomes. Alternative futures scenarios explored possible long-term trajectories. An optimistic scenario envisioned the global treaty catalyzing dramatic plastic production reductions, approaching 50% decrease by 2040, alongside comprehensive collection and recycling infrastructure. Biological degradation would treat legacy pollution and handle non-recyclable materials, processing perhaps 10% of global plastic waste by 2050. Cumulative effects over decades could substantially reduce environmental plastic loads, particularly in oceans where floating debris would be actively remediated. A pessimistic scenario saw treaty negotiations collapse amid disagreements between producer and consumer nations. Plastic production would continue growing, reaching 700 million tons annually by 2040. Collection and recycling infrastructure would expand slowly, constrained by insufficient financing. Biological degradation deployment would remain limited to niche applications in wealthy regions, treating perhaps 2% of waste globally. Plastic pollution would continue accumulating in all ecosystems, with increasingly severe ecological and health consequences. Intermediate scenarios between these extremes seemed most probable. The treaty would establish frameworks and aspirational goals, but implementation would vary widely by country. Production would grow but more slowly than historical trends, perhaps reaching 550 million tons by 2040. Biological degradation would deploy at meaningful scales in some regions while remaining absent in others, treating perhaps 5% of global plastic waste by mid-century. Environmental plastic loads would stabilize or decrease slightly rather than growing exponentially, a modest but meaningful improvement. The pathway forward was ultimately social and political as much as technical. The testbeds and subsequent deployments had demonstrated that biological plastic degradation could work at achievable cost with manageable risk. Whether humanity would choose to deploy these technologies at scales sufficient to meaningfully reduce pollution depended on governance decisions made in treaty negotiations, national legislatures, corporate boardrooms, and municipal councils. It depended on public willingness to accept novel technologies despite uncertainties. It depended on sustained research funding, technology transfer to where pollution was worst, and integration into comprehensive strategies addressing plastic across its life cycle. 5 years of intensive research and deployment had transformed biological plastic degradation from speculative laboratory curiosity to operational technology with demonstrated capabilities and quantified limitations. Bacteria could indeed degrade synthetic polymers, slowly but measurably, offering new tools for addressing one of the defining environmental crises of the 21st century. The decades ahead would reveal whether these tools would be widely adopted, carefully refined, and responsibly integrated into global efforts to end plastic pollution. The science had delivered possibilities. Politics, economics, and collective human choice would determine what became of them. The technical capabilities of engineered bacteria represented only one dimension of addressing plastic pollution. Equally critical were the human behaviors producing hundreds of millions of tons of plastic waste annually, the corporate decisions perpetuating linear production-consumption disposal models, and the policy frameworks either enabling or constraining these patterns. Biological degradation might offer tools for treating pollution, but preventing it required transforming how humanity related to synthetic materials that had become ubiquitous across every domain of modern life. Consumer behavior studies revealed the complex psychology underlying plastic use. Environmental concern, the emotional and cognitive awareness that plastic pollution damages ecosystems and harms organisms, was widespread across demographics. Surveys in multiple countries documented that 70 to 85% of respondents expressed concern about plastic pollution and supported efforts to reduce it. However, concern alone poorly predicted actual behavior change. The attitude-behavior gap, where people professed environmental values but continued unsustainable practices, reflected structural barriers and psychological complexities. Perceived consumer effectiveness, the belief that individual actions can meaningfully contribute to environmental sustainability, emerged as a key psychological driver determining whether concern translated to behavior change. When individuals believed their choices mattered, purchasing products with minimal packaging, carrying reusable bags and bottles, avoiding single-use items, they were significantly more likely to adopt plastic reduction behaviors. Conversely, when people felt their individual actions were inconsequential compared to systemic forces like corporate production decisions, they experienced futility that paralyzed action. Research examining single-use plastic reduction in Thailand documented these dynamics through the theory of planned behavior framework. The study found that attitudes, the degree to which individuals evaluated plastic reduction favorably, significantly predicted behavior. Subjective norms, perceived social pressure from family, friends, and community to reduce plastic also influenced actions. Perceived behavioral control, the anticipated ease or difficulty of reducing plastic use, was particularly important. When reusable alternatives were readily available and affordable, behavioral control was high and plastic reduction increased. When alternatives were scarce or expensive, control was low and behavior change was limited regardless of attitudes. These findings highlighted that consumer behavior change required addressing structural barriers, not just individual motivation. Cost represented a major obstacle. Reusable containers, bags, and bottles required upfront investment, while single-use plastic was often free or extremely cheap. Studies found that cost of reusable alternatives negatively influenced plastic reduction attitudes, with higher prices discouraging adoption. Availability was equally critical. If stores did not stock package-free products or bulk buying options, consumers could not reduce plastic packaging even if motivated. The social practice perspective examined plastic use as embedded in everyday routines rather than isolated individual choices. Research following 20 adults through diary records and workshops revealed that reducing single-use plastic was often practically difficult within existing social systems. Food provisioning practices relied on packaged goods available in supermarkets. Convenience food for busy schedules came wrapped in plastic. Social norms around cleanliness and food safety reinforced plastic packaging as hygienic. Changing these practices required not just individual willpower, but reconfiguring the material infrastructures and social expectations structuring daily life. Digital communication strategies showed promise for accelerating behavior change at scale. Social media campaigns creating challenges that invited users to reduce plastic consumption for specified periods, share experiences, and encourage others generated engagement and awareness. Gamification incorporating points, levels, and rewards for sustainable actions leveraged psychological motivation. Transparency in communicating product materials, environmental impacts, and disposal methods enhanced consumer trust and loyalty towards sustainability-committed brands. The research suggested that digital engagement combined with educational content and incentives could significantly reduce single-use plastic reliance. However, the emphasis on individual consumer responsibility risked obscuring the structural forces shaping consumption patterns. Consumers operated within systems designed to promote disposability, with pervasive marketing reinforcing cultures of convenience and single-use products. Effective solutions required shifting focus from individual blame to corporate accountability and system-level redesign. This critique aligned with broader environmental justice perspectives, emphasizing how pollution burdens fell unequally across communities. The United Nations report titled "Neglected: Environmental Justice Impacts of Plastic Pollution" documented how vulnerable communities disproportionately bore environmental degradation from plastics. Marginalized populations experienced severe impacts at all stages of the production cycle, from extracting raw materials through manufacturing to consumption and disposal. Indigenous peoples faced displacement for oil drilling and natural gas extraction via fracking, contaminating potable water sources. Coastal communities dependent on marine resources confronted livelihoods threatened by plastic-infested fisheries. Informal waste workers in developing nations sorted plastic waste by hand with minimal protection, exposing them to toxic chemicals and health hazards. These environmental injustices reflected power imbalances, where economically and politically marginalized groups lacked voice in decisions about plastic production, use, and disposal. The report emphasized that environmental justice required not just technical solutions, but ensuring affected communities participated in decision-making and had access to credible judicial systems for seeking remedies. This perspective challenged top-down technology deployment models, arguing that communities on pollution frontlines must help design and control remediation strategies affecting their environments and health. Corporate sustainability commitments represented another critical human dimension. Major consumer goods companies had announced ambitious plastic reduction targets, often pledged to achieve by 2025 or 2030. Common commitments included making packaging 100% recyclable, reusable, or compostable, incorporating specific percentages of recycled content, reducing virgin plastic use by defined amounts, and supporting collection infrastructure development. These voluntary pledges generated positive publicity and responded to consumer and investor pressure for sustainability. However, tracking showed that most companies were falling short of stated targets. Systemic challenges, including lack of recycling infrastructure for flexible plastic films, contamination, reducing recycled material quality, and higher costs of recycled versus virgin plastic impeded progress. Some companies responded by investing in circular economy infrastructure, funding collection and sorting systems, developing advanced recycling partnerships, and supporting plastic offset schemes. These initiatives aim to complement reduction efforts by ensuring that plastic still used would be recovered and recycled. Plastic offset schemes, emerging as one corporate strategy, involved companies funding plastic waste removal equivalent to their plastic footprint. Similar to carbon offsets, plastic credits were generated by projects collecting and recycling waste, particularly in regions with inadequate infrastructure. Critics argued that offsets allowed companies to continue producing plastic while appearing sustainable, potentially delaying more fundamental business model changes. Supporters contended that offsets mobilized finance for waste management where it was desperately needed, generating real environmental benefits even if not addressing root causes. Extended producer responsibility programs represented more systemic corporate accountability mechanisms. EPR schemes required producers to finance end-of-life management of their products, creating incentives to reduce material use, design for recyclability, and invest in collection and recycling infrastructure. Well-designed EPR programs significantly improved recycling rates and reduced plastic pollution. In the United States and globally, EPR for plastic packaging was expanding, with successful programs providing blueprints for broader application. Key factors determining EPR effectiveness included scope and coverage, with comprehensive schemes avoiding loopholes that let producers circumvent responsibility, adequate fees that reflected true end-of-life costs rather than nominal charges, and robust enforcement ensuring compliance. Poorly designed EPR schemes with limited coverage, insufficient fees, or weak enforcement proved ineffective and sometimes counterproductive. The regulatory design details mattered enormously for whether EPR achieved intended accountability or merely provided greenwashing opportunities. Policy interventions beyond EPR included bans on specific single-use plastic items, recycling targets and mandates, and procurement requirements favoring recycled content. Single-use plastic bans implemented in numerous jurisdictions targeted items like bags, straws, stirrers, and food containers. Evaluations found that well-enforced bans could substantially reduce consumption of targeted items. However, effectiveness varied with enforcement capacity, availability of alternatives, and comprehensiveness, avoiding loopholes. Bans also raised public awareness about plastic pollution, signaling government commitment, and potentially catalyzing broader behavior change beyond targeted items. The regulatory effectiveness question probed whether current policies were adequate to address plastic pollution at necessary scale and speed. Most analysts concluded that existing regulations showed limited effectiveness due to fragmented approaches and failure to address systemic issues. Regulations that focused only on waste management while allowing continued production growth faced inherent limits. Policies emphasizing individual consumer responsibility without constraining plastic production at source could not curb pollution given the massive scale of manufacturing. Comprehensive approaches combining production caps, product design standards, waste management requirements, and circular economy incentives were needed but remained rare. Political influence from fossil fuel and petrochemical industries represented a major barrier to stronger regulation. The plastics industry, closely tied to oil and gas sectors, mounted aggressive lobbying against regulations that would limit production or mandate sustainability requirements. Analysis of the global plastics treaty negotiations found that fossil fuel and chemical industry lobbyists dramatically outnumbered environmental advocates and even exceeded national government delegations. At INC 5, approximately 220 industry lobbyists were registered, more than any single country's delegation. Industry representatives infiltrated negotiations, pressuring officials to weaken production limits and promote voluntary commitments over binding regulations. Reports documented intimidation tactics, including lobbying national delegations to replace independent technical experts with industry-aligned representatives. Major companies including ExxonMobil, Dow, BASF, and SABIC were actively expanding plastic production capacity even while participating in treaty negotiations, viewing plastics as profitable replacement for fossil fuels in energy and transport sectors transitioning to clean alternatives. Industry projections anticipated plastic production doubling or tripling by mid-century. Civil society groups characterized this dynamic as corporate capture of environmental governance, with foxes guarding the henhouse. They called for strong conflict of interest policies, disclosure requirements for lobbying activities, and procedures ensuring affected communities rather than industry representatives shaped treaty provisions. The struggle over the plastics treaty exemplified broader tensions between corporate power seeking to preserve profitable but environmentally destructive business models and social movements demanding transformative change. Recent health research added urgency to demands for action. Studies published in Nature Medicine and other leading journals documented alarming bioaccumulation of microplastics and nanoplastics in human tissues, including brains, livers, and blood. Autopsies of decedent human brains found microplastic concentrations 12 times higher than in liver tissue, with polyethylene particles most abundant. The data suggested that plastic accumulation in brains was increasing over time, with samples from individuals who died more recently containing higher concentrations. Mechanistic studies revealed how circulating microplastics caused harm. Particles in bloodstreams were phagocytosed by immune cells, which then caused obstructions in brain capillaries, forming thrombi that reduced blood flow and impaired neurological function. Mice exposed to microplastics exhibited behavioral abnormalities and reduced locomotor abilities, lasting weeks. The research demonstrated that microplastics disrupted tissue function not necessarily through direct chemical toxicity, but through physical mechanisms interfering with circulation and cell biology. These findings transformed microplastic pollution from abstract environmental concern to concrete human health threat. The accumulation of evidence about environmental injustice, corporate responsibility failures, regulatory inadequacy, political obstruction, and health impacts created intensifying pressure for fundamental system change. Biological plastic degradation technologies could contribute to managing pollution, but they operated within and could not substitute for addressing the human behaviors, economic structures, power dynamics, and political processes producing the crisis. The human dimension required recognizing that plastic pollution resulted from specific choices by identifiable actors, corporate executives, policymakers, consumers, that could be changed through education, regulation, economic incentives, and social movements demanding transformation. Technology alone was insufficient. Comprehensive solutions required engaging the full complexity of human systems producing and potentially reducing plastic pollution. Chapter 26: SYSTMS and Complexity. Standing before the assembled researchers at the 5-year symposium, Dr. Sarah Miller, an ecological systems theorist who had observed the testbed programs from design through deployment, delivered a challenging presentation. Her central thesis was uncomfortable: the persistent framing of plastic pollution as a technical problem requiring technical solutions fundamentally misunderstood the nature of the challenge. Plastic pollution was not a discrete problem with identifiable causes and correctable solutions, but rather a symptom of systemic dysfunction in how industrial civilization related to materials, waste, and planetary boundaries. Systems thinking provided frameworks for grasping this complexity. Unlike linear thinking that traced simple cause-effect chains, plastic production causes plastic pollution. Systems perspectives recognized that environmental challenges emerged from interactions within complex adaptive systems involving ecological, social, economic, and political dimensions. These systems exhibited characteristic properties, including feedback loops, where effects influenced causes, nonlinear dynamics, where small changes could trigger large effects, emergence, where system-level patterns arose from countless micro-level interactions, and resilience or fragility depending on system structure. Applying systems thinking to plastic pollution revealed multiple interconnected feedback loops. Positive feedback loops amplified plastic production. As production increased, economies of scale reduced costs, making plastic cheaper and more competitive with alternatives, which drove further production increases. Investment in plastic manufacturing infrastructure created sunk costs, incentivizing continued operation to recover investments. Accumulating environmental damage from plastic pollution generated research funding and public concern that sometimes paradoxically enabled industry to position itself as part of solutions through voluntary commitments and technology initiatives, deflecting regulatory pressure while maintaining production. Negative feedback loops theoretically could stabilize or reduce plastic production, but were often weak or delayed. Environmental regulations constraining plastic faced industry lobbying that delayed or weakened implementation. Consumer backlash against plastic pollution could shift purchasing, but was blunted by lack of affordable alternatives and deeply embedded convenience culture. Biological degradation technologies potentially created negative feedback by reducing accumulated pollution, but slow degradation rates and limited deployment meant effects were minimal compared to ongoing pollution inputs. This system's view suggested that effective interventions required identifying and strengthening negative feedback loops while disrupting positive loops driving production. Production caps directly interrupted the positive feedback loop of increasing scale, reducing costs. Extended producer responsibility shifted economic incentives, making pollution costly rather than externalized. Design standards requiring recyclability or biodegradability created market pressure, rewarding sustainable innovation. These policy interventions aim to restructure the system itself rather than merely treating symptoms. The system's perspective also highlighted unintended consequences and rebound effects. Rebound effects occurred when efficiency improvements enabled increased consumption that offset intended benefits. For example, if biological degradation made plastic pollution less persistent, this might reduce pressure to minimize plastic use, potentially increasing production and net pollution despite treatment. Careful system design was needed to prevent such perverse outcomes. Cross-sectoral connections meant that plastic pollution could not be addressed in isolation from other environmental challenges. Plastic production consumed fossil fuels, linking it to climate change. Agricultural runoff patterns influenced microplastic transport in watersheds. Ocean plastic interacted with marine ecosystems, affecting biological carbon pumps relevant to climate regulation. Air quality in communities near plastic manufacturing facilities reflected environmental justice dimensions. Addressing plastic required considering these multiple connections and seeking co-benefits, interventions that simultaneously advanced multiple objectives. Cities implementing systems approaches to air and water quality demonstrated this integrated thinking. Rather than treating air pollution, water contamination, and climate adaptation as separate problems requiring separate solutions, systems-oriented cities identified shared drivers and invested in interventions delivering multiple benefits. Green infrastructure, including bioswales, addressed stormwater management, reduced urban heat islands, improved air quality through vegetation, and created recreational spaces. Similarly, plastic pollution interventions could be designed for co-benefits, bioswales capturing plastic debris while managing runoff. Wastewater treatment, removing microplastics while reducing nutrient pollution. Behavioral campaigns, reducing plastic while promoting overall sustainable consumption. Systems thinking emphasized that effective solutions required multi-stakeholder collaboration and fostering partnerships across public, private, and community sectors. Environmental problems spanned organizational and jurisdictional boundaries. Municipal governments controlled waste management but could not dictate packaging decisions made by corporations operating globally. Corporations designed products but depended on consumers purchasing them and public infrastructure collecting and recycling them. Communities experienced pollution impacts but often lacked power to regulate polluting industries. Addressing plastic required coordinated action across these actors. The Global Plastics Treaty negotiations represented an attempt at systems-level international coordination. The treaty framework aimed to address plastics' full life cycle from production through design, use, and disposal. Provisions under negotiation included production targets limiting virgin plastic manufacturing, design standards promoting recyclability and durability, waste management requirements ensuring collection and processing, and financing mechanisms supporting technology transfer and infrastructure development, particularly in developing nations. This comprehensive approach reflected systems thinking principles that isolated interventions at single points in the plastic life cycle would prove insufficient. However, the treaty negotiations also illustrated how political and economic systems resisted change even when systemic problems were recognized. Industry lobbying aimed to fragment the comprehensive systems approach, accepting waste management provisions while blocking production limits. Petrostate governments aligned with industry interests, using procedural tactics to delay and weaken treaty language. The resulting political struggle was not primarily about technical questions of what would work, but about power, whose interests would prevail in redesigning systems that generated enormous profits for some while imposing costs on many. Complexity science offered additional insights beyond standard systems thinking. Complex adaptive systems, which included ecosystems, economies, and societies, exhibited properties that made intervention outcomes difficult to predict. Small changes could sometimes trigger large system transformations through cascade effects, while large interventions might produce minimal change if systems were resilient to perturbation. Tipping points represented thresholds beyond which systems rapidly transitioned to qualitatively different states. Could biological plastic degradation deployment trigger tipping points toward rapid pollution reduction? The testbed experience suggested not at current scales and performance levels. Degradation rates were too slow and deployment too limited to substantially reduce plastic burdens. But what if technological breakthroughs dramatically accelerated degradation, or what if deployment scaled 100-fold? Complex systems theory suggested that answer was fundamentally uncertain, dependent on myriad interactions, impossible to fully model. Conversely, might plastic pollution itself reach tipping points triggering ecological collapse? Research on microplastic impacts on ocean biological pumps raised this concern. If microplastic interference with phytoplankton and zooplankton populations substantially reduced carbon sequestration, this could create positive feedback loops accelerating climate change, which in turn disrupted ecosystems further. potentially cascading toward major regime shifts in ocean ecology. Again, genuine uncertainty precluded definitive predictions, but the possibility warranted serious precaution. Adaptive management offered strategies for intervening in complex systems despite uncertainty. Rather than requiring complete understanding before acting, adaptive management emphasized learning while doing, implementing interventions at scales enabling monitoring and adjustment, tracking outcomes systematically, and modifying approaches based on observed results. The testbed strategy exemplified adaptive management, deploying biological degradation experimentally, monitoring comprehensively, and using results to inform scaling decisions. However, adaptive management faced limitations when interventions were potentially irreversible. Releasing genetically modified organisms into environments where they might persist and spread created risks that could not be easily undone if problems emerged. This was where precautionary principles clashed with adaptive learning. Strict precaution argued against any action where severe harm was possible, even if improbable. Adaptive approaches accepted some risk as necessary for advancing knowledge and developing solutions. Navigating this tension required sophisticated governance balancing learning against precaution. Systems thinking also illuminated why past environmental governance often failed to prevent crises. Governance structures were typically fragmented into sectors, environmental agencies, agricultural departments, energy ministries, health authorities that addressed issues within narrow jurisdictions. This fragmentation meant that systemic problems crossing boundaries fell into governance gaps. Plastic pollution involved petroleum extraction, chemical manufacturing, product design, retail distribution, consumer behavior, waste collection, and environmental contamination, each regulated by different agencies or not regulated at all. Integrated governance structures were needed to match the systemic nature of plastic pollution. The Global Treaty Framework attempted this integration at international scale. National plastic action plans could integrate across domestic agencies. Regional authorities could coordinate across municipal boundaries, but creating integrated governance required overcoming entrenched institutional structures, professional identities, and political interests vested in existing fragmented arrangements. The human dimension remained central to systems perspectives. Complex systems involving humans were shaped by culture, values, power, and meaning-making, not just material and energy flows. Changes in worldviews, how societies understood humanity's relationship to nature and conceptualized concepts like waste and value, could be as important as technological or policy changes. Environmental movements had worked for decades to shift cultural narratives from viewing nature as resource for unlimited exploitation toward recognizing ecological limits and interdependence. Systems thinking about plastic pollution ultimately demanded humility about prediction and control. The systems were too complex, involved too many actors and interactions, and exhibited too much emergence and nonlinearity to permit confident forecasting. Biological plastic degradation might contribute to addressing pollution or might prove marginal. Global treaty negotiations might catalyze transformation or collapse into symbolic gestures. Consumer behavior might shift rapidly toward sustainability or remain locked in convenience culture. Multiple possible futures existed, and which would materialize depended on countless decisions and chance events unfolding over coming decades. What systems thinking could provide was not certainty, but rather frameworks for navigating complexity more intelligently. By recognizing interconnections, feedback loops, unintended consequences, and emergence, systems perspectives enabled more sophisticated intervention design. By acknowledging uncertainty and promoting adaptive learning, systems approaches avoided rigid plans that would inevitably fail when confronting messy reality. By emphasizing cross-sectoral collaboration and co-benefits, systems thinking mobilized broader coalitions and generated multiple rationales for action. The plastic pollution crisis would ultimately be addressed, if at all, not through single solutions, but through systemic transformation involving technology, policy, economics, culture, and collective human choices. Chapter 27, RESONS 4 Hope. The data from 5 years of biological plastic degradation research could support either optimism or pessimism, depending on what one chose to emphasize. Bacteria could degrade synthetic polymers, a discovery that would have seemed fantastic decades earlier when plastics were assumed permanent. But degradation rates were measured in months and years, painfully slow against production counted in hundreds of millions of tons annually. Techniques existed for treating plastic pollution, yet deployment remained limited, and technology alone could not address systemic drivers of the crisis. Standing at this uncertain juncture, neither triumphant optimism nor despairing pessimism seemed appropriate. What was needed was hope, not naive expectation that problems would resolve easily, but rather informed commitment to continue working toward better futures despite uncertainty and difficulty. Environmental psychologists studying climate action had identified hope as a crucial psychological resource enabling sustained engagement. Hope increased perceptions of collective efficacy, the belief that coordinated group action could achieve meaningful change, which in turn motivated participation in collective action. Research with Italian university students examining climate protest participation documented these dynamics. The study found that hope led to increased collective self-efficacy, which then predicted greater likelihood of participating in climate strikes and protests. This mediation pathway was significant. Hope alone was insufficient if it did not translate to belief in collective power. But when people combined hope with perception that their contribution enhanced collective efficacy in achieving desired outcomes, they were mobilized to participate. Applied to plastic pollution, similar mechanisms appeared relevant. Hope that plastic pollution could be reduced through whatever combination of technology, policy, behavior change, and social movement pressure was necessary psychological foundation. This hope then needed to support beliefs in collective efficacy that coordinated action across researchers, policymakers, businesses, and citizens could achieve meaningful impact. Those beliefs in turn motivated specific actions like participating in policy advocacy, supporting businesses with sustainable practices, reducing personal plastic consumption, or contributing to cleanup efforts. Importantly, hope oriented toward solutions rather than dwelling on problems proved most effective. Research by Ojala and colleagues found that among young people, hope specifically directed toward climate change solutions served as significant precursor to enhanced perceptions of collective efficacy. This solution-focused hope was realistic rather than Pollyannish, acknowledging problems while maintaining confidence that pathways forward existed. The biological degradation research exemplified solution-focused hope, confronting plastic pollution's scale and persistence while demonstrating that tools for addressing it could be developed. What grounds for hope existed as the third decade of the 21st century progressed? Several developments suggested positive trajectories. The Global Plastics Treaty negotiations, despite frustrations and setbacks, continued advancing toward comprehensive international framework. Over 175 countries had committed to ending plastic pollution through legally binding instruments, an unprecedented level of international consensus. Even if final treaty text fell short of ambitions, the process itself elevated plastic pollution as global priority, requiring coordinated action. National and subnational policies were proliferating. Single-use plastic bans, extended producer responsibility schemes, recycled content mandates, and other regulations were being implemented across diverse jurisdictions. While each individual policy had limitations, the aggregate effect was growing regulatory pressure on plastic production and use. This created business certainty that sustainability requirements would strengthen over time, incentivizing corporate investment in alternatives and circular economy infrastructure. Innovation in materials science offered alternatives to fossil fuel-based plastics. Bioplastics derived from renewable resources like corn, sugarcane, and agricultural waste were advancing in performance while reducing carbon footprints. Materials such as polylactic acid, polyhydroxyalkanoates, and starch blends could be composted or biodegraded under appropriate conditions. Market forecasts predicted increasing acceptance, higher production capacity, and growing market share for bioplastics in coming years. Applications were expanding from niche products to mainstream packaging, agriculture, automotive, and consumer goods. However, bioplastics faced their own sustainability questions. Using agricultural land and crops for plastic production competed with food production. Some bioplastics required industrial composting facilities that were not widely available, meaning they ended up in landfills where biodegradation was minimal. Life cycle assessments showed that bioplastics' environmental benefits varied considerably depending on feedstocks, production processes, and end-of-life management. Bioplastics were not magic solutions, but components of more complex materials transitions. Technological innovation continued in biological degradation itself. Machine learning was accelerating enzyme engineering, potentially delivering variants with dramatically improved activity, stability, and substrate range. Metagenomic mining of environmental samples from polluted sites worldwide was identifying novel organisms and enzymes with plastic-degrading capabilities. Synthetic biology tools enabled constructing organisms and pathways impossible in nature, potentially creating superdegraders or designer consortia. While none of these advances guaranteed breakthroughs, they expanded the possibility space for what biological approaches might achieve. Social movements were growing in scale and sophistication. Youth climate activists had mobilized millions globally, many linking climate and plastic crises as symptoms of unsustainable economic systems requiring transformation. Organizations advocating for Break Free from Plastic Coalition united thousands of groups worldwide working to reduce production, promote reuse, and hold corporations accountable. Indigenous groups and frontline communities most impacted by pollution were increasingly vocal in demanding justice and participation in decisions. This expanding social mobilization created political pressure that could not be easily ignored. Corporate commitments, while often falling short, indicated that business leaders recognized sustainability was becoming competitive necessity. Major brands faced consumer expectations, investor pressure, and regulatory requirements pushing them towards circular economy models. Some companies were genuinely innovating with refillable packaging systems, plastic-free alternatives, and recycled content integration. Others engaged primarily in greenwashing, but even performative sustainability suggested that markets were rewarding environmental positioning. Over time, competitive dynamics might favor companies making authentic progress. Academic research communities had expanded dramatically to address plastic pollution from every conceivable angle. Thousands of researchers worldwide were studying plastic chemistry, environmental fate and transport, ecological impacts, degradation mechanisms, health effects, social dimensions, policy effectiveness, and technological innovations. This generated accelerating knowledge accumulation that informed better interventions. Open science practices, including data sharing and collaborative networks, enabled researchers to build on each other's work rapidly. Success stories from particular locations demonstrated that meaningful progress was possible. Rwanda's comprehensive plastic bag ban, enforced since 2008, had dramatically reduced plastic litter and become model for other countries. The European Union's Single-Use Plastics Directive was driving significant reductions in consumption of targeted items across member states. Cities like San Francisco and Seoul had achieved recycling rates exceeding 60% through comprehensive programs. These examples showed that well-designed policies vigorously implemented could work. Perhaps most fundamentally, awareness of plastic pollution had penetrated public consciousness globally. Documentaries showing ocean plastic gyres and wildlife suffering from ingestion reached hundreds of millions of viewers. News coverage of microplastics in human brains and blood generated widespread alarm. School curricula incorporated plastic pollution education. This consciousness was prerequisite for collective action, problems that remained invisible could not be addressed. The fact that plastic pollution was now highly visible, politically salient, and subject of international negotiations represented progress from decades when it was overlooked. Yet hope had to be tempered with realism about challenges. Plastic production continued growing, not declining. Industry lobbying remained a powerful force blocking regulatory action. Economic systems incentivized disposability and consumption. Technological solutions, including biological degradation, faced limitations that meant they could only be components of broader strategies. Environmental inequities meant pollution harms fell disproportionately on marginalized communities with least power to protect themselves. Climate change was simultaneously intensifying, creating interconnected crises that complicated each other. Grounded hope, the form most useful for sustaining engagement, acknowledged these difficulties while refusing to accept them as permanent. It recognized that addressing plastic pollution would require decades of sustained effort across multiple domains: technology development, policy reform, economic restructuring, cultural change, social movement pressure. It accepted that setbacks were inevitable, that negotiations would frustrate, that technologies would disappoint, that policies would be weakened. But it maintained commitment to continuing the work because the alternative, accepting accelerating pollution and its consequences, was unacceptable. The biological plastic degradation research community, having spent 5 years documenting both achievements and limitations, exemplified this grounded hope. They had demonstrated that bacteria could degrade synthetic polymers, expanding human capabilities for addressing pollution. They had identified challenges that constrained what biology alone could achieve, informing realistic expectations. They had developed governance frameworks for managing ecological risks, enabling responsible deployment. They had built international networks for knowledge sharing and technology transfer. They had engaged publics and policymakers, contributing to informed decision-making. Looking ahead, they committed to continuing research improving enzymes and organisms, expanding understanding of environmental degradation processes, optimizing deployment systems, and monitoring long-term outcomes. But they also recognized their work as contribution to much larger collective endeavor requiring participation from countless others. Biological degradation would not solve plastic pollution alone. Combined with production reduction, improved product design, enhanced waste management, policy reform, corporate accountability, behavior change, and social movement pressure, it could be a meaningful piece of comprehensive response. The reasons for hope were not guarantees of success. Humanity might still fail to address plastic pollution adequately, allowing continued accumulation that degraded ecosystems and harmed health for generations. But failure was not predetermined. The tools, knowledge, and social mobilization existed to change course if collective will could be summoned. Hope was the psychological resource that could help summon that will, sustaining belief that action mattered and that different futures were possible. In that sense, hope was not passive optimism, but active commitment to continue working toward better outcomes despite uncertainties, creating the future through persistent collective effort rather than waiting to see what future would arrive on its own. The plastic pollution crisis existed within broader context of humanity transgressing multiple planetary boundaries, the biophysical thresholds defining safe operating space for civilization. The planetary boundaries framework identified 9 Earth system processes critical for maintaining conditions that enabled human development over the past 10,000 years. These included climate change, biosphere integrity, land system change, freshwater use, biogeochemical flows, ocean acidification, atmospheric aerosol loading, stratospheric ozone depletion, and introduction of novel entities. Plastic pollution fell primarily under the novel entities boundary, referring to synthetic chemicals and materials with potential for unwanted geophysical or biological effects. Recent assessments concluded that this boundary was transgressed, with chemical pollution, including plastics, exceeding the planet's capacity to assimilate these substances without fundamental system disruption. Microplastics and nanoplastics had spread to every ecosystem from Arctic ice to deep ocean sediments, to remote mountain lakes and even human tissues. But plastics' impacts extended beyond the novel entity's boundary to affect other planetary systems. Climate change was directly linked through plastic production's greenhouse gas emissions and waste incineration, releasing carbon dioxide. Ocean acidification was potentially exacerbated by plastic debris interfering with marine organisms' calcium carbonate shell formation. Biosphere integrity suffered as plastic ingestion and entanglement harmed wildlife populations across terrestrial and marine environments. The interconnections meant addressing plastic required considering its role in multiple boundary transgressions. The Safe and Just Operating Space framework built upon planetary boundaries by integrating social justice and human well-being dimensions. While planetary boundaries defined upper limits for resource extraction and environmental pressure, social foundations defined lower limits, ensuring all people could meet basic needs including food, water, health, education, and political voice. The space between social foundations and planetary boundaries represented the safe and just zone where humanity should strive to operate. Currently, humanity simultaneously transgressed planetary boundaries through environmental overshoot and fell short of social foundations through deprivation affecting billions. Plastic pollution exemplified this paradox. Overproduction and waste generation breached novel entities' boundaries, while production facilities and waste dumps concentrated environmental harms on marginalized communities lacking voice in decisions affecting them. Addressing plastic required simultaneously reducing environmental pressure and ensuring justice. Intergenerational justice added temporal dimension to planetary boundaries thinking. The framework emphasized that future generations possessed inherent rights to healthy environment, clean resources, and stable climate. Present generations bore ethical responsibility to act as environmental stewards, preserving conditions enabling future well-being. The principle of intergenerational equity demanded that environmental burdens and benefits be distributed fairly across generations, avoiding transfer of problems to those not yet born. Plastic pollution violated intergenerational equity through multiple pathways. Persistent polymers accumulating in environments would burden future generations with contamination they did not create. Chemical additives in plastic leaching into ecosystems could affect human and ecological health for decades or centuries. Greenhouse gas emissions from plastic production contributed to climate change, whose worst impacts would fall on future generations. These impacts occurred because present generations prioritized short-term convenience over long-term consequences. Acknowledging obligations to future generations meant adopting precautionary approaches when facing potentially irreversible harms. Releasing engineered organisms capable of spreading and evolving created risks, extending far into the future. Even if immediate impacts appeared manageable, long-term consequences over decades or centuries remained deeply uncertain. Intergenerational responsibility demanded extreme caution when interventions might alter planetary systems in ways affecting generations thousands of years hence. Yet complete precaution could itself violate intergenerational obligations if it meant accepting continued plastic accumulation. Future generations would inherit not just risks from biological remediation, but certain harms from persistent pollution if present generations failed to act. Navigating this tension required weighing different risks and uncertainties, making difficult choices without full information. Vision 2050 initiatives by business coalitions and sustainability organizations attempted to articulate positive long-term futures within planetary boundaries. These visions described worlds where 9 billion people lived well without transgressing ecological limits. Achieving this required wholesale transformation of energy systems toward net zero carbon, materials flows becoming circular, with waste eliminated, food production sustainable and equitable, providing healthy diets for all, and living spaces thriving in harmony with nature. For materials and waste specifically, Vision 2050 pathways called for material collection and recovery improving exponentially, flow of waste into environments ending with nature restored, consumption becoming circular and regenerative, and technological advances deployed responsibly driving efficiency and transparency. Applied to plastic, this translated to production aligned with actual needs rather than perpetual growth, designed for durability and recyclability, comprehensive collection and reuse systems, and biological or chemical degradation, treating materials genuinely at end of useful life. Zero waste movements provided more radical vision, eliminating waste concept entirely. The Zero Waste Masterplan outlined how communities could transition from linear disposal systems to circular regenerative economies. Strategies included preventing waste generation through better design, extending product lifetimes through repair and reuse, composting organic materials, recovering resources through recycling, and only as final resort treating small residual fractions. The goal was not merely reducing waste, but creating resilient local economies that replenished rather than depleted planetary resources. These ambitious visions faced formidable obstacles, including entrenched economic interests profiting from disposability, political systems responding to short-term incentives rather than long-term sustainability, and cultural norms glorifying consumption. Yet the visions served essential functions, articulating alternatives to current trajectories, mobilizing action around shared aspirations, and providing frameworks for coordinating across sectors and scales. Paradigm shifts in worldviews represented perhaps the deepest level of transformation required. The dominant social paradigm viewing nature as resources for unlimited human exploitation had driven both material prosperity and environmental degradation, characterizing the Anthropocene. The new ecological paradigm challenged this anthropocentric view, recognizing humans as one species within an interconnected web of life subject to ecological limits. This shift from human domination to interdependence implied profound changes in values, ethics, and behavior. Rather than maximizing extraction and consumption, societies would prioritize sufficiency and stewardship. Rather than externalizing environmental costs, economies would internalize them through pricing and regulation. Rather than viewing waste as inevitable byproduct, systems would eliminate waste through circular design. The anthropocentric paradigm shift did not mean diminishing humanity's significance, but redefining our place within larger ecological context. It meant moving from domination toward mutual respect and reciprocity with natural systems. This shift required recognizing intrinsic value of all life beyond instrumental value to humans, acknowledging ecological limits as real constraints, not obstacles to overcome, and accepting responsibility for planetary stewardship given human power to affect Earth systems. Evidence suggested such paradigm shifts were already underway in segments of society. Environmental movements had worked for decades propagating ecological worldviews. Youth activists demanded systemic change rather than incremental reform. Indigenous communities maintained and shared traditional ecological knowledge, embodying sustainable relationships with nature. Academic disciplines from ecology to economics increasingly incorporated systems thinking and planetary boundaries perspectives. But paradigm shifts occurred slowly and unevenly, often requiring crises to catalyze broader adoption. Climate change impacts, biodiversity collapse, and plastic pollution visible in every environment might constitute such catalyzing crises. Whether these would drive transformation toward ecological paradigm or trigger reactionary reinforcement of anthropocentric values remain to be seen. For biological plastic degradation specifically, operating within planetary boundaries framework meant several imperatives. Technologies should minimize additional environmental burdens through energy efficiency, avoided toxic byproducts, and genetic containment, preventing unwanted organism spread. Deployment should prioritize treating existing pollution rather than enabling continued production. Integration with broader circular economy transformation ensured biological degradation, complemented rather than substituted for production reduction and waste prevention. Most fundamentally, any technological intervention must be evaluated against contribution to or detraction from safe and just operating space. Did it help bring humanity within planetary boundaries while meeting social foundations? Did it honor intergenerational obligations by reducing long-term environmental burdens? Did it embody paradigm shift toward ecological thinking or perpetuate technological optimism, avoiding systemic change? These questions provided ultimate criteria for judging whether biological plastic degradation represented genuine progress toward sustainability or merely another technofix, delaying necessary transformation. Chapter 30, The Road Ahead. The final presentation at the 5-year symposium was delivered not by a scientist, but by Maya Torres, a 23-year-old environmental activist who had participated in the Urban Testbeds Community Advisory Board as undergraduate student. She opened with a simple question that cut through days of technical discussion. What kind of ancestors do we want to be? The question reframed the entire plastic degradation enterprise, shifting focus from technical capabilities to moral responsibilities, from what we could do to what we should do and why. Torres argued that the testbed experiments, for all their scientific value, mattered primarily for what they revealed about humanity's capacity to take responsibility for damage caused by previous technological choices. Plastic pollution represented the material legacy of a civilization that had prioritized convenience over consequences, short-term profit over long-term well-being, individual benefit over collective thriving. Biological degradation technologies offered opportunities to begin remedying that damage, to clean up messes created over decades of heedless disposal. But technology alone could not make current generations good ancestors. That required fundamental changes in how societies produced, consumed, and disposed of materials. It required confronting power structures that concentrated wealth through environmental exploitation while externalizing costs onto vulnerable communities and future generations. It required cultivating humility about human knowledge and technological capacity, recognizing that complex natural systems often defied attempts at control and optimization. The question of what kind of ancestors to be connected directly to intergenerational stewardship principles. Every generation inherited a world shaped by predecessors' choices and would bequeath a world shaped by its own to successors. The quality of that inheritance, ecological health, resource availability, climate stability, defined relationships across generations. Current generations possessed unprecedented power to affect planetary systems through technology, industrialization, and population, creating correspondingly unprecedented responsibility to exercise that power wisely. Judging wisdom required long-time horizons extending beyond individual lifespans or political cycles. Decisions made today about plastic production, biological remediation deployment, or circular economy investments would echo through decades and centuries. Truly sustainable choices prioritized outcomes across multiple generations over maximizing present benefits. This temporal extension of moral consideration challenged ingrained human tendencies toward temporal discounting, valuing immediate gratification over distant consequences. Educational systems needed to cultivate long-term thinking and intergenerational consciousness. Environmental curricula should emphasize connections between present actions and future conditions. Case studies of historical societies whose short-sightedness led to collapse, Easter Island, Norse Greenland, Classic Maya, could illustrate consequences of exceeding local carrying capacities. Conversely, examples of cultures maintaining sustainability across centuries through traditional ecological knowledge could demonstrate that long-term stewardship was achievable. Youth engagement in environmental decision-making provided institutional mechanisms for representing future generation interests. Since young people would inherit consequences of today's choices, their voices deserved weight in shaping those choices. The urban testbed's inclusion of student representatives on governance boards exemplified this principle. Expanding such representation to national and international policy forums could help counter short-term political incentives. The role of hope in sustaining long-term collective action remained essential. The magnitude of plastic pollution and broader environmental crises could induce paralysis through overwhelming despair. Hope provided psychological foundation for continued engagement despite difficulties and setbacks. But this hope needed to be grounded in realistic assessment of challenges rather than naive optimism that problems would resolve easily. Research on climate activism identified that hope functioned most effectively when linked to collective efficacy, the belief that coordinated group action could achieve meaningful change. Individual consumer choices, while valuable for consciousness raising, were insufficient to drive systemic transformation. Collective action through social movements, political organizing, and institution building could alter power structures and policy frameworks in ways individual actions could not. The proliferation of climate strikes, environmental protests, and youth activism demonstrated growing collective mobilization. Movements like Fridays for Future had engaged millions globally, creating political pressure governments could not ignore. Similar mobilization, specifically targeting plastic pollution through initiatives like Break Free from Plastic, was building. These movements provided both practical force for change and sources of hope, sustaining participants through long struggles. The road ahead for biological plastic degradation wound through terrain both technical and social. Technical development would continue improving enzymes, optimizing organisms, and refining deployment systems. Second and third generation projects would address limitations identified in test beds, scaling up where successful and pivoting where necessary. Academic research would deepen understanding of environmental degradation processes, ecological impacts, and governance frameworks. But technical progress alone was insufficient. Social and political dimensions required equal attention. Policy frameworks needed to create enabling conditions through regulations incentivizing circular economy, extended producer responsibility schemes funding waste management, and international cooperation addressing transboundary pollution. Economic structures needed transformation toward valuing sustainability over growth, internalizing environmental costs, and distributing benefits equitably. Cultural change was perhaps most fundamental, yet most difficult. Societies needed to shift from disposability culture toward durability, from individual consumption toward collective well-being, from anthropocentric domination toward ecological partnership. These changes could not be engineered or legislated, but emerged through countless daily choices, conversations, and cultural productions shaping values and norms. Education, art, and media played vital roles in cultural transformation. Documentaries revealing plastic pollution's extent reached mass audiences and shifted public consciousness. Literature and film exploring human nature, relationships cultivated empathy and imagination. School curricula teaching systems, thinking, and ecological principles formed worldviews of rising generations. These cultural interventions complemented technological and policy changes, creating conditions where sustainability became common sense rather than radical proposal. The Global Plastics Treaty negotiations represented critical juncture where technical knowledge, policy frameworks, economic interests, and social movements converged. Treaty outcomes would shape plastic production, design, management, and remediation for decades. The negotiations tested whether international cooperation could transcend narrow interests to address collective challenges. Success would demonstrate global governance capacity. Failure would indicate continued primacy of national and corporate interests over planetary well-being. Regardless of treaty outcomes, work would continue across multiple scales and domains. Researchers would advance scientific understanding and technological capabilities. Municipalities would implement local interventions from bioswales to reuse systems. Companies would innovate business models and materials. Activists would organize movements demanding justice and sustainability. Individuals would make daily choices aligned with values. These dispersed efforts might seem inadequate against pollution scale, but systems thinking suggested that transformation emerged from accumulation of countless small changes that eventually reached tipping points, triggering larger shifts. No one could predict which intervention would prove catalytic or when critical mass would be reached. The imperative was to continue working across all available fronts, trusting that persistent effort would eventually move systems toward sustainability. The 5 years since initial bacterial inoculations in landfills, oceans, and cities had taught that plastic pollution was simultaneously a technical problem requiring innovation and systemic dysfunction requiring transformation. Biological degradation offered valuable tools but not sufficient solutions. Comprehensive responses required integrating technology with policy, economics, culture, and ethics. Most fundamentally, the testbeds demonstrated that addressing environmental problems created by humans demanded that humans accept responsibility, acknowledge harms caused, commit to remedy, and transform practices to prevent recurrence. This was ultimately moral and political work as much as technical. Technologies like biological degradation could assist, but could not substitute for difficult choices about production limits, consumption patterns, and distributional justice. What kind of ancestors would this generation be? The question remained open, answer determined by choices made and actions taken in years ahead. If current generations reduced plastic production, built circular economy systems, deployed remediation technologies responsibly, and transformed cultural relationship with materials and waste, they might be remembered as generation that turned crisis into opportunity for transformation. If they continued prioritizing short-term convenience over long-term consequences, while hopping technology would clean up messes without requiring systemic change, they would be remembered, if at all, as generation that knew better but failed to act accordingly. The biological plastic degradation research community, having devoted years to developing tools for remediation, understood their work's value and limitations. They had shown that bacteria could degrade synthetic polymers, expanding human capabilities. They had identified constraints preventing technology alone from solving pollution. They had built governance frameworks for responsible deployment. They had engaged publics and policymakers in informed dialogue. Looking forward, they committed to continuing research while advocating for comprehensive strategies. Better enzymes and organisms would be engineered, but primarily to complement rather than replace source reduction. Deployment would expand where appropriate, but always within frameworks prioritizing safety and justice. Knowledge would be shared internationally, supporting technology transfer and capacity building. But they recognized that their contribution, however valuable, represented a small part of much larger collective endeavor. Ending plastic pollution required participation from all sectors of society, from producers to consumers, policymakers to activists, scientists to artists. It required working simultaneously on immediate interventions and long-term transformation, technical fixes and systemic change, local actions and global coordination. The road ahead was long and uncertain. Success was not guaranteed, and setbacks were inevitable. But giving up was not an option for those who accepted intergenerational responsibility. The work would continue in laboratories and landfills, policy forums and protest marches, classrooms and communities, wherever people gathered committed to creating sustainable future worthy of bequeathing to generations yet to come. The bacteria would keep degrading plastic, slowly and incompletely, while humanity grappled with the harder work of transforming itself.